all. Once again, welcome back to the workshop on Advanced Drug Design and Medicinal Chemistry 2022. Today, once again, we have good fortune that we have Dr. Pankaj Soni from Pfizer with us. He is a senior research advisor in the Bioprocess Development Group. He has done PhD from Naipur Mohali, then he was a Ramanujan Fellow and received Innovative Young Biotechnologist Award from the Government of India. Then he was a faculty at IMTech Chandigarh for some time, then worked at Coca-Cola and then moved to Pfizer, where he is currently working for the past 7 years or more. And he leads the protein engineering group at Pfizer. He has also done postdoc from Los Alamos National Laboratory and Max Planck, Germany. His research interests are tailoring enzymatic properties by protein engineering and directed evolution to evolve novel, improved biocatalysts for industrial applications. And therefore, his expertise are in protein design, directed evolution, biocatalysis, and biofuels. Today, he will talk about smart protein engineering for the synthesis of drugs and drug intermediates. Over to you, sir. Please begin.
Thank you so much, Vaibhav, for your kind words. Like I proposed the title Smart Protein Engineering for Synthesis of Drug and Drug Intermediates, but I thought maybe I'll add some more to it. So I want to talk about not just pharmaceuticals, but also about nutraceuticals and biofuels. And so during my talk, I'll give you a brief flavor of like what's biocatalysis, what was the classical approach of doing that thing, and then what's the modern enzyme engineering way of approaching this problem. And then in protein engineering, I'll talk about the different methodologies like the random mutagenesis, rational knowledge-guided mutagenesis, as well as the semi-rational protein engineering. Bulk of my talk would be on the semi-rational protein engineering, and I'll give you few examples of how using semi-rational protein engineering, there have been a lot of commercial success in pharmaceuticals, in nutraceuticals, as well as in biofuels. So let's start with catalysis. I think for this audience, I don't need to introduce what is catalysis. We all know that like a substrate is converted to product by a catalyst, which depending on the nature of the catalyst could be a metal catalysis or heterocatalysis or organocatalysis, or if driven by light, it could be photocatalysis. But here we'll talk majorly about the biocatalysis. That is the substrate to product conversion by enzyme or microorganism whole cells or immobilized cells. So what actually is biocatalysis? Biocatalysis is when reactants are converted to products by enzymes. Those enzymes can be in different forms. They can be in the microorganism, or it can be in a lysate or purified enzyme. And so for any reaction to happen, like a particular biocatalyst is needed, and this comes from a cycle of isolation and optimization. So like we want to do a reaction and we want to make very specific product out of that substrate, but there is no known enzyme known. So how would we begin this whole process? So it starts with isolation. So we look in nature for enzymes that can do a similar reaction. And then we'll spend a lot of time on screening to find the right enzyme that can do the reaction. And once the enzyme is found, we'll optimize the reaction condition, and then the whole process is done. And if they say the process economics is directly proportional to the time spent on screening. So the more time you spend on screening, a robust biocatalyst is found, the better are the process economics. So when enzymes started replacing chemical catalysts in the processes, there's always this discussion between biochemists as well as the chemists. Both used to feel that one or the other way is better. And there has always been that debate over that. So definitely, like the biochemist or the protein chemist would demonstrate that biocatalysis have a lot of advantages over the chemical synthetic routes. One of the main reasons is enzymes have high degree of chemical precision. Like they have high degree of chemo, regio, and stereo selectivity. An enzyme is very specific to its substrate, It's very specific to where on the substrate the reaction happened, and also the stereoselectivity of the product. And this high precision gives it the big advantage of making simplified processes. Unlike chemistry, where you have to do a lot of blocking, deblocking steps, enzymatic reactions are very simplified reactions. So many a time, like a 10-step chemical synthetic route have been changed to like a 2 or 3-step enzymatic step. And also another big advantage is enzymes operate in mild reaction conditions, like a buffer, normal pH buffer, pH 7 kinds, and then like temperature of 25 or 30 degrees Celsius under aqueous conditions. Unlike chemical catalysts that where they would need many time high pressure or high like presence of organic solvents. So this also was making it very attractive to a lot of process people. But at the same time, because of some of these properties of enzymes, there were few limitations of these biocatalytic processes. Because enzymes are highly selective, so they have very limited substrate scope. On one hand, you have a chemical catalyst like suppose the sodium borohydride. You want to do a reduction. It can do a reduction on a variety of substrates, but at the same time, a ketoreductase would be very picky on what substrate it would reduce. So that is a challenge. Definitely, they work only under a very normal range of pH and temperature, and they lack stability in organic solvents and thermostability. And these both are also very important to processes, because in the real scenario, lot of those substrates are more soluble in organic solvents. If enzyme is not stable in them, how would they function? And similarly, lot of processes might not be working only under a very narrow pH or a temperature condition. So these limitations have always hindered the use of enzymes in the processes. And over years, the biologists have worked on it. Microbiologists have put in a lot of different efforts to design processes that can speed up the isolation of microorganisms with diversity of enzyme properties. And similarly, biochemists have spent a lot of time on studying about the enzymes and solving crystal structures, understanding the mechanisms. And then there are protein engineers who would tweak the properties of enzymes. So I'll briefly cover those in coming slides. So how would we address these limitations that I showed on the last slide? The classical approach used to be you go and isolate new microorganisms from nature, from regions like we anticipate that there will be microorganisms that can produce desired enzymes. So we'll either follow the enrichment and isolation procedures. This was the classical approach. And then a metagenome approach came. I'll talk about these in the next slide. And however, the modern way of doing it is like do enzyme engineering. From the same parent, make changes and so engineer the say existing pool of enzyme and create the properties you desire. And we call it laboratory evolution of enzymes. So let's start with the classical approach of enrichment and isolation. So you want to do a reaction and you don't know where we'll find the enzyme. So the first step to start with is think of what substrate or what product you are using, what kind of reaction it is. So accordingly, like suppose, again, let's go back to alcohols and ketones. So suppose you are looking for alcohol dehydrogenase enzyme. So collect samples from the effluent treatment plants of, say, a brewing industry, or spray chemicals in a small land and collect samples of soil every on a timely basis, and then perform enrichment and isolation in the lab. And just to give you an example of one of our very old work, so in this work what we did was we were looking for an enzyme that could do this reaction. So this ketone needs to be reduced to make S-alcohol, which will be used to make this drug, which is Cymbalta, the antidepressant of Lilly, the blockbuster. So they have multi-billion-dollar sales of this drug. So we developed the first bioprocess of this drug. And just to give you how we started, at that time, there was no enzyme that could do this reaction. So we went by the classical approach. We went to nature, isolated microorganism from the effluent treatment plant samples. We could isolate 46 new organisms and the basis was like on the basis of if they are consuming the substrate, like we were feeding them this substrate, and if we are seeing the product, then among them, 25 were consuming the substrate. Now they are consuming the substrate, but are they making the product we are looking for? So only 13 were making the product. So the rest were degrading the substrate. And among those 13, are we getting the desired selectivity? Because we are looking for this S-selective reduction, and only 3 were. The rest 10 were like doing R-selective. And then among those 3, then you look for which one is the fastest and show highest catalytic efficiency, and that is your biocatalyst of choice. So that's how classically this work used to be done. This whole thing took us more than like 18 months. Now let's look at the other approach to that. So the other approach is the metagenome approach. What happens in this case is from a place of land which you feel has the exposure to the desired substrates or products or the chemicals, you collect the sample, collect the soil sample and do genome extraction. So you isolate the whole DNA of the organism there. And then you chop the pieces of DNA and then stitch them into a plasmid, like bacterial artificial chromosome to create back libraries. So now these back libraries have pieces of DNA, and then you try to find which piece of DNA has the desired activity. Wherever, like feel free to stop me if anyone has questions, but otherwise if like say someone is typing or anything, maybe they can keep themselves muted.
Okay. Suman ji, can you check who is?
Okay,
so
please
carry on.
Yeah, so you create those libraries, and then by activity tests, you try to figure out on selective plates to see which one is showing. Like in this case, there is a substrate and agar plate, and they try to figure out which one is making, say, orange colony, and the one that is having the highest activity is darkest orange. So then that's how you find in which clone you have the desired gene, and then you go forward with that enzyme. So these are the two classical approaches. And again, there's a lot of screening efforts in this, and that takes time. And the modern approach to it is enzyme engineering. These two scientists have big contribution in the field. Frances Arnold, who is at Caltech in US, so she won the 2018 Nobel Prize in Chemistry for her contribution to enzyme engineering. And the other person is Manfred Rietz, who's at the Max Planck Institute in Germany. And again, he big contribution to the field, developed a lot of smart protein engineering tools. I feel fortunate to have worked with him during my first postdoc. And so the approach is, in enzyme engineering, we do a directed evolution of enzyme or protein for desired properties. Like we are looking for the enzyme that can do certain specific reaction. So we'll use a parent enzyme and we'll make changes in that enzyme and do screening or selection to see if we have made that. And how it is done, like we have a parent gene. On that parent gene, we make library by different methods. I'll show you in coming slides. And those library of mutant genes in a test tube, then they are transformed into a microorganism by electroporation or different transformation methods to get different, say, bacterial or yeast or different microbial colonies. Then these colonies are picked in 96 or 384 well plates, and they are tested for activity or selectivity. And so this is one round of evolution. Like you start with a parent and you find the enzyme mutant that is better than the parent. So this is first round. Then this is the parent for second round, and you do this same again. So these are the different rounds of evolution you perform to till you get what you want. And that's where screening and selection helps you a lot. And there are different approaches of enzyme engineering. There are random methods. If you have no clue about your enzyme, you only know the sequence of the gene, nothing else. So then these approaches are very good, like error-prone PCR and DNA shuffling. And on the other hand, if you know a lot about your enzyme, like there's a lot of good crystal structure information of your enzyme with the substrate, with the cofactor if needed. So if you have a lot of good information on the enzyme structure as well as mechanism, then we can do site-directed mutagenesis, make changes in single amino acids and make specific mutants to create an activity. And then an approach is you know something, but you don't know a lot. So then there's a middle ground. You do semi-rational protein engineering. In coming slide, I'll explain them more and give with some examples. Let's start with error-prone PCR. So I hope the audience knows what is PCR. So PCR is polymerase chain reaction, like it's a molecular biology step introduced, like a method introduced by Carey Mullis, who got Nobel Prize for this, and it can be used to create millions of copies of a DNA. Actually, nowadays everyone must be knowing what is PCR because we all must have got done the COVID PCR test, so rapid PCR test. So that's one of the applications of these PCR. In this case, normally PCR would create multiple copies, millions of copies of your piece of DNA with high precision, no mistakes. But here we want to create lot of mutants from our parent. So we want to make the PCR do mistakes. And how we'll do that? We'll use, say, wrong combinations of the DNT piece. We'll add magnesium or manganese different concentrations, or the polymerase that makes mistakes. And then we have some random mistakes happening, and then we have a random library, and we screen that library first on activity. Like in this case, if you get loss of like a bigger circle is the higher activity, and then those mutants are then tested, suppose in this case by HPLC, to find the selectivity. And just to give you another example, there are lots of papers on error-prone PCR. This used to be the classical way input, like one of the first approaches of protein engineering. And there are many papers on error-prone PCR-based protein engineering. This is just one such example. So here in gray is the wild type, is the parent. It is tested on 4 different substrates: palmitate, stearate, laurate, and butyrate. And in this side, we are showing the specific activity. So as you can see, like in each here, like from gray to blue is the first round of evolution. Then green is second round, red is third round. So as you see, like as we move from one round to the next, the activity is increasing for all different substrates. So they could successfully engineer this enzyme without the knowledge of any structure or mechanism, and by doing random mutagenesis and testing thousands of clones and doing 3 rounds of evolution. The other approach is DNA shuffling. So DNA shuffling is like, you have suppose 3 parent gene. One has one property that you like, the other has another property, and third is another different property. And you're looking for the like a mutant that has a combination of these properties. So what happens in this case is, like suppose this has high activity, this has high selectivity, and this has high stability. So you chop these pieces of DNA by an enzyme called DNase, and then these pieces of DNA, then you stitch them together by primerless PCR. They are stitched together in a random orientation. We don't have any control there. Wherever they want to stick, they stick. And then you amplify those, like you make millions of copies of these pieces. And then you test them by activity. Like in this case, they wanted to convert a galactosidase to a fucosidase, and they did 7 rounds of DNA shuffling. They will chop their pieces and then shuffle them and stitch them. And it worked for them. So now let's move to the next approach, rational mutagenesis. What happens in rational mutagenesis is, like, there's a lot of crystal structure data. We precisely know this is the enzyme pocket. When I say enzyme active site or a pocket, I mean the place where the substrate goes and sits, or the cofactor goes and sits, and the reaction happens. So in this case, this is for the cofactor. The cofactor is sitting here, and from the crystal structure data, we precisely know that it is interacting with which amino acid residue of the enzyme. And then in this case, like suppose we know these are the amino acids, so you make changes in that very, like say this threonine 34 is changed to glutamate, arginine 33 to tyrosine, lysine 37 to alanine, so and vice versa. So these specific changes, and this leucine 67 isoleucine, these specific changes are made in protein. So very like few single mutants are made, and then their activity is tested. So it's a very nice study. Like they precisely knew where the pocket is and how the substrate is, the cofactor is sitting in the pocket and what residues it's interacting. And they studied the interactions and 34, 37. And wild type had this. Here they made 2 amino acid change. And see, the target here is this enzyme is NADPH specific. And for like a process, because it's a 2-step process and the second step is NADH dependent, so they thought it makes more sense for both enzymes to be having the NADH specificity. So what they did in this case is, like here they made 2 amino acid change, and you see like in wild type there is no like in light gray is the ratio of activity for NADH to NADPH specificity, and in the darker like gray is the turnover number. And as you see with this mutant, they could increase the activity and also make it slightly specific for NADH. Next one, more NADH specific. The third one, very. So see, they could increase the turnover. They could also change the specificity. So it's a very beautiful example of how, with a good understanding of the structure and mechanism, you can tweak the enzyme properties and change the specificity and activity of an enzyme. But the challenge is, it's not as simple as it looks. Like in routine, like a rational mutagenesis, success is very low because we think we are very smart, but nature is way smarter than us. So that's where semi-rational methods have been very successful. What happened in their cases, that we have a protein structure, and even if we don't have a structure, we create homology model. There are a lot of softwares available, like Webhub is one of the experts. Then so there are a lot of molecular modeling softwares available that a lot of computational biologists used. Using those tools, you create a homology model of that enzyme and then do docking studies. So, and from those substrate enzyme docking studies, you understand how the substrate, where is the pocket, how the substrate is sitting in what orientation, and what residues it's interacting with the enzyme. And from that, you make some hotspot selections of amino acid, which you are going to change. In this case, unlike in site saturation, where we are changing one amino acid with one amino acid, In this case, we change that one amino acid with all 20 amino acids. So we make libraries of mutants. And then we screen, these are the size saturation mutagenesis libraries made by PCR. Then they're transformed and we get the colonies and then screen them. Just to give you some example of our work on this. So this is crystal structure of Candida antarctica lipase B. It's the most used enzyme in a chemical industry, Candida antarctica lipase B. So a lot of chemical processes are with this enzyme. There are a lot of crystal structures available with many different substrates. And so we use that information. Like this is a very typical lipase with a catalytic triad of serine, histidine, aspartate. Good understanding of the mechanism was there. On the basis of that, we identified these amino acids and we grouped them under different sites, A, B, C, D, E. And we were looking to create enzyme mutants that can make both enantiomers of the products. So using this, like this is one of the substrate in this paper, you will find we tried it on more than 20 substrates. So using the same parent, when the parent has almost negligible or almost no activity for say one, we have created enzyme mutants by a couple of rounds of evolution. So we use there's a parent that has an E value of 1 for S selective, and so we did 1, 2, 3, 4. 4 rounds, and we have an enzyme that has an E value of 72. So highly selective and highly active S selective mutant. And similarly, here also, we reached a highly selective R mutant. So from the same parent, by making changes in the pocket, you can create new activities, you can create enantio complementary enzyme mutants, and you can increase the activity by several folds. So this is the kind of work that addresses the challenge which I was showing you initially, that enzyme has the limitation of narrow substrate scope. Definitely, the parent has narrow substrate scope, but by enzyme engineering, we can solve this problem and make it very broad substrate specific and highly active. Now coming to the other challenge that I mentioned, the enzymes are not stable at high temperature or organic solvents. So this is another example from our previous work where like there is this enzyme Bacillus subtilis lipase B. This has moderate stability. Like this was stable at 42 degrees Celsius for like 30 minutes. And so what we did here was like one of my colleagues, he developed an approach called B-FIT, B-factor iterative test. So from the enzyme, you understand which regions have weak spots, like high flexibility. Like the crystal structure data of the enzyme gives you B factor data, and the high B factor mean that spot is very flexible, and a low B factor mean that the spot is very rigid, that amino acid is very rigid. So in this case, from the enzyme crystal structure data basis of B factor, we identified different hotspots that were having high B value. That means they were very flexible. So herein, like we made libraries of these mutants, and this is like T5015 value. Like when exposed to that temperature for 15 minutes, it loses its activity to half. So in 5 rounds of evolution from wild type, 1 round, second, third, fourth, fifth, we got a mutant that has a T5015 value of almost like close to 100, 90, I think 98 point something. So that's the power of enzyme engineering. An enzyme that was moderately stable, having a low T5015 value, could become thermostable by making changes in some of the regions. And from the same enzyme, when we tested for organic solvent stability, because see the logic in stability, whether it's for thermo or for organic, is like in organic solvents, the solvent forces pulls apart the enzyme and denatures it. So in this case also, if we can increase the inherent strength of that enzyme by making changes in the flexible region, we can increase the inherent strength and make it more stable. So these mutants, when tested on different organic solvents, there also we saw a trend that this is far more stable in organic solvent than this. So here we address the other challenge of like with the enzyme, that when people say they are not stable in high temperature or organic solvent, you can do semi-rational protein engineering and make them stable. Now let me give you a couple of commercial examples of this. There are many, like in my career also, like at different places, we have developed processes using enzyme engineering that save over 25 millions per year for the companies. And many other companies or researchers have done that. And here you give you one of the best examples in the field. Like this is work from a company called Codexis in California. Like they are one of pioneers in protein engineering. And this is for a drug sitagliptin. This is an anti-diabetic molecule sold in by brand name Genovia by Merck. And the chemical catalyst process were like this. From this substrate with the rhodium and high pressure and the high solvent, they got a 97% EE. Then came thought of like, okay, how about we do it by enzyme? At that time, like this could be done by an enzyme called transaminase that can do like keto to this amine. But how would we do that? There's no enzyme that can do this, that can accept this kind of substrate and high substrate charge. So the company started with a small substrate and see if they can do this. So they picked a transaminase, tested it on a smaller substrate, and saw the desired activity. Then increased the length of the substrate on the real substrate, and over rounds of evolution, they could and parallelly saw a lot of structure. And so they could engineer this enzyme and develop the commercial process for this molecule and having a EE of 99.95%. So way better than a chemical process and not harsh or not high pressure like that. And this like very eco-friendly. So this is what green chemistry is about, and a lot of companies are moving towards that. So giving you another examples from nutraceuticals. So I think a lot of you must be drinking Coca-Cola and may have seen this kind of bottle, Coca-Cola Life. Like it's a green label on that, and it has stevia sugar. That's a 0-calorie sweetener, and there's no sugar there. And so stevia comes from this sweetener comes from stevia leaves, and those plants you have to grow acres of stevia leaves to get some like few grams of that sugar. So that's like from the commercial viability, there was a need to develop a process for that. So we, like and many other scientists, we collaborated and we put together a process for developing the best stevia sugar. And so parallelly, other researchers developed in yeast, we developed in E. coli. So what happened here is like starting from glucose to steviol, stevioside, and then three glycosylation events happen. And these glycosyl transferases, a lot of protein engineering was done on these to develop this molecule. So this is commercial and have a huge value. This stevia sugar has a market value of $485 billion per year. These are the structures of these enzymes and the different mutations and how to make it work. And we have a patent for that. So more details are there if you need. And then coming to another example, like biofuels. So there have been a lot of push on like different generations of biofuels. The one of them is like cellulosic biofuels. So what happens is like from solar energy, a lot of feedstock is grown, and that feedstock, like say in our case, corn cob or jatopa or all those like plant waste. So that is then boiled in a boiler with to like it's chemical and physical pre-treatment. So what happens there is like there's high acid and a lot of enzymes are added, not different cellulases, so that you release the different sugars from these plants. And then those sugars, then different set of fuel producing microorganisms act on them to make different kinds of biofuels. So when I was at Los Alamos, we were involved in a process to develop and engineer enzymes, improve their activity and stability for the enzyme pre-treatment. So when we look at that, like this is how it looks like. So the cellulose polymer is like that, and different cellulases act on them. There are beta glucosidases, there are cellobiohydrolases, and endoglucanases. So the big need here is the enzyme should be highly stable in high temperature, high acid, and very active. So that's where like you apply those different approaches of protein engineering and create such enzymes. Okay, let me wrap up what I have said over last 30 minutes or so. I hope I could pass on the message that enzymes are robust biocatalysts to carry out any desired reaction with high degree of activity and specificity. And there are a lot of advanced protein engineering methods available that have helped scientists develop, like create pathways for synthesis of pharmaceuticals, nutraceuticals, a lot of fine chemicals, flavors, fragrances, biofuels. So, like in the beginning, I showed you like the chemists think they are smarter and the biologists think they are smarter. But it's the combination, like they have to work together, that will make it work. Like it's a combination of both chemistry and biocatalysis, because there will be steps for the synthesis of the substrate that where the chemistry is better, and then there might be steps where enzyme has a higher specificity and activity and selectivity to give a green sustainable process. So they go hand in hand. So that's all from my side. Thank you everyone for your kind attention. I'll be happy to take any questions.
Thank you very much. That was really a wonderful presentation explaining all the advances in the protein engineering field and how directed evolution and other methods are playing a part. So are there any questions from the audience? Something from the YouTube? So yeah, let me ask myself. So what role do you think the computer aided design or is now going to play in this area, because the computational abilities are improving every day and we are able to generate mutants and do long set of series of simulations. But I can understand that each enzyme will come with its own mechanism of reaction and simulating it. So there cannot be a general framework in which you just plug in the protein and get the answer. But one can you think of areas where a generalized solutions can be built?
Definitely. I see, like, it's a very good question, Vaibhav. Thanks for asking. I see Dr. Bhaitam is on, like, I can see his name on my screen currently. So he's one of the pioneers in the field. Actually, he's the one who introduced us to this subject like 15, 20 years back. So as such, all this, like, especially say in this pharma space, computer aided drug design is very core to it, like whether it's to study the receptor ligand interactions, or even at our level where we are doing the protein metabolic engineering. So lot of this all, like, we develop a lot of algorithms, we work with a lot of computational biologists to help us with that, and like use those tools to see and identify the best orientation for the substrate to sit to make these reactions happen. So definitely they are very integral.
Okay, so yeah, thank you. Anybody would like to add or ask a question? Okay, with that, then I would like to once again thank Dr. Pankaj Soni for coming us, and special thanks because you are coming at an odd time for you. Uh, so thank you once again.
No worries. It's all my pleasure. Thank you so much, Vaibhav, and the organizing committee for giving me this opportunity. I really loved having a chat with you guys. Thank you.
Bye.
Bye.
Okay, so now our next speaker is also already with us, Dr. Suman. Can you hear us?
Yes, I can hear you. Yeah.
Okay. Thanks once again for joining us. And also for him, also it's very odd time for him in the night in the US. So thank you, special thanks for that. So let me introduce Dr. Suman to the audience. He is assistant professor at the School of Pharmacy, Computational Science, University of Texas at El Paso in the US. He has done PhD in chemistry from the University of Texas, El Paso. His research interests are understanding non-covalent interactions between protein ligand complexes, general drug discovery, genomics, and also personalized medicine. He also does a lot of machine learning and AI and uses computational tools and develops computational tools for docking, pharmacophore modeling, MD, and metabolism predictions and QSAR. And currently he is working on specialized software tools for molecular cellular information processing and decision making. And also during the COVID pandemic, he is working with the COVID-19 Drug Discovery Consortium in finding drug molecules for the treatment of COVID-19. And today he is going to talk about accelerating drug discovery using AI and ML algorithms. With this brief introduction, I once again thank Dr. Suman and invite him to start the talk. Please share your screen with us. You are muted. Okay, good. We can see this screen.
Okay, great. Okay, so I assume you're seeing my screen right now. Great, and first of all, I want to thank Dr. Vaibhav Dikshit for this invitation. For us, it's about like 10:20 right now. So it's in inconvenient time, but it's not that bad actually. So I'm really glad that I was able to join and share some of the things that we've been doing in our lab, and also some of the excitement that we have about artificial intelligence and machine learning that can accelerate drug discovery. So artificial intelligence and machine learning. Okay, so why we need that? So drug discovery is a very complex process, and so a drug to bring into the market, typically it's gonna take around 10 years and billions of dollars. So the idea is we wanted to expedite this process using predictive models. So the artificial intelligence and machine learning, it can be applied at various levels. So not just in one level. So in the early stages of drug discovery. So for example, if you look at a target identification, there are several omics data that we can use, and we can predict what are the targets that are novel targets that could be important for a given certain a disease that you are interested in. So it can be very helpful in identifying the pathways, identifying the targets in the very early stages of drug discovery. And we can also use artificial intelligence and machine learning algorithms in the early stage drug discovery to find different molecules, lead molecules, and also in the lead optimization process. And not just that, but also in the clinical trials and preclinical trials, if we can predict things before we do the experiments. So there is a huge level of excitement about this AI/ML in the recent days, and it can be applied in various levels of drug discovery. So with that, I wanted to talk a little bit about like the definitions of what is AI, and you all have heard what is AI, ML, and deep learning. So what is the differences between these? So first of all, if we look at what is an artificial intelligence, simple definition is like, okay, a program that can sense, reason, act, and adapt. So if you look at ourselves, like we have an intelligence, and like, okay, so humans have an intelligence. So we call that as a natural intelligence. So computers usually they don't think, they don't have that ability. But like if we create programs that can think, that can sense, reason, and adapt, so if they can do some of the things that we do, then we call that as an artificial intelligence. So we are developing an artificial intelligence. The next thing is machine learning. So if we look at machine learning is a subset of artificial intelligence. So if you look at machine learning, just the machine learning part, it's actually an algorithm that can predict something. So usually it's a predictive model. So it learns from the data. So usually in machine learning, you need some data for it to learn, and then it will predict. If we develop a model, by using that model, we can predict some of the properties or whatever you're trying to do. And deep learning is actually more of a newer one. So deep learning, again, it is a subset of machine learning. Okay, in the deep learning, rather than a simple models, like we have a different layers. Usually we call this as a neural networks. So it is, again, the deep learning is more powerful because it can, with the increase in data, it can learn more, because a normal traditional algorithms, machine learning algorithms will at certain point will stop learning, whereas deep learning, with the increase in data, they will be able to learn more from the data. So AI, it's not new actually. It's been there for a very, very long time. So if you look at, it's there in 1950s. There was a lot of early excitement, but then afterwards, like the excitement, there wasn't that much excitement. It wasn't took off during that time because at the early stages in the 1950s, like, okay, the algorithms were there, but like we did not have like a lot of data, and we did not have much of computation power. So those two were lacking during that time. But in the recent times, now we have like, okay, very advanced computers and like highly powerful computers, and also we have like an increase in data that we have collected in the last several decades. So there is more increase in data, there is more increase in computation power, coupled with the newer algorithms. Now the AI became more powerful. So that's the excitement that we are seeing, that AI is transforming several fields, and we are interested in drug discovery. So we are trying to adapt AI technology to advance drug discovery research. So if we come back to drug discovery, okay, one of the main thing in drug discovery is like we wanted to predict the activity of a given molecule or a property of a given molecule. So what we look at is like, okay, if we have a set of compounds, if we know the activity of those compounds, we try to construct a structure-activity relationship. So we have a structure, a couple of structures, and if we know their activity, so we try to calculate like, okay, structure to A to its property, we try to relate them. So if you are pharmacy students, like, okay, you study like a lot of the structure activity relationships in your class, So here, like if we wanted to quantify those activity relationships, structure activity relationships, then we call them as QSAR. So this stands for quantitative structure activity relationship. So where you're relating structure to the activity and you're doing it quantitatively. So you're trying to see, like, if we change a molecule, a group on the molecule, what would be the activity change for that particular molecule. So these are QSAR. If we predict a property, then we call it as a QSPR, quantitative structure property relationship. And these QSAR, QSPR, people have been using for a very long time for the activity or property prediction. But now we have more advanced machine learning algorithms. So we are just applying those to fit the data. So you have several molecular structures, and you have a biological response or a property. So we are trying to correlate these two. So here is a simple correlation that we are doing experimental data versus predicted data here. In this case, we are trying to predict this, come up with an equation where we can predict for unknown molecules. So usually the way we do this is like, okay, we take we take the compounds, molecular structures, and extract their features. So features can be different features. It can be it can be a chemical feature or a physical chemical features, or it can be a pharmacophoric features, or it can be a fingerprint features. So we extract the features from the molecules, and then we try to put those relationship to its activity or property that we are interested in. And we correlate these using different machine learning algorithms. So here, like, okay, simple multilinear regression was used for a very long time, but we have these advanced machine learning algorithms where we are using like nonlinear methods to correlate the data. So if we look at machine learning, we can look at machine learning. We can classify machine learning into two different types. One is an unsupervised learning, and the other one is supervised learning. Unsupervised learning, it's basically a clustering. So usually in unsupervised learning, you don't know exactly like, okay, what you're looking for, but you project your data, you cluster them, you put them together, and then you figure it out based on the data. For example, like, okay, if when you cluster the data, then you will get a group of compounds together. Then you know, if you know the properties of a compound in that group, then you can predict the one that is closer to that also has a similar property. So you're predicting based on the cluster. So you're trying to put things together. So that's unsupervised learning, and supervised learning is what we are more interested than unsupervised learning. In supervised learning, this can be divided into two different types. One is a classification, and the other one is regression. So classification. Classification simply means the name itself has the definition. So it just classifies the given set of data. So for example, in drug discovery, we are interested in if the molecule is active or not. So if it will bind to a given target or not. So those are the type of questions that we are asking, or like if the given drug will metabolize by certain enzyme or not, or if the given molecule is a toxic or not. So those are the different classifications that we we are usually interested in drug discovery. So if we have a classification problem, then we use this supervised machine learning to classify it. Again, like, okay, if you look at like, okay, computer science, like, okay, people are like looking at images and classifying various ways, like, okay, if it's a dog versus cat or whatever it is, so human versus other things. So it's a simple classification. And if you look at a regression, this is more continuous. So basically here you're predicting a number rather than you're telling yes or no. In classification, you're basically telling yes or no. In regression, in regression, we're usually looking at at something called we are actually predicting a number. For example, if you are predicting a binding affinity, okay, KD value or a KI value, okay, then that is a regression problem. So, so in classification, again, like here, like, okay, we are looking at gene. Here is an example. So the gene, here we are classifying whether it's a disease gene or a healthy gene. So this is a classification problem, whereas a regression problem, so in this case, like we're looking at if you have this gene, how many days, like, okay, this patient has survived. So here, like, okay, we are looking at a number. We are coming up like with a number, how many days the patient survived. So so that is an example of a regression. And then we have like a machine learning models. Like we have different types of machine learning models. Algorithms. So here, like, okay, so there is a linear, when you're fitting the data. So I mean, linear fitting is a simple fitting, but there are also several non-linear methods that we could use to classify the data. So for example, like, okay, if your data is like this, the linear model will not be a very efficient way to classify the data. So like you need a nonlinear method here to classify the data. So if you look at different, if you look at a machine learning model development process, whenever you have a data, you actually take your data and you split into 3 different types. You want like a training set and a validation set and a test set. So you have a data, so you extract your features and you split into a training set, validation set, and test set. And it is very important to have 3 different sets, which is a training, validation, and test set. So what you do is, like, by using the training set, you actually develop, you model your data and you come up with a machine learning model. But once you have the machine learning, so you wanted to test. Once you have a model, every time you have a model, you test it with your validation set first. And once you validate it, okay, if it looks good, then you just go ahead and test with test set. But usually you do this a couple of times. You train it again, try to see, like, okay, on the validation set a couple of times, and then you, at the end, when you are satisfied, you take that model and use the test set to check the model performance. Okay, why we need validation set and test set, why we need both these sets is, so validation set is informing you your model while it is training, whereas test set, you will use it only at the end. So you do not use this during the training process, because this validation set is kind of also the part of the training set. It's not exactly the training set, but it is helping you in the training process. So that's why you have the validation set. And once you have a model, that's what, like, okay, you use those machine learning models to do your prediction. Like, for example, if you want, before you do that any experiment, if you have a data, if you want to check a compound, whether it's active or not, if it's a classification model, then you extract its features and predict using the developed model, and then you get your response. So this is a typical machine learning process. So here I just wanted to touch a little bit on this graph, knowledge graph mining, and this is picking up like more and more in the recent days. So knowledge graph mining, where, like, okay, you can collect data from various databases and put the data sets together, and that are machine learning ready, and you can predict the models. So this knowledge graph mining is getting more and more popular in the recent days. And also, just to show the power of AI and machine learning, so this was a paper published 2 years ago, where this company called In Silico Medicine, they developed molecules against a target using AI algorithm in 21 days. They developed the models pretty quickly, and then they synthesized in 21 days, and like they tested, and within 46 days they were able to design, synthesis, and test the models all together in the matter of 46 days. So they were able to come up with some of the molecules within several days. So another one of popular AI example is protein structure prediction. Protein structure prediction is very important for drug discovery. If you know the structure of the protein, then you can design the molecules that can really fit into them. So protein structure is very important, not just drug discovery, for also various other things. So for this one, so this is a long-standing problem. Like, okay, people have been using a lot of homology models, but AlphaFold algorithm, which is developed by DeepMind, which is a subsidiary of Google, they come up with this AlphaFold model, which has an now accuracy pretty close to the experimental accuracy, or like very exactly similar to the accuracy of experimental results. So this was one of the major breakthrough in the recent times, very recent times. So there was Cas13 and Cas14. And Cas13, like, they really outperformed compared to the first place. Like compared to the second place, they really outperformed. And actually in Cas14, they did even better compared to AlphaFold 1. So AlphaFold 2, their algorithm did really well. And like here, they employed machine learning. It's not just a machine learning; it's a combination of machine learning and other things. But the machine learning was a critical component of this algorithm, AlphaFold. And also, you can see automating chemical synthesis using artificial intelligence. So this is very important. Like synthesizing molecules is a very important process in drug discovery, and not only just drug discovery, but synthesizing molecules is very important in various other industries as well. So now, like, there are algorithms like Manifold, which can actually do the retrosynthesis and give, like, okay, for any given compound, it can give like a retrosynthetic pathway, and to give like what is the easiest way to synthesize the compounds. And then here, like IBM, they have this robo reaction for chemistry, where like, okay, the AI can automatically synthesizing compounds. Here is an example of Novartis, where like they were using deep learning to classify compounds based on like treating. They have a lot of data where like compounds were treated with the cells, and they were classifying whether the given compound will have an activity towards those given cells or not. So those are, again, like different machine learning algorithms have shown, and like, okay, AI applications. So again, like, let's dive a little bit more deeper into AI and drug discovery. So actually, if you look at drug discovery, it's a multi-parameter optimization task. So it is not just like, okay, that you're just doing one thing. Like, okay, for example, the drug, if it is really potent and if it has a high activity towards the given protein, that is not the whole solution. The thing is, we need to look into whether the drug is it safe, and like, will it pass the cell membrane? Is it soluble? Does it have optimized drug metabolism properties, pharmacokinetic properties, toxicity? So we need to optimize the given compound for all the other things as well. So that is the biggest challenging task here. Like, okay, so it is a multi-parameter optimization task. So what are the ways like we can find drugs that have like optimal properties? Okay, here if you look at like, okay, so you, for example, if you want a drug with all the desired properties, and like, okay, so that is where like we wanted to reach. That's a bullseye. That means like a compound that has all the optimal properties for the target we are looking and have all the desired properties. So if you try to do experiment or like large scale simulations, okay, so we won't be able to reach this. Like, okay, so if we do high throughput screening, we can only screen so many compounds. But if you look at our chemical space, the chemical space is getting bigger and bigger. So right now, like, okay, the drug-like molecules can be about 10 to the 60 compounds. So that's like a huge number of compounds. So we can screen this, we can enumerate the compounds, we can do, say, example, virtual screening like docking. Okay, so by using those techniques, like we can come closer to the properties. So we can have several hits, and like we can look at all the properties of those top hits, and we can get up to pretty close to what we want. But there is an inverse design, like rather than like, okay, just screening and trying to get a compound, hoping that it will have a compound with all the desired properties, we can actually design a compound that have exactly the properties that we want that compound to have. So for this, we can do, this is called as an inverse design. So here, like, okay, in this case, like we are designing inversely, where like, okay, the compound to exactly what we want. So here we can use artificial intelligence algorithms. So we call this as generative chemistry. Because we're generating molecules, so we have different type of algorithms, so variation autoencoders, and like, okay, we have GANs and reinforcement learning. So these are different algorithms that can be applied in optimizing compounds to get a newer compounds that have exact properties, desired properties. So let's dive into some of these algorithms, how they work. So for example, a deep reinforcement learning algorithm for de novo design. So here, this is an algorithm called reinvent. So this algorithm, so first how it works, this algorithm, to know this, okay, first let's look at a chemical structure. So you all know, like, okay, this chemical structure. So if you look at this molecule, that is a two-dimensional structure. So if you want to put, like, okay, if you wanted to feed it to the computer so computer can understand, so what we usually use is a one-dimensional structure rather than a two-dimensional structure. Of course, like you all know, a molecular formula, which is a one-dimensional structure, but it does not have all the information how the atoms are connected. So there is this other representation called SMILES, which some of you might have, if you have a little background in cheminformatics, you might be very familiar with SMILES. So here, like SMILES, the SMILES represent this molecule in this case. So this is like a one-dimensional structure. So this is SMILES. So here is an example of an isomeric SMILE for the same molecule. So when you use isomeric SMILE, it also has a stereochemistry involved in them. And then there is it's not just SMILES, so there is also other few other representations, 1D representation. For example, standard INCHI is one representation, and INCHI key is another representation. And there is a newer representation called selfies. So this is again the newer representation. Selfies is much more suitable for machine learning algorithms, for AI algorithms. So the way these work is, like, for example, like, okay, here is a reinvent algorithm. I just wanted to give a little glimpse for you guys to understand how this algorithm actually works. And we are actually using most of these algorithms we are borrowing from other side of the computer science, for example, like natural language processing. Most of the algorithms here we are seeing is from computer vision or natural language processing. So here, so first, so if you look at SMILES, so this has information, like here, like oxygen is connected to a carbon, and this is connected to a carbon, and so on. So here, whatever these are, it is showing up here. So, so in simple terms, like, okay, the way natural language processing algorithms would work, if an algorithm is there to generate names, Okay, for example, if there is an algorithm to generate names, the way we train the computer, the way this algorithm is trained to give up new names. For example, if we want to generate a new name, so first we need to feed the algorithm with some of the names, and so it knows what are actual names and what ones are not actual names. So so we're using similarly, like, okay, we're actually taking compounds, compound structures, and we are telling the algorithm, okay, these are the real structures and these are like not real structures. So it over the period of time, like every time, like, okay, so the computer randomly generates some compound, or like if you're generating a name, so computer randomly generates some name, and then we have a agent that tells whether it is a compound name or not. So in this case, in this algorithm, this was generated using Campbell data. So if Campbell is one of a very good resource for ligands of small molecules. Usually it has data of several small molecules and their activity towards different proteins and so on. So Campbell is a large cheminformatic database. So it has like several compounds that are like drug-like compounds. So in this algorithm, so all the compounds from the Campbell are taken, and then it is fed to a Smiles generator. So Smiles generator, so like the algorithm first generates different molecules. So each time it generates a molecule, then there is an agent that tells whether it is a good molecule or not, and then you have a classifier built in here that has that will tell whether the molecule has desired properties or not. So each time a molecule is generated, you look at whether it has the desired property or not. If it has a desired property, it will take it. If it doesn't, then it will go through the process again. It will generate a new molecule again. So this is a process where it's an iterative process where a molecule is generated. And it will be checked for its property, and if the algorithm does not generate a molecule with desired property, it will do it again, and so on until you get molecules with the desired properties. Here is another algorithm. Here is another algorithm, which is a generative algorithm. This is using autoencoders. Okay, the way this works is here, for example, here you have a lady. So you're taking a picture of a lady and you're trying to generate the same picture. So the way you do it is like, okay, first you encode and then you decode. So first, how do you encode? So look at the features of this this girl. So here the features of this girl is so she's a female. She has a brown hair. She has a makeup. She doesn't have sunglasses, and so on. So those are her features. So you encode all the features and use those same features and generate same person again. So if you generate the same person again, so that's what, like, okay, you're encoding and you're decoding. So here is a molecule. Okay, you encode all the features and then you're decoding it to get the same molecule. Okay, but we are not interested in generating the same molecule. We wanted to have some change in the molecule. So the change in the molecule and with the desired properties. So you just wanted to modify the properties. So here, what we are doing to do that in encoding and decoding, what you do is, for example, here is a lady. So we wanted to generate a picture of a lady very similar but with the slightly different properties. For example, so here is the information you're encoding, and then just to change something. So here she doesn't have a black hair, she has a brown hair. Let's just change that. Okay, and then you try to generate this the person. Same person with slight modifications. So you have a modification. You have a picture the way you want. Okay, the person, you have the same person, but like you have a with some modified properties. So the properties are modified. So you are using same thing for the compound. So for example, you have a compound that is active towards, for example, COVID. So you want, but it is not soluble, for example. So what you want to do is like you want to encode it and just change the property here in the latent space where you increase its solubility. Okay, and then you decode it. So you get another compound with small change in here, but that will have a property that you're looking for. So here is an algorithm where like you're encoding and decoding, and here is a latent space. In the latent space, you're moving the molecule in such a way that you will get a desired property. So here is a desired property. So you're optimizing the property in the latent space. So this is an example of a generative chemistry here where we are using autoencoders to generate molecules with desired properties. So here is one of the applications. So there are several applications. Okay, our lab. Okay, what we are doing. So we one of the applications that we recently developed and published in Nature Machine Intelligence is a platform to estimate anti-SARS-CoV-2 activities. So here, what we did was like we took the data, and there were 11 different assays for which we developed. Machine learning models where like you can, for any given drug molecule or a drug-like molecule, you can predict its activity, whether it will be active in each of these assays or not. So we have a web portal for this one. Actually, it just got down today. It's under maintenance, but you can check like in a couple of days. It should be up and running. So this is our web portal where like people can submit a smile or just the name of the drug or just by using a PubChem CID. Here is another tool that we developed. It's called LigandNet, where we developed machine learning models for various drug for various targets. So these are some of the tools that we used in doing that. So we developed machine learning models for about 800 targets. So these targets include like, okay, some of them are GPCR, nuclear factor receptors, kinases, enzymes, and so on. So for about 800 human targets, we developed machine learning models. And here are the results of those. So most of our models, like we check using F1 score and ROC-AUC. This goes from 0 to 1, and like most of our models are pretty close to 1. So these are pretty very well optimized and have a predictive power. And here is another web portal where like, okay, people can submit a smile, and that can result in it will run through those 800 different models, and it will give up whether it will be active or inactive for that particular protein. Here is another project. We call it as OpenDMPK, so where we are predicting like drug metabolism and pharmacokinetic properties of different molecules. So in here, so if you look at like, okay, there are different various phase 1 and phase 2 enzymes. So where like, okay, we were looking at whether the drug molecule will be active or inactive for that particular will be a substrate or not for that particular metabolic enzyme. If it's not a substrate, okay, then that's it. But if it's a substrate, we wanted to look at the drug-drug interactions. If it's and also we are interested in sites of metabolism. If it's a substrate, we wanted to look at the sites of metabolism, and then we want to predict the metabolite and also the toxicity. Actually, we collaborated with Dr. Vaibhav Dikshit on this before, where we were actually developing for sulfotransferases and some of these enzymes. So here is an example of sites of metabolism prediction on FMO. So I don't want to get too much into details, but here we have a web portal for these as well. For a given molecule, we're able to predict different metabolic properties. Just wanted to see how am I doing on the time. So Vaibhav, can you just let me know how many more minutes I have?
You have another 15 minutes. It's not that bad.
15 more minutes?
Yes.
Okay, great. So we've been working with the docking algorithms as well, so scoring functions, so where we applied deep learning. As most of you know what docking is, so usually this is a fast calculation where we look at whether the drug will bind to the protein or not, and we estimate the binding affinities if it binds. So basically, so we try to take the drug molecule and put it on the protein and estimate the binding affinities. The idea here is like, okay, so with the drug protein complex, when the drug bind to the protein and form the drug protein complex, usually the energy is released, and we are trying to estimate like how much energy that is released, the free energy. So if you look at a docking program, it usually has two components. The first one is a search algorithm, and the second one is a scoring function. So usually search algorithm, what it does is it takes the drug molecule, and so here is an example of search algorithm where it takes the drug molecule and put it on a protein. So if you look at this here, we have a drug molecule that is put on this protein like at various positions. So, and the next component is the scoring function. So at any given pose, what would be the energy binding affinity? What would be the binding affinity between the drug and protein? So that is calculated using scoring function. So in here, if you see like the drug is moving in various places on this protein, at every protein, like okay, at every position, we calculate the binding affinity. So the binding affinity at one position can be low, at another position can be high. The idea here is like we're trying to find the lowest energy possible conformation. So lowest possible energy conformation. So the lowest possible energy conformation, ideally that would be the natural pose of that ligand. So that is what we are interested in the pose. To design the compounds. So to estimate this binding affinity, there are various scoring functions. So scoring function, again, scoring function is a equation to calculate the binding affinity. So there are different kinds of scoring functions. There are like, okay, DOC is an example, which is a force field scoring function. If you know molecular dynamics, okay, most of these are used in molecular dynamics. These are force fields scoring functions. There is knowledge-based scoring functions. There are empirical scoring functions, and now machine learning scoring functions are proven to be even more accurate. And so because they're able to learn information from data. So we've been applying this machine learning scoring functions. Recently, we've been developing machine learning scoring functions, but in the past, we developed some of these force field-based scoring functions. So here is an example, like, okay, one of the things that we did early on before we applied machine learning was we incorporated something called as a halogen bond. So for example, if you, I'm not sure if you guys are familiar with the halogen bond. Everybody know what is a hydrogen bond, but I just want to show a little bit about halogen bond. That is our earlier work. So here is an X. So if you look at an X, if this X is, for example, chlorine, this chlorine and this oxygen. So here is a drug molecule within the binding site. So if this X is a chlorine, this chlorine and this oxygen, okay, usually you would think that these two are a repulsive force because chlorine is an electronegative atom and oxygen is a electronegative atom, so you will have a repulsion. But if this chlorine is here where like, okay, this CXO angle is 180. If this angle is 180, then what you usually see is on this chlorine on this other end, you will have a positive charge. This is called as a sigma hole. So for example, you have a chlorine here. So this chlorine, we think like, okay, this is an electronegative atom. It's all negative, but it's not all negative. The negative charge is usually on this side. So on the other side, on the distal end, you usually see a positive charge. So because of this positive charge and this oxygen here, so these two actually attract each other, and actually this will form a favorable force. So this bond is called usually CXO. That's so this bond is called as a halogen bond. So here, look at this, where if it's a H, X is a H, the IC50 is 0.29. If X is a chlorine, H versus chlorine, then the IC50 is lower. That means like it's a better drug when you have a chlorine in this place instead of a hydrogen. So this type of bonds are called halogen bonds, and like most of you might be familiar with Autodoc Vena. Vena is a very popular, one of the widely used docking program. So this does not account. If you look at the scoring function, the way Vena estimate the binding affinity is by using repulsion, hydrophobic hydrogen bonds, and there are these van der Waals terms, Gauss1, Gauss2, but it does not account for this kind of interactions. So one of our earlier work was like, okay, so we incorporated these terms. For example, here you have a hydrogen bond, but excuse me, you do not have a halogen bond term. So again, sulfur also exhibit this kind of polarization. So we incorporated, like we studied like for halogens and for sulfurs, and we incorporated those into the scoring function of Vena. And we actually published two papers, one in JCIM and the other one in JChemInf, where we incorporated this this halogen bonding terms and sulfur bonding terms into them. And so the original one only has so many terms, and like then we added like, okay, halogen and sulfur terms into their scoring function. So here is an example. Here is like, okay, you can see this is Vena, and here is a Vena XP. We call like, okay, our program as Vena XP. When you have halogens, like actually you will see here. In this case here, you have a protein, and the one in green is a crystal ligand, and this one is a dockered pose. So we actually try to hold this area to see like, okay, if we get the natural pose. So this molecule does not go into the binding pocket because it is seeing that as a repulsion in this case when we dock with Vena. But when we dock with Vena XP, we actually could see like, okay, one of the poses directly superimposes on the natural crystal ligand pose. So this was one example where like, okay, we tried to incorporate some of the terms that were not present in the scoring function, and we modified to get more accurate results. So that was our earlier work. So we wanted to check like, okay, can we get better with the machine learning scoring functions? So we actually used a deep learning here where like, okay, we train the data on the PDB bind data set, which is actually a crystal structure protein. It's from protein. It's from PDB protein database, but actually they have separated like, okay, there is this PDB bind data set, where like they got all the structures from the PDB and that have a binding affinity with them. So here we try to extract different features such as close contacts, electrostatic interactions, binding pocket flexibility, hydrophobic contacts, hydrogen bonds, and pi interactions and salt bridges. And we try to train this. So we trained about 55,000 neural nets, and we tried to take like our top 10 networks. Try to take we took top 10 networks, and then we were able to look at our predicted versus experimental. We were able to get like within the experimental accuracy, where like our RMSE is about 1.15 kilocalories per mole on our test set. So this is like very close to an experimental results. So all the data and the algorithms. So usually like we are an open source enthusiast. So what we do is like all the algorithms and everything, like okay, we put it on our GitHub page where people can use these algorithms to make to benchmark our models, and also so our models can serve as benchmarks. And also if people wanted to make them even better, we highly encourage that. So we put it on our GitHub page. And another thing is like, okay, so most of the time, like when we are developing open source, these have like a lot of dependencies. So we actually use a containerized system. It's called Docker. So just wanted to mention this containerized system, and it's very important. Like why containerized system? You need to have a containerized system for your applications, is because if you containerize it, then anyone like okay, because before we're using Python 2.7, now we're using Python 3.5. So every time like okay, the dependencies change. Like if you containerize it, then you can your code always will work. Here is another example that we just recently published in JCIM, is where like we developed a machine learning model for biased signaling. So most of you are familiar with what a GPCR is. GPCR is a G protein coupled receptor, but these G protein coupled receptors, when a drug can bind to the receptor, they will show downstream signaling. So this downstream signaling can be two different types. It can be through G protein, which is alpha, beta, gamma subunits, or it can be through beta arrestin. So some drugs will activate both pathways. Some drugs will activate only one pathway. Some drugs will activate the other pathway. So it is very important for us to know the given drug molecule will activate this pathway or this pathway, because one could be a therapeutic effect, the other one could be a adverse effect. For example, we have opiates. Okay, so one could be relieve the pain, the other one could cause addiction and constipation and so on. So here it is very important to design the drugs that are biased towards one side signaling that we are looking for. So this is a very important for drug discovery, because most drug molecules bind to GPCRs. So we developed a model for this one. It's called BiasNet, where like okay, for a given drug molecule, you can look into it to see whether it will be a G protein biased or a beta arrestin bias. So those are some of the examples that we've been developing. We have developed recently. So just wanted to acknowledge my lab members So here is my lab members, and like, okay, so I have appointment in computer science as well. So like I actually work with a lot of students from computer science who does a lot of the work from our group, and different funding agencies. Actually, we have a recent NIH funding, and we have NSF funding, and TAC is a Texas Advanced Computer Center that we use a lot of computation for. So with that, I will stop my presentation, and I'll be happy to take any questions at this moment.
Thank you, Dr. Suman, for a wonderful talk. So I believe there is a question in the chat box. So it says, my query is that how we can test our model when experimental data is not available, like biological assay.
So if we don't have a biological data, then you will not be able to develop a machine learning model. So for the machine learning, if you don't have data, you will not be able to do it. But for example, you can use AI algorithms. For example, if you wanted to develop a molecule, if you know for example, like if you want to do de novo design, like if you know the structure of the protein, maybe that could be a start where like, okay, you try to develop a molecule and do a docking and see if it can fit into the structure of the protein. But if you don't have a structure of the protein and if you don't have biological data, in this case, you won't be able to do anything.
Okay, so I guess, can you answer the second one also?
Okay, so next question is, thank you for giving brief on MLAI in drug design. Okay, question, how to tackle problem associated with data processing by ML? Okay, in case of poor quality data, over or underfitting of data, or sometimes lack of training data. Yeah, so lack of training data, again, the answer is I already given with the first question. Okay, so data, so a lot of time people think when we're saying like, okay, data scientist or like a machine learning expert, there are a lot of different algorithms. But the thing is, the data is really, really important. If you don't have a good data, it is hard to do anything. So data is very important, and data cleaning. So actually, a lot of time in our efforts, we spend a lot of time cleaning the data. So if we get the data from multiple resources, what we do is like, okay, so we make sure like, okay, the data, we harmonize the data in such a way that like, okay, the data is high quality. So again, poor quality data will result in poor performance of the model for sure. So the quality of the data is highly, highly important. And another question in there is like over and underfitting of the data. Okay, so this is where like, okay, if you are a machine learning expert, you should know whether you're overfitting or underfitting. So you look at your so while training, so that's what like you will be looking at like, okay, how your model is performing on the validation set. So the validation set will help us in this over and underfitting of the data.
Yeah, very well explained. So I guess we have few questions from YouTube chat also. So I request Dr. Farhana Rosie to read out those to you.
Hello. Thank you, sir,
Yes.
for the informative talk. There is one question from Shweta Sharma. The question is, I will read for you. Is coding and decoding tools are available, or it is developed by your group?
So I guess the question is about the auto encoders. So yes, there are different algorithms that are available out there. So we are taking the most advanced ones, and like, okay, so we actually in our lab, what we do is we try to modify them so it can work for our problems. So just I'm pulling up my other slide. Yeah, for example, this is a slide where I was talking about encoding and decoding. So there are different algorithms. For example, here is one example where this is a paper where you can look at some information. So most of these algorithms are present in other applications. So if we look at what Facebook and what other people are doing, so technology companies, so Google. So we take some of the advanced algorithms and we're trying to fit those here for our problems, biological problems and drug discovery problems. So most of these are coming from the other side of the computer science. So these algorithms, again, there are several papers that people are publishing, and this is a very new field. So for drug discovery, so most of these algorithms are very new. So we take those and we implement our own methods into it.
Yeah, so I myself have a query about if you could enlighten us about that. So these models are developed with a totally different purpose. So would you think that that could come in the way of applying them in drug discovery, like opening
Yes.
a Coke bottle with a screwdriver instead of the opener?
Yeah. So very good question, and that is actually the problem that we face, because it's not made for this; it's made for something else. But the thing is, what we try to do is so we make our screwdriver work as an opener. So that is the challenge that we have to we deal with. So for example, here I showed here is an algorithm where this is used for generating images. So we are trying to use to generate molecules. So these images, so this is worked with the pixels. Okay, so we need to go in there and understand the code, and here we are, instead of pixels images, we are feeding smiles and trying to get smiles instead of image to image. And here in this case you're just changing the features here. So here we are developing different machine learning algorithms, so we are trying to incorporate those in here, where we are in the latent space, we are trying to give it generate a molecule in such a way that we get this particular property. So this is the challenge that we have. So it's not everything done for us and given to us. So, but we are trying to modify this in a way that we want it. So again, like the generative chemistry, this technology, it's not matured yet. It's just very recent, in the last 1-2 years. These are getting better and better. These are being developed. Not even better, these are being developed. And one of the main challenge in these are actually AI can develop molecules. It can come up with some molecule, but we need to synthesize these molecules, because that is the biggest challenge. So we need to so we also incorporate into this where a factor, because there are different factors. So one factor we say is the molecule needs to be synthesized. So there is a score that we incorporate. It's called a synthetic accessibility score. So can we synthesize it? Okay, how easy it is to synthesize? So that is one of the factors that we put it in, because that is our challenge. That is not challenge in the computer science, but it's our challenge, a chemistry challenge, a drug discovery challenge. Yeah, but that's a very good question, and that is a challenge that we face. Yeah.
So any other question we have on YouTube? Okay. So one more thing I got me interested in this slide that you showed, that For synthesis also you have machine learning and
Yes.
developing. Uh, I believe that these machines are not going to replace organic chemist anytime soon, but certainly they will make the space competitive, and one would have to become really expert to remain relevant in the field and job ready.
Yes. So, okay, so
Because
this is
the
another.
machine the repetitive task now that was not possible.
Yeah. So, I mean, there is a lot of times like we get this question like, okay, the machine learning and AI gonna replace the jobs, replace humans. Actually, it won't. It will never replace humans. It will never replace jobs. But the thing is that the job market will shift. Okay, so it's so instead of doing some repetitive task, that will be done by the machine, and like okay, some human will be asking the machine to do those jobs. So. For example, Amazon uses like okay, there's a lot of like okay robots okay for doing like a lot of labor within their warehouses and so on. So in those cases, there a lot of people will say like okay that it is replacing human jobs, but actually it's replacing labor job like we're like okay very low paying jobs with high paying jobs where like because a lot of people are writing the code for it and like okay they're so higher paying jobs. So there is always there is a shift in technology. Because of the improved technology we will do a better kind of job, so the humanity will do like okay better kind of jobs. But it's not again like okay for chemistry it is not replacing organic chemists. It is informing organic chemists like okay what is the best way to synthesize the compound, and like okay so what is the instead of we are pipetting it like okay the machine will pipette it, that kind of thing. So it makes our life easier, but it is not taking the jobs. If it takes jobs in one way, it is generating jobs in the other sector for the same purpose.
Yeah, thank you. That helps clear the confusion in the minds of audience, if any, including me. Thank you so much. And finally, we thank you once again for joining us at the late hour. And although it was inconvenient for you, we are grateful that you could make it. Thank you so much.
Yeah, it is my pleasure. Again, I'm glad I was able to join. Yeah, thank you all for attending. So have a nice rest of your day. Thank you.
Thank you. Bye-bye. So we already have our next speaker, Professor Chakravarti, with us. And who better than Dr. Kapil Leshwar to introduce him? So I request Dr. Kapil to please introduce our speaker to the audience.
Thank you, Dr. Dixit, and very good morning to all of you. So it's my pleasure to introduce our next speaker, Professor Rasid Kumar Chakravarti. Professor Rasid Kumar Chakravarti has been a founder, faculty member of NIPER SAS Nagar and had been intimately associated with the development of the institute by every possible step. He was the head of the Department of Medicinal Chemistry, NIPER SAS Nagar, for more than 2 decades. After his superannuation in August 2019, he was the visiting professor in the Department of Chemistry, IIT Ropar. He is currently an emeritus fellow and DAE Rajaramanna fellow in the School of Chemical Sciences, Indian Association for Cultivation of Sciences, Kolkata. His research interest lies in the area of synthetic organic chemistry, which include sustainable methodology development. And on the medicinal chemistry side, his research area covers on various thrust areas, which include various communicable and neglected diseases, as well as various non-communicable diseases. He has published 181 research paper with a total H index of 63, as well as he has filed 42 patents. He has guided more than 40 PhD students, and he has delivered more than 150 lectures in various national and international platforms. He is the recipient of various prestigious honors and awards, such as Indian National Academy of Sciences, Fellow of the Indian National Academy of Sciences, Ranbaxy Research Award, Indian Society for Mass Spectrometry Eminent Award, Chemical Research Society of India CRSI Silver Medal 2022, to name a few. I was fortunate to work under him during my PhD studies. With this brief introduction, I would like to invite Professor Chakraborty to deliver his lecture. Sir, please.
Kapil, do you hear me?
Yes, sir. Yes, sir.
Yeah, but probably you are not being able to see me. I am trying to sharing my slide, but somehow it is not coming. Then probably I have to log out and log in again. Let me try. Do you see my slide?
No, sir, we can't see your slide.
Fine. No, I know this link is a little bit. What I'll do, I'll log out and log in again, right?
Yes, sir. Yes, sir. Yes.
You let me know when I should proceed.
Yes, sir, we are live. Please start.
Should I continue now, Vaibhav?
Yes, sir. Yes, sir. Please continue.
Okay. I take this opportunity to thank Professor U.S.N. Murthy, Director, NIPER Guwahati, and his team, particularly Dr. Vaibhav Dixit and Dr. Kapila Sachet, to give me the opportunity to be with you. My plan was that to be with you in person, but because of the unavoidable situation, we are meeting virtually. And not merely to discuss about anti-inflammatory scaffold. This is an opportunity to share a few messages to you. So in this context today, I thought that I will put before you that new anti-inflammatory scaffolds, particularly through sustainable medicinal chemistry approach. So as you see that there are two keywords, inflammation and a sustainable medicinal chemistry. Inflammation is not new to this gathering, but we know that inflammation generally is considered to be a natural defense mechanism of any living system, but there has to be a very good balance between the beneficial effect and the adverse effect of the inflammatory cascades. And when this balance is not there, which leads to uncontrolled inflammation, and that gives to various kinds of disease pathogenesis. More directly impact is found to be in two disease conditions, which we call rheumatoid arthritis and a chronic asthma. But in general, and there is inflammatory bowel disease also, it is believed that for majority of the diseases that onset of the pathogenesis is due to the inflammatory cascade. And to control this disorder, relatively a small number of targets have been identified till date. So these are a few targets which are very commonly and maybe popularly used. And out of these, that enzyme-derived targets, particularly cyclooxygenase enzyme with a specific attacking the isoform specific COX-2 that leads to the therapeutics for to control rheumatoid arthritis, and PD4 in general, phosphodiesterase inhibitors that gives you to control another disease condition, which is commonly called a COPD or maybe asthma also, although there is a critical difference between asthma and a COPD. But when we started working on the PD, I think about 2001 or so, at the time only asthma or a COPD was the on target. But recently, that COVID outbreak also has shown that there is a very good scope to reassess the therapeutic potential of the PD inhibitors, because it has been found out that PD4 or PD5 inhibitors are found to be clinically useful for the treatment of pathologies that are similar kind of symptoms, particularly post-COVID condition like thrombosis, inflammation, and fibrosis. So that is the reason there has been a hope that SARS-CoV-2 infection can be to some extent managed by using PD inhibitors alone or in combination with other drugs that can give you beneficial effect to treat COVID, or at least what it can do, it can curb or block the COVID-19, particularly with reference to slowing down the disease progression as well as the severity of the condition. But we are yet to delve into this aspect, but that rekindles their further interest in dealing with the PD4 inhibitors apart from its classical use. Second buzzword was sustainable medicinal chemistry, which I want this audience to be a little bit careful in listening to and getting a message in this regard. Now, why a sustainable medicinal chemistry? We know that this is the popularly known value chain of the drug discovery, and this is actually the journey of medicinal chemistry starts from the lead generation, because target identification, generally the medicinal chemistry, they borrow from the molecular biology. So this is going to be the role of the medicinal chemist. And maybe in the today's context, that the central role of the medicinal chemistry is much more broader, as we see that medicinal chemistry has to cross talk with the other very intimately related discipline, so that we can think about what could be the most effective way to generate a new chemical entities. Now, within again the core expertise of medicinal chemistry lies primarily synthesis of analogs around the lead molecules. First to generate the lead molecules and then synthesis analog around the lead molecules, while parallelly, you are going to interact with all the diverse expertise so that you can think about how a candidate drug can come, or maybe it can be put at least to the in vivo toxicological evaluation. So this happens to be the what Vaibhav was talking today, that yes, synthetic organic chemistry looks to be the core expertise and the core exercise in the medicinal chemistry discipline. And that has been debated long back, and ultimately people have realized that organic chemistry is the central stage in the drug discovery. Only thing is that there are various other areas has to be amalgamated, then only we can think about a really more effective molecule. So, but the question is that we know that other day Dr. Sastry also was telling that we have a chemical space. Chemical space is a limiting. Yes, chemical space is there, but still synthesis is the bottleneck. So synthesis is the bottleneck, and why reason? What you require, that Me Too scaffold probably may not be able to give you the fruitful candidate, although it's still one approach for that. What is required? A completely new carbon skeleton, which is called carbogen, new completely structured. Till date, only way to get a completely new structural scaffold is from the natural sources, and we know that how difficult, how challenging it is to find out such kind of natural product. So once in a century, you get a molecule like Taxol. So you have to think about that how to synthesize the compound in such a way we can generate a druggable scaffold, but which might have a new kind of a carbogenic structure. Apart from that, the recent problem or recent concern is that all these things looks to be dealing with all the chemistry and etc. But doing the chemistry in the right way is the another concern, which is the today's concern, which in this context, the sustainability is going to come. So how to avoid the adverse effect of the chemical research on the environment? So why we are concerned about that? Now, let me take you a few examples, like impact of chemicals, which includes drugs as well as pharmaceuticals on the environment. We'll come back to the chemicals. Now, this provides you all the data come from the industry. E factors means what is the waste we are going to put into the environment. So all of these four figures are related to the chemical exercise, but we take out the oil refining because it may not be under the domain of the pharmaceutical or medicinal chemistry. All the other three, bulk chemicals, fine chemicals, and the pharmaceuticals, all they are related to either medicinal chemistry or the process chemistry with reference to the drug development. And we see that these are the most polluting exercise, but unavoidable exercise. So that is the reason the concern is going to come that there is a need to integrate or address the sustainability. Sustainability becomes a very global term in terms of any kinds of development, societal development. But when this aspect is critically analyzed in the domain of the chemistry, then it called a green chemistry, become a popular name. So to integrate or address all those issues during the drug discovery and development. In the development, probably it comes automatically, but in the discovery, which is ignored, which is called discovery chemistry. Why? Now, this again provides you a very popular funnel called discovery funnel, which says that roughly around 10,000 compounds, and these 10,000 compounds are not Me Too compounds. These are diverse scaffold for that, and perhaps here the luck comes, although scientifically it's very difficult to accommodate the word luck, but fact is that yes, luck, because there are so much parameters which is not a proper control of the drug development chemist. So one compound might come. So think about that exercise which has been done, or the polluting exercise has been done to synthesize Baki remaining 9,999 compounds. So those are basically useless, but we have to do this exercise. Otherwise, we cannot go into this one. So we have to address those things that, and that is called a sustainable medicinal chemistry. And this has been recognized maybe a couple of decades back. And that is the reason it highlights the green chemistry tools are going to influence the medicinal chemistry and any chemistry research-based organization. So how to do it? Now, one obvious way to handle will be that what are the major polluting substances we deal with in making any chemical substances, and in this regard, implication of the solvent become very high. So this figure says that for any typical chemical manufacturing processes, that a solvent amounts to the major mass utilization. More than 85% of the mass utilization come from the organic solvents. They are supposed to be recoverable because they are low molecular weight, low boiling solvents, but their recovery efficiency varies from 50 to 80%, generally average close to 60%. So that means 40% of this material is going where? It is going to the environment, either to the air or to the aqueous body. So that is the reason there has been a proposal that can we develop eco-friendly solvents which are alternative to the common volatile organic components or even solvent-free reaction. Solvent-free reaction might face a problem in the heat transfer issue in the bulk production. So that is the reason people will go for. So what are the way out? Again, it is the potential issue in the industrial sector. So there are various pharma industry. They have come up their solvent selection guide. This is a Pfizer solvent selection guide, which will tell you that commonly solvent used in our research laboratory, college of medicinal chemistry or organic chemistry research laboratory, looks like that those are not going to be the desirable one. And the preferred one is going to be the first row, and in which we can see that water is at the top of the table, which very rarely an organic chemist, synthetic organic chemist or synthetic medicinal chemist would like to have a wet environment in the laboratory. So this perhaps try to tell that water might be the ultimate green solvent. So there are various other alternative reaction media has come. So these are the 5 commonly used for that. And generally, we try to deal with at Naipur Mohali. We used to deal with water, ionic liquid in a different context also, and fluorous alcohol. So these are the 5 major types of alternative reaction media. Now, Vaibhav also talked about today, doing the AI for that, whether AI is going to replace the organic chemistry, right? So very nicely answer has been given by the speaker. But what has been observed over there, that medicinal chemists do everything other than the chemistry. And that is the reason this article has come. So what it tells you that it prompts for further synthetic research in the synthetic area, suggesting chemistries that are at present underrepresented, but are in clear need of robust, reliable, and widely applicable need to be readdressed. Even it says that one, further improve the already tried and tested procedure, which are already employed for that, methodologies means organic reaction, where there is a clear deficiencies in the area such as atom economy, safety, and etc. So it is not like that. All the organic chemistry has been done. Chemicals, no chemical space exploring. Now coming back to this one, what AI and machine learning does? Yes, machine learning provides you to probably give a finer way to adapt a particular synthetic route so that it will minimize the time and etc. But on the basis of what? On the basis of the data which has been generated by organic chemists or medicinal chemists, they have been put together. Like today, we know that what are going to be structural alerts, because all the unsuccessful data of the toxicology data that has been come together. So if those data are not there, then we do not have the database to predict whether this molecule has a toxicophore or not. Exactly the same thing. If your backward Hartwig reaction is not discovered, then there is no way your AI or machine learning will take into its synthetic step Buchwald and Hartwig reaction. So what it does, it is the database, and now it has a different way for that one. So point is like that one, it is never going to replace a synthetic chemist. Only thing is that there might be various ways to do the same synthesis. It will give you some way out for that one so that you do not have to do all those exercises manually. And most importantly, how do you do it? You need to do how to carry out that reaction, and that computer does not do it. It is all your exercise which is coming from the organic chemistry school or synthetic medicinal chemistry school, then only you discover the reaction. So again, there also, it is not like that. All the organic synthesis has been done and nothing you can do it, so you borrow it. It is, and that is the reason this article has come. Now, on the top of that, there is. So now the question is that, so what is the suggestion? Now, this is a suggestion again came from this article. Read it very, very critically. So what are the sustainable practices? Now, basically, there are 2 practices. One is, although it is a maintaining the record, and first is the synthetic strategy, CH activation, aqueous chemistry, biocatalysis, at Naipur Mohali, we initiated a very good way biocatalysis. Then this is called another way to do the multi-component reaction, telescoping for that. Right. We know the CH activation is a buzzword in today's synthetic chemistry. I'll come back to this one, and aqueous chemistry. So we realized this thing earlier before this came into the comprehensive suggestion for that. So at Naipur Mohali, we initiated wherever there is a possibility to use alternate reaction media or to address the sustainable chemistry while in the discovery chemistry. In alternating reaction media, water, we do not aggressively propose ionic liquid as a reaction media, but we propose a different way to use the ionic liquid, non-solvent or organocatalytic and fluorous solvent. But today I will discuss about use of water and various catalytic processes. Also, it gives a sustainability. So we have developed various solid supported protic acids, and transition metal catalysis as well as nanocatalysis. Copil has contributed significantly in my laboratory to initiate the nanocatalysis oriented work. But today I am not going to talk about those works which Copil has contributed. But we'll take under the transition metal catalysis, I'll take an example of the CH activation while addressing the sustainability in the discovery medicinal chemistry. So now coming back to the first agenda, like alternative reaction media, sustainability using aqueous chemistry. It's not new. As a matter of fact, the very first organic synthesis is actually fulfilling the two basic criteria of green chemistry called 100% at atom efficiency as well as a reaction in an aqueous media. I'm referring to Euler synthesis, ammonium cyanate to urea, right? But probably in the true sense organic synthesis, where we are going to make from one organic starting material to the another compound, this was perhaps reported first in 1931 by Diels and Alder for the Diels-Alder reaction. When it was repeated about 15 years later on, Woodward and Baer, they found out there is a difference in doing the reaction. There is a change in endo-exo selectivity. So much so, it was realized the implication of aqueous chemistry. And as early as 1914s, US patents were filed for Diels-Alder reaction in water. But probably the most triggering incidents came through this observation by Koenig and Huisman in 1973, who distinctly proved a beneficial rate acceleration in water on Diels-Alder reaction. And perhaps the credit goes to Ronald Breslow. In 1980s, he made it to common knowledge to water as reaction media to, in general, the organic chemistry people. So thereafter, there has been a lot of studies, and based on that, we can find out that there are examples in which performing a reaction in aqueous media do accelerate the reaction, occasionally change the selectivity also, and people have used application in organic synthesis, application of aqueous reaction to synthesize material. May not be the final compound, but at least the key starting material. And then what could be the possible role of water? Now, it has been proposed that hydrophobic effect is a major effect and occasionally hydrogen bonding effect. But when we started thinking about implementing the aqueous chemistry in the discovery medicinal chemistry, we started thinking what could be the specific role of the water. And we realized that all these proposals were relevant to only one type of reaction, which is the Diels-Alder reaction, not has been integrated to any other organic reaction in general. So we had the question that what could be the role of the water? That means a molecular level interaction of water with the substrate so that we can get all those advantages in a rational way. In this regard, we have developed one very important reaction called en-Bock protection reaction. You call any organic synthesis where you have to manipulate the nitrogen amino group, or in peptide synthesis, we have to protect, and most commonly en-Bock protection, P-Bock protection is utilized, promoted in aqueous media without taking any catalyst. And the reason behind that is that water can activate the Bock anhydride through hydrogen bond formation, and to give suppose the transition state 1, and subsequently it is going to reorganize the nucleophile in such a way that now oxygen of the water is going to undergo hydrogen bond formation with the NH hydrogen, so there is activating the nucleophile, and through the transition state 2, in which both the electrophile and the nucleophile has been activated, that's why we call dual activation, the reaction can proceed. It's a very beautiful reaction. It is a self-indicated reaction. You do not have to do any kind of analysis to find out whether the reaction is going or not. No TLC, nothing like that one. As long as the reaction progresses, you can find out the effervescence of carbon dioxide, and when the reaction stops, then there is no effervescence. If the product is solid, it will precipitate out. You need not to do any further purification. Almost you can get analytically product provided it is a solid material. In case of liquid, you have to triturate it. So it is a very good reaction. Right? So it works very well. Then one cello does not make it a summer, so we need a further validation. And for some reason, we are looking for this kind of, if not the target molecule, but intermediate of the target molecule. And classically, this can be done by a Michael addition reaction. We got developed half a dozen of dense metal catalysts for this purpose. But since the target molecules are going to be subjected to enzymatic assay, and enzymes are very much susceptible to a presence of trace amount of metal, particularly transition metal impurities, and sulfur is a very strong acceptor of a trace amount of the transition metal. So we were looking for a completely metal-free, particularly transition metal-free synthesis. So we thought that one, can we put into use the dual activation role of the water and see whether it works? And it works very well. Right? So, and we have propose the role of water as amphiphilic, means electrophile nucleophile dual activation by cooperative hydrogen bond formation reaction. And subsequently, we found out that our proposal has been used to explain some other gas phase reaction in which acetaldehyde is reacted with hydroxyl radical, which is promoted by a single water molecule through the kind of proposal which we have given. Only thing is that they have done a very critical computational analysis and tried to prove that one, that this, when water undergoes dual activation through this transition state, like hydrogen bond donor as an acceptor, then it provides is a lowest energy transition state. That is the reason single water molecule catalysis takes place. This is of theoretical interest, but practical interest of our to explain to use our concept to explain the was found out in this case in the marine natural product. These toxins, normally brevitoxins, saxitoxins, all the toxins, they have actually a polyether kind of molecules in which the tetrahydropyran moiety is repeated at 10 or 20 molecules are there. And those people try to mimic that why they are, how to make it in the laboratory. So it is basically intramolecular nucleophilic ring opening reaction of an epoxide by a suitably positioned hydroxyl group. So any attempt people try to do in the laboratory, it either gives the 5-member ring closure product like THF or a mixture of THF and THP using all the organic solvents. And finally, they propose that it can be only happening in the water, and water actually undergoes a dual activation through the hydrogen bond formation reaction. That is the reason why in the marine natural product you get a 6-member THP. There also they have taken our proposal. Wait me for a minute. Pratim, I'll call you back. I am in a webinar now. আমি তোমাকে পরে ফোন করছি। ঠিক আছে। So that was the first proposal in the literature. When we tried to put that one, that dual activation role, only those 2 papers came. So after organic letters completed 20 years of its publication, they published a virtual issue called a Platinum Anniversary Collector's Edition. What they did, a group of editors, they have picked up one particular paper in a particular year, and with those 20 papers, they have the Platinum Anniversary issue. And in 2006, they have picked up this particular paper, that NT-Bock formation in water media. And this is the only representation of the Platinum Anniversary Collector's Edition of the organic letter from India in the last 20 years. That doesn't mean that on other papers are not there. So that means it tells that this proposal has such a far-reaching consequences. So at the time the students are not there, but I have shared with the student, and they feel very happy that they have contributed some fundamental knowledge, and that also working in an institute which is not a classical organic chemistry or basic research institute. And that is the motto of NIPER. So you do not do a me too research, you do something where you can have an impact in the context of the scientific achievement. So we had our own example, and we tried to implement all the aspects of the aquatic chemistry, and we try to do discovery medicinal chemistry. So we have done a lot of process chemistries also, like totally all water chemistry. But today I will try to put an example before you, like how we can implement the sustainable medicinal chemistry and developing a new chemistries also at the same time with PDE4 inhibitors, particularly asthma and COPD. So while discovering, so we know that by this time inflammation, we know that there are two targets popularly used by the medicinal chemistry, COX for different disease pattern and PDE4, and particularly in today's context, PDE4 has a little bit more importance. It might provide you a little bit advantages in treating the SARS-CoV-2. We are yet to initiate our activity in this regard, but plan to do. So we are looking for, searching for a novel PDE4 scaffold, maybe about 15 years back. And we found out that whatever the molecules in an advanced stages, at the time, it was piclamilast and the roflumilast, they were in the advanced clinical trial. So later on in 2014, roflumilast won the race and piclamilast didn't because of the much beneficial effect compared to the adverse effect for that. So still there is a lacuna of the enough number of PDE4 inhibitors. In 2012 or 14 it got approved. So we are looking for at the time before it becomes approved, that what could be the new scaffold for that. Rolifram is the first generation, a prototype molecule, but it is a non-selective inhibitor. So we did preliminary in vitro screening. We realized by this time that this upper part of the molecule called northern region or the moiety A looks to be a conservative. Only thing you can do, you can make a combination, various combination of this alkyl group in the catechol ether moiety. So only scope you have to make a suitable replacement, bioisostatic replacement, is the southern part or the moiety B. It is a heterocyclic moiety. So we did a quick virtual screening. So what could be the most appropriate replacement as a heterocyclic scaffold, which is going to recognize by the PDE4 active sites. And in this regard, we found out that benzothiazole scaffold is going to be, so we have compared both oxazole and thiazole. We found out thiazole is very comfortably sitting in the active site. So we can club, covalently attach the heterocyclic as a benzothiazole to them. So design part is over. Now comes the another important and unreplaceable activity of the medicinal chemist, which is going to be the how to make it. So if we go for, we find out what are the various procedure. There are various approaches for that, and none of them are going to qualify in the context of the today's sustainability. So by this time, the microwave assisted has become one technique for a green chemistry protocol. We adapted that, it works, but there is some disadvantage where depending upon the stability of the carboxylic acid, sometimes it undergoes decarboxylation, so naturally you end up with the mixture of product, but it works very well. Many people utilize our chemistry to make their own starting material. So we wanted to do it in a very milder condition. So when you realize that what is that kind of reaction, the kind of reaction is nothing but an electrophile-nucleophile combination. So our earlier two example, Bock protection as well as that thiamycalation reaction also is a electrophile-nucleophile combination, and where water finds enough scope for dual activation, activating both the electrophile and nucleophile. So we thought that can be put into use, and in fact the reaction works very well. Simply heating these two substrate in water and 110 degrees centigrade gives the product. Here also, depending upon if the compound is solid, it will precipitate out, and you may not need to do any further effort for the purification. So to realize whether water provides any beneficial effect, we found out, this is a classically monitored by the TLC, that when the solvent is water, then within 10 minutes the reaction is complete. So in terms of the rate or acceleration, water provides a beneficial effect compared to the classical organic solvents. In terms of the selectivity, the reaction proceeds via the intermediate formation of the dihydro compound called benzothiazolene. And here we found out through GCMS analysis that when the reaction medium is water, practically no accumulation of the undesired product is found out in the final product. In all other cases, varying amount of the undesired product is there. So here also we found out that water provides you a selectivity also. So this is the reaction condition. But then we became a little bit more critical. we found out that these are the 5-point agenda to sustainability in terms of the energy. So in terms of the energy consumption, it is not fulfilling properly. So can we carry out the reaction without heating? So then it comes the classical concept of the organic chemist. We are carrying out an organic reaction in aqueous media. So naturally it is going to be insoluble. So why don't you take a solubility enhancer? We started it with this preliminary expectation. So what we did, we use various kinds of surfactants which are used as a solubility enhancer, neutral, cationic and anionic surfactant, and monitor the reaction by GCMS to see that what is the extent of the desired product 3A versus the undesired product 3B. And we found out these 3 anionic surfactants, SDS, SDC, sodium deoxycholate, and sodium dioctyl sulfosuccinate, SDOS has been abbreviated at. They provides the best result, where in case of SDOS, almost no accumulation of the undesired product. In all the other cases, it gives at least, so that this gives more than 90% or more than 85% yield of the desired product after, of course, isolation for that. So then we became a little bit critical. So we found out, is it due to the solubility? So when we have carried out the same reaction, but we take SDOS, but we change the solvent from aqueous media to non-aqueous media, we found out in all the cases the reaction is a very clear reaction mixture. But product outcome is not good, means we get a very poor yield of the desired product. Why now? Because we found out undesired product D is a significant amount. So it is not due to the solubility at all. So whenever we are taking organic solvent, a clear reaction mixture, but it is not reflected in the product outcome. So it may not be at all due to the solubility enhancing ability of the SDOS, which is doing the job over here. So to have a critical insight regarding the specific role, so we did what is called that various selective reactions. The first 3 entries are reaction is performed in water without any SDOS. We found out that within 60 minutes the reaction is over, means there is no starting material, but we get a major product is 3B. So cyclocondensation takes place in the absence of SDOS in water, but moment we are giving SDOS, then we get 100% in terms of the desired product. Then we thought that what is happening now, there is a dehydrogenation, we call it oxidation also. So if that be the case, if we pass on oxygen gas to the reaction mixture, then it should make the reaction faster. In fact, it happens. D means we have taken water, SDOS, and bubbling oxygen into the reaction mixture. Instead of 60 minutes in open air, it takes 30 minutes, which tells that yes, in fact, it is helping to capture the oxygen, or maybe for capture the hydrogen which is being evaluated to give the product. Then we thought that is it possible then oxygen can do the job. So next 3 data are you take water but no SDOS and bubble oxygen into the reaction mixture, and found out no, we get an undesired product 3B as the major product. The next 3 agendas are there. We have taken SDOS, we have taken water, but water is a degas and under argon atmosphere, and there also you get almost 1:1. So what you need the availability of the air or oxygen, you need to have water, and you need to have SDOS. So perhaps what SDOS is doing, it is capturing oxygen from the air and delivering to the reaction mixture so that it can easily eliminate the hydrogen and consume the hydrogen to make the water molecule. So if that be the case, the relative catalytic efficiency of the various surfactant should be depending upon their oxygen uptake ability. So what we found out, yeah, I'll do it, right? I'll quickly. So we found out that the reaction is all those things is going to be very effective catalyst. So it works, right? So we could find out. So what is the reason? This might be the reason that we can accommodate the oxygen, water as well as SDOS. So to prove it, we found out that this intermediate can be isolated by can be proved by the corresponding ESIMS technique. So this is the intermediate, and as the reaction goes, there is a increasing of the concentration of the supramolecular assembly. So that tells you that this is going to be actually catalytic species. So this is exactly how the reaction takes place. So the reaction works. So what we had to make the molecule, we have either this method or these two methods, depending upon the availability of the starting metal, you can do it. So with this, we made the compounds, and we did find out that this is the best effective molecules, but not this is the most effective molecule amount we have done, but this is not the best effective molecule. So we have actually the lead structure. Now comes the lead optimization, what changes we can do without taking the right starting material. So I have talked about that you have to develop new chemistry, but at the same time, there has been, you read this article for that, right? Where have all the new reactions gone, all the CH activation and etc. So it tells that you have to employ all those reactions in the real reason for which you have done, and only real reason is that can they find a place in your drug discovery pathway or not. And that is the reason this article, Nature Review, it tells you that there is a necessity to have an intense crosstalk between the synthetic and medicinal chemists in the industry and academia, so that you can enhance the impact of these methodologies in the future drug discovery. So there is a need to integrate all those CH activation, and this article also proposed that CH activation. So we adapted a late stage activation. I'll complete it within maybe a couple of minutes time without giving details. So what we need to functionalize. So we have by this time that one way we can do it, that if we go for when we did the molecular docking, we found out there is a lot of pocket solvent pocket is there, and Dr. Sastry has published a review article which he says that you can make a good selectivity towards PDO4 compared to PDO4D, B versus D, if we can replace a lot of water molecule from the solvent pocket. And one way will be a bulkier group. So one way to introduce the bulkier group is the aromatic moiety, which is going to be hydrophobic also. It is going to replace the water molecule. But we thought that instead of having only hydrophobic moiety, if there is some other moiety which can have an interaction, so arylation reaction. So there are a lot of CH activation for arylation reaction, and we did find out that these are the disadvantages, and there is a scope to develop a new chemistry also at the same time. So what we did, we developed a completely without using any co-oxidant, using oxygen as a molecular oxygen as a oxidant, and the reaction works very well. I'll quickly go for that. That the reaction proceeds via the CH activation. It provides by a KH-KD study by NMR. By this time, we have developed a very good expertise in utilizing mass spectrometry. It provides you a real-time analysis. With the ESIMS analysis, also provides a very good indication that the reaction proceeds via the CH activation. I'll not go further details for the mechanistic, right? Let me go further. So with this, we develop a new way also to make the target molecule benzothiazole. So with this, what we did, we adapted both the techniques. In some cases, the NHPI mediated works. In some cases, it doesn't work. It depends upon the reactivity. Then we take the classical one, in which we take instead of NHPI oxygen, we take the tertiary butyl hydroperoxide, and we have prepared the molecules. This is where that generally when you have a very bulkier substitution over here, then we found out a lesser yield in our catalytic version, where we are taking a molecular oxygen for that. So with this, we have various ways how to handle the chemistry and adapted the CH activation techniques. And we had a larger number of molecules which is showing a very good PDE4 inhibition, more than 80%. Few molecules have been taken further to find out their selectivity, and now further is going to be take them for the in vivo testing. So what I tried to present before you that adapt a sustainability in the medicinal chemistry exercise, and one easiest approach will be our most obvious and desirable approach is going to be manipulating the reaction media, and to this extent water has been proposed as a reaction media for performing organic reactions, but molecular level understanding how water provides these advantages were not known. In this context, we have proposed amphiphilic, means electrophile nucleophile dual activation through cooperative hydrogen bonds, and it is a conceptual advancement in understanding the aqua chemistry, as has been demonstrated by utilizing this concept for the why THF containing toxins are produced in the marine natural product. Generally, aqueous chemistry, people try to adapt a surfactant only for the reductionist view, like it is going to be solubility enhancer. In this figure, I say that no, do not have a very conservative attitude, you can think about, you might find out a new chemistries, and in this regard, looking beyond the role of the surfactants from the solubility enhancer, we found out it is a non-Him model of the dioxide activation. And this chemistry we have been adapting for various kind of oxidation, which is going to replace the transition metal-based catalysis. And the molecular basis of understanding of the role of in this case has been found out by mass spectrometry. Yes, we have utilized the aqueous chemistry to find a new molecular scaffold as a potential therapeutic to manage asthma or COPD. And lead optimization has been done by taking the new advancement of the organic chemistry, particularly late stage functionalization using CH activation. Right, we know the chemistry. So again, that no matter how much machine you do it, it is not going to replace your chemistry attitude for that. And particularly, modern life depends upon the petrochemicals and all the pharmaceuticals. But looks like that the current processes, whether finished product or trying to get the product in terms of the new drug development, that they are not a sustainable, and the sustainable way, which is called green chemistry approaches, aims to tackle this problem, right? But it is not merely replacing one catalyst by another catalyst, replacing a organic solvent but aqueous media. At the heart of green chemistry is innovation and the discovery of new chemistries, not merely an incremental improvement of the traditional synthetic methodology. The word green in green chemistry refers to phrase young and new. And I think by proposing the first time from Naipur Mohali, the dual activation role of the water, which has found applicable in understanding various kinds of aquatic reaction, we have found out that no, it is in fact the innovation, and by adapting the CH activation techniques, in which not only the CH activation has been employed for the late stage lead optimization, but identifying a new way for oxidation by using molecular oxygen. So this demonstrates that these are not merely a methochemistry, you have an innovation. So this has to be kept in mind while we are doing a research, particularly in a flag-bearing institute like NIPERS. So all these are the students, and here is Kopil. I didn't have a scope to discuss about his chemistry. And Naisargi actually, she did the water-mediated reaction, but earlier there are two students like Sune and Rajkumar. Rajkumar is Central University Bhatinda, who has done the earlier that dual activation role. Lot of all-water chemistry for the synthesis of process chemistry has been done by Damodar and Dinesh, who is on NIPER Ahmedabad. Sudipta has done a lot of fundamental work in the ionic liquids, which is he is in Department of Chemistry, Delhi IIT, but I didn't get a scope over there. And Brahma, who is in the industry, he's in a very responsible position. He has developed a lot of transition metal-based catalysts, particularly leading to the cardiovascular drugs. Right, so keep in mind, this institute, the motto is innovative ways in pharmaceutical education and research. And I'm very happy after contributing as a founder faculty for the last close to 25 years of service. I have again relocated myself. It's not merely my alma mater, in Indian Association for the Cultivation of Science. Now, this is much before could realize that importance of the interdisciplinary. We talk about every other interdisciplinary. Now, he is a Indian Association for the Cultivation of Science is not only the first research institute in the country, it is the first research institute in sciences in Asia. And this institute was not conceived and was not instituted by any chemist, by not any physicist. It was thought about by a interdisciplinary person called Mahendra Lal Silkar. He is a medical practitioner, he's an allopathy doctor, used to practice homeopathy. More than 150 years ago, this gentleman felt the necessity, though he is a biologist by profession and by training and knowledge, he felt the necessity to have a culture of the basic research, fundamental, initially physics and chemistry was there, later on biology was adapted. So that tells you that interdisciplinary, if you have to do that one, you have to come out from your complementary compartmental attitude for that And that is the reason why NIPER, within 10 years of its existence, become a brand name for that one. So it embraces the science and science only. They do not go for that one, that you are a pharmaceutical scientist, you are a molecular biologist, or you are a basic organic chemist. Wherever your expertise is required, you have to embrace it, and then only you can really contribute. I think that's what I had to share with you. Yeah, I am done, Kapil.
Thank you very much, sir, for your kind words of advice and guidance. I'm definitely sure students have many more questions to ask to you, but because of the lack of time, and we need to keep to the schedule also. So we skip, and we are sure that you will visit us personally, and there we can have long sessions of discussing chemistry.
I plan to do that.
Understanding more details. So with your permission, sir.
Yes, sure. You proceed.
With your permission, sir, I introduce our next speaker.
Yes, sure.
That is Dr. Jagannath Mondal. He works at TIFR Hyderabad, where he is associate professor. He has done PhD from the Department of Chemistry in University of Wisconsin Madison, and done postdoctoral research at chemistry department in Columbia University. He is also associate of the Indian Academy of Sciences Ramanujan Fellow, and there are many awards. In the interest of time, I am skipping that. His current research is wide spectrum of computational chemistry and biophysics, ranging from a computer simulation of biomolecular recognition processes to explore genome biophysics. Today, he will talk about computer simulation of protein small molecule binding kinetics in real time. With this short introduction, and I am also aware that he is not totally well today, but therefore I am expressing additional gratitude. Thank you, sir, for having patience and being with us. Sir, please, over to you. Thank you.
Yeah. Can you hear me? Hello?
Yes, sir, we can hear you and see your screen also.
Thank
Please
you.
go ahead.
Thank you, Dr. Boivov, and thank you very much for your kind invitations. And it's an opportunity for me to talk about what we are doing in our research. And as you already pointed out, we don't do any wet chemistry in our institute. In my lab, actually, we are looking at small molecule recognition with protein by using computer simulations. But the thrust that we are trying to emphasize on is basically on real time. So we'll slowly figure out what we are in real time, what we mean actually. But in this community, I think this does not need any introduction. That the biomolecular recognitions in the context of in the context of drug design is actually one of the most ubiquitous phenomena that everyone is interested in. And as far as the recognition process is concerned, protein ligand binding is actually one of the most well studied and well sought after system that is actually being looked into. So, what I'm trying to show here is that I'm trying to show here is very simple schematic of its binding site, which is protein, and we have a small molecule ligand which is supposed to, complex and try to form, protein ligand bound structures. And, in a very simple chemistry, one can actually write it in terms of also a reversible reactions. And, the point is that it actually undergoes binding, and then it also tries to, get out and try to basically release this and try to unbinding to form these things. And the equilibrium between these two actually decides the efficacy of the molecule. So what actually we are actually interested in our group, we are actually interested in what are the kinetic pathways that this small molecule takes to eventually get these particular locations. So we know that these actually the crystal structure which actually tells you that where the ligand is going to basically be bound. But what we really wanted to see, if we really look at in a completely backward way, we'll ask that how does the small molecule for the entire protein, why it is only exploring that particular location and getting in there. So that is what actually we are exploring, and we are trying to figure out how it actually what are the pathways it basically uses to finally get into the pocket. And accordingly, what we are going to do in today's lecture, we are trying to talk about two stories where we are trying to where we have tried to answer this kind of questions. In the story one, which is basically system which is quite well regarded as a test bed for many of the computer simulation and experiments, which is a system called T4 lysozyme, which was originally devised by Brian Matthews and his co-workers in 1992, where they tried to do a small mutation in a 99th residue, which created this particular mutant, and that actually started showing that that particular small molecule, that small mutations can actually become very efficient binder for many small molecule or aromatic molecules. So this is basically one of those molecules. So this is basically called a solvent inaccessible cavity. But what we generally know is that for this particular system, the binding is quite rapid. So this is one particular system that we have explored and we are going to talk about. Near the end of this particular part, we are going to talk about one joint venture with an experiment and simulation, where we are trying to look at how small molecule can actually act as a sensor for certain bacterial protein, and we are going to show that how the knowledge that we gain from computer simulation can be tested or validated and can set up a crosstalk between the experiment and theory. So this was mostly the first part was basically done in our group in collaboration with certain NMR groups in TIFR as well as in University of Toronto. The right hand side thing that we are going to talk about later part is going to be presented in collaboration with Dr. Ruchi Anand at IIT Bombay. So with this overview, I'll just first talk about this, what is the thing about T4 lysozyme and why it is very interesting. So the thing, as I told you, this system, if you look at the structure, it looks actually completely solvent inaccessible. But there has been a paper which is on 1996 by Frederick Dahlquist, where they actually did quite a lot of NMR experiment and tried to point out that the title of the paper was very catchy. It was basically showing that the access of the ligands to cavities within the core of a protein is rapid. So that actually, by rapid, what they mean is that if you look at the biomolecular rate constant, it will be 10^6 to 10^7 molar inverse second inverse, and they estimated the barrier height of the activation barrier, which is 2 to 5 kcal per mole, and that is actually quite interesting from a computer simulator perspective, because what people generally will apparently think that if it is really a buried cavity, then it will be very difficult for ligands to get inside. But for this particular system, which was shown, is that the rapid binding is something that is quite paradoxical. So so accordingly, what we really wanted to check, what actually how does this small molecule actually gain rapid access to this particular solvent inaccessible cavity, and what are the pathways for this particular getting there actually. So now if you really want to ask a computer simulation person, and basically this community, people will say that there are many different approaches that people have taken over the time, and the way I try to draw them is basically I try to put it on an axis which is called computational accuracy, and the other one is called computational expense. By computational expense, I mean that the kind of resource that you need, the kind of time that you need to really spend to do this kind of exercise. So on the right, on the bottom hierarchy of this thing, I am just showing something called rigid body docking, which everyone is quite familiar with in this audience. What we do is that we have a target already decided by either the structural biologist or a crystallographer, that this is the location where the protein is supposed to bind. And then we have this small molecule, and we try to see, if I try to basically use these lock and key based approaches, can we fit in there? If it fits in there, then we will say that this is most likely going to be a good structure, and otherwise not. Now the advantage of this particular thing is that this is computationally ultra fast, and actually in fact right now you can do it in a decent laptop also. But the disadvantage is that it is kind of decided by the user that where it is supposed to be explored, and that is where you are going to explore, and that actually basically is the one of the disadvantageous situations. Now the other approach, which actually will say that computationally more accurate but computationally also expensive, is that okay, we can do one thing. We might see that the ligand is basically there inside the cavity, but we can try to look at the reverse process, where we can try to accelerate the unbinding of this particular ligand by some technique which is molecular dynamics, but adding external forces, and that actually has been done, and that we can do something by technique called metadynamic simulation, which we are not going to talk about in this particular lecture. But the problem with this particular problem is that this will be dependent on how are you going to pull it out, and so that will also be a biased approach. So now on the top of this hierarchy, one of the wish list that people can think about is that if I really want to figure out where the small molecule is actually bind, the one particular approach will be that, why don't we try to simulate this entire small molecule, looking around and basically binding its own location. And that actually, in principle, can be done in molecular dynamic simulations, because if we can do it, that will provide you the true real-time dynamics. But the disadvantage is going to be very expensive, because you have to wait for almost for a long time, and these are very stochastic process. Many times you might not see the result which you really look for actually, because you have to wait for a long time. But what actually the problem is that this MD simulations, as I think most likely has been discussed in this workshop, this is actually computationally very simple, because if you really want to simulate any any object inside a solvent, all you have to do is that you have to just basically provide an initial configuration, you have to define the interaction potentials, and then you have to just use Newtonian equation to basically move them together. And if you can move them together in real time, you will basically be able to figure out the trajectory or the entire history of the small molecule or any particular large system to move around, and that will basically help you to figure out what actually is going on. But the problem is that why it is not so easy, it's mostly because, especially in protein link and binding dynamics, why it has not been so useful is mostly because the underlying phase space or the underlying landscape that we generally look for is extremely extremely rugged. And that actually is because the systems are generally very complex. You will see many other intermediate which might actually can be looked into. And accordingly, what actually has been the problem is that it is very difficult that in a molecular dynamic simulations you'll be able to see this kind of events so easily. Okay, so then what actually has happened over last 5 or 6 years, there has been more development on the software, but there is also very important development that has taken place in also hardware. And one of this particular development has been in the graphics card. So what used to be this popular thing called video gamers, who used to use these game ready drivers which are called GPUs, now is being used in a computer, and people are rewriting their program so that you can actually make sure that computer reads also the graphics card processed programs, and then you try to basically see whether you can accelerate it, and that actually is showing pretty big acceleration. And this is one of those GPU based computing facility that we have built in our institute, which actually help you to accelerate the overall computing in a significant way. And this is exactly what we did actually in our project. So when we started this particular simulation, what we did is that we'll try to see whether it is possible for us to look at the entire process of the small molecule, which is benzene, finding and binding in this particular cavity, and whether we can do everything in all explicit water simulations, and can we actually look at how this actually finally gets into this cavity. So, and we basically used a fully all atom MD simulations, and basically this is one of the simulation trajectory that I'm trying to show you here. So what we find is that during the simulation, which is basically about 2 to 3 microsecond long in this particular, which is quite long, at least when you start at that time, you will basically see that the small molecule is basically touching different location of the protein. It also is flowing around in the water, and then when the time is right, you will start seeing that the small molecules start basically on its own looking at and basically binds in a particular cavity, which looks almost like the cavity that was close to this L99A actually. So this actually was one of the simulation trajectory that we basically could generate, and then we thought that why don't we first check how good is this result actually. So one way to check is that you can actually have the crystallographic structure of this particular simulation pose already crystallographic pose structure already available. Then why don't we basically do one thing? Why don't we try to overlay our final bound simulated pose with this crystallographic pose? And what we found is that if you look at the root mean square deviation between the crystallographic and the simulation pose, they're almost within an angstrom level resolution actually. That means that the computer simulation has been able to predict the actual bound pose that you really wanted to look for. And very importantly, that actually what you have done, now you have the access to this long simulation trajectory, which will also tell you the pathway that it has taken during its basically binding to this particular cavity. Now what we did is that we actually did not stop it in one simulation. We tried to reproduce the entire process, and we carried out multiple trajectories. In all trajectories, we found that simulation length changes goes from almost 2 microsecond to almost 8 microsecond. But in all the cases, the ligand binding actually happens. And the RMSD, if you look at in all the cases, the ligand binding happens at a precise location as what you basically see in a crystallographic pose. So that actually tells you the simulations are reproducible, and you can actually then trust the simulations and try to look at what actually is going on. Okay, so so what actually we found is that when we now went back and we basically try to look at what actually is going on as far as the ligand binding is concerned, what we found is very interesting. We found that if you look at one of these stimulation trajectories, if we look at this one, you will see that the small molecule is binding through these two helices, which is the red and the blue, and these are called helix 4 and helix 6 of this particular gate. And if you look at another simulation trajectory, you will see that in this particular case, the ligand binding is happening through another two helices, which is basically called helix 9 and helix 7, and they are quite opposite actually. So this is actually quite interesting because it is saying that unlike the common approaches that people generally talk that is the only dominant pathway that is actually possible for a ligand binding to happen, it is actually showing that there is multiple pathways by which the ligand binding can actually take place. And what we find is that this is almost like a conformational selection, that there is an opening that is actually being taken place and then the ligand is getting there. Now, to characterize what kind of opening it is there, what we did is that we actually calculated or estimated the distance between these two different helix, the helix gate opening, which is this black curve. So if you see the helix gate opening by this black curve, and if you try to overlay the ligand approach to the cavity, you will see that for a particular moment, the ligand actually just opens up for a while. Sorry, the helix gates are just opening up for a while, which is this black curve increasing, and that is the moment that the ligand takes, and basically during that particular time, ligand is basically jumping to this particular pocket. And if you look at the time that actually takes, you will see that this time is about 50 to 100 nanoseconds, which is very, very tiny, short time that actually the ligand gets to get inside this pocket, but that is sufficient for this ligand to open actually. So this is subtle, but this rare opening of the interhelix gate, which is basically driving this particular process, and this is happening for multiple independent trajectories actually. So now, we try to look at, and we basically saw that in all cases actually, this kind of gate opening is actually quite common. In all the cases, one of the gates are opening, but this has to be different other gates. So if we look at individual trajectory, in all the cases we found that the ligand binding is basically quite different, and that actually tells you that there are multiple pathways, and the rare opening of this interhelix gates is actually the key driver for this kind of process actually. Okay. Now, being a more like a simulation or the physical chemist, what we really wanted to see is that can we actually now exhaustively sample the entire space and try to ask ourselves how many pathways the ligand will follow to achieve this final bound state, and how much is the contribution of each of the pathways, and can we actually, more important, as we try to point out, can we predict the long time equilibrium kinetics and the thermodynamics. So in this particular case, one particular approach which is becoming quite popular, which is called developing Markov state model, so that one can actually tell this entire story in a quantitative way, but in a more simple language. So what actually, I'm not going to talk about Markov state model in this particular, in this particular series, but this is basically mostly, contributed by two groups, which is Vijay Pandey and Frank Nguyen, and they are students and they're postdocs. And basically what actually you do is that you have lots of independent simulations which is done here, and what you do is that you use those raw data, and then you try to couple them into certain discretized microstates, and from those microstates you try to look at what is the equilibrium between those macrostates that is being taken place between them actually. Okay, so essentially what actually underlying this particular Markov state model, what lies, you generally try to look at independent simulation trajectories, you try to cluster them into certain states, and then what you calculate something called this transition probability matrix by counting how many transitions you see between two states. So if you have green states to red states or green states to blue state, what is the time, how many transitions you generally see, and then what you generally apply, you apply this key feature of Markovian process, where you say that if you have a short time trajectory for which you know the transition probability, you'll be able to extract what will be happening at the long time trajectory by by using this short time data actually. And that actually is the power of Markov state models, that it allows you to predict the long time property of this particular situation. So this is exactly what we did in our simulation. So this was mostly done by my former postdoc Navjit Hala. What actually he did is that we already had quite a long simulation trajectories. We spawned many short trajectories for different locations of these particular systems, and then we basically had almost a 60 microsecond simulation trajectories that we really could play with. And then what we did is that we developed such a Markov state model on this particular data set, and that actually gives rise to a very simple but very quantitative story. It tells you that the entire result that you are finding can be can be simplified into a very quantitative but very four state model. In one of these models, let us call these four state pictures. So these are MS0, MS1, MS2, and MS3. MS0 is the unbound state. MS3 is completely, fully bound state, and MS1 and MS2 are two intermediate. And what we find is that these intermediates are actually appearing near this helix 4 or helix 6 or helix 7 and helix 9. And very interestingly, it also tells you that what is the contribution of each of this particular pathway. And you can also figure out there are also possibility of direct transition from unbound to bound, and there is also intermediate driven transition, which is also contributing. And there are also, two intermediate taking place in a particular transition and finally giving rise to one particular transition. And what we can do is that we can also extract what is the lifetime of each of these intermediate. And if you look at this intermediate lifetime, they are actually very small. They are about 69 to 742 nanosecond, and it tells you that these multiple pathways by short-lived intermediate is leading to this hypolysozyme. Okay, so what you can actually do is that you can also extract the kinetic constants from this particular Markov state model by calculating something called mean fast passage time, and you can calculate the k on and k off, and you can also extract the delta G. And what we basically see is that if you calculate all this quantity, generally the order of magnitude comes up to be very similar, and that actually tells you that the Markov state model essentially captures most of the the redetermining step that you are looking for in this kind of ligand binding simulation. Okay, and what we actually extracted is that you can also extract, if you try to do a very simple transition state theory on this particular model, you can also extract what is the barrier height for this kind of process, and this turns out to be only 5 RT. So together, if I now try to tell this entire story together, so the question was why in this particular solvent inaccessible cavity the binding was so rapid. So the answer will be that there are multiple pathways that this ligand is trying to take, plus there is the low activation barrier. Together, you are basically essentially getting a very rapid binding. So that actually tells you that even though the cavity is extremely buried, but ligand has its own way to devise multiple pathways which are low in activation barrier, and that will ensure that the ligand can actually still get in there. Okay, so but what we learned from this particular entire exercise is that it is very, very stochastic, very complex and rare event, at least rare in the computers. And then the protein conformation is also changing while the ligand is searching its designated cavity, and large statistical sampling is actually required. So what we really wanted to figure out, is there a way that we can actually discover an optimal reaction coordinate which will identify these multiple ligand binding pathways in hypolysozyme. So in this particular case, we try to use machine learning approaches to see whether we can actually answer these kind of questions. So we already had a lot of simulation data in our hand, and then what we did is that we actually started thinking that can we figure out a CV which will basically display this multiple ligand binding pathway on a low dimensional landscape. So for that, we actually use this approach of artificial neural network, which is basically called autoencoder, which is self-supervised machine neural network. In this particular case, what we are doing is that, let us say you have an initial image, you are trying to reproduce the same image, and in between you are basically having a very compressed bottleneck layer, which is your essential data that you are looking for. So this is almost like a dimensional reduction technique, but using artificial intelligence. And what we did is that we actually had many of the simulation trajectories, and we just extracted the protein and ligand distances from all the simulation trajectories, and then we actually implemented that inside a machine learning framework where we are trying to reduce the dimension by using certain weights and biases to certain bottleneck CV, which is chi1 and chi2, and then again regenerating the original input, and we are trying to see whether what is the optimal value of this weights and biases, which will give rise to the minimum cost for this kind of work, so that you will lose least amount of information. So this is exactly what we did in our cases, and what actually turned out that if you now project your simulation data on this chi1 and chi2, you'll basically see that you basically get a very rugged landscape which is basically completely divided into certain unbound state and in bound state, and you do discover small intermediates which are very, very tiny in the populations, but they actually do exist. And then what you can do is that you can now actually try to project your simulation trajectory on these particular data sets, and you will see that actually the simulation data does capture this kind of ligand binding in one of these pathway. You can also do another simulation trajectories, and you can also project it on this particular surface, and you will see that the ligand is actually really capturing going through these intermediates, and basically then essentially it is basically going to the the completely bound pose actually. And what will happen is that if you wait for a long time, it will basically take some time to transition between these states, and then it will finally get to the bound pose actually. So this is actually it tells you that there are multiple pathways, and these pathways can be easily captured also in a low dimensional but very complex collective variables. All right. So the take-home message for this particular part is that we try to capture the the real-time dynamics of a particular ligand inside a cavity, and we try to show that the machine learning tools can actually help you to figure out the collective variables, and the simulation actually help you to reconcile the decadal paradox from the static crystal structure and NMR data. Now, what I'm going to do for the next 5 or 10 minutes, I'm going to give you a small update on what we actually got recently engaged with is in looking at whether we can provide insights to experiment for a particular bacterial protein which is known to sense small molecule like phenol actually. So this was done with Ruchi Anand's group at IIT Bombay. So what we knew from her group is that this protein is called MOPR, and this particular protein actually has 3 different domains. We are basically looking at the one is something called the sensor domain, and in this particular domain, this particular domain is known to selectively and sensitively sense phenol. And very interestingly, this is actually only senses phenol so accurately. Any other aromatic molecules, it will basically kind of try to discourage binding actually. So we really wanted to see how does the phenol sensitivity is so selective in this particular cases, and can we actually figure out what is actually happening. So we actually thought that why don't we figure out what is the way that this particular ligand binding is taking place, and what is the structural basis for the selectivity shown by this phenol. Okay, so for that we did the similar kind of exercise. In this case, we actually had the N-terminal domain simulated. N-terminal domain simulated, and what we found is that this phenol is actually very interestingly roaming around, and then it is basically finding a particular gate which is designated by one leucine and one one methionine between one beta sheets and one basically helixes. And then what actually find that if you wait for long enough, it will basically sit there properly and basically get in there actually. And that actually told you that okay, in this particular case, there is the phenol sensing is happening by the phenol, and the simulation can actually capture the trajectories. But the question is that why it is so sensitive? That was the question actually that we really wanted to target. And that's why what we did is that we first carried out multiple simulation trajectories, and what we found is that unlike the previous one, here we find mostly one particular pathway. There is also another pathway which is pretty less minor, but there is one particular pathway which is actually taking place in most of the cases. And then when we try to dig around, and we found that what is happening, there is there is a particular, originally there is this buried cavity. There is a channel which is being formed, and that channel is being basically used by this particular phenol to basically get inside this particular pocket. So this is almost similar to what we basically found earlier in the T4 lysozyme case. What we found is that when the ligand is getting inside, there is this channel opening, and that channel is actually basically being closed. And when we really focused on the channel, we found that there is actually 2 residues which is playing an important role. One is this L119 and one is M110, and this is phenol. And what we find is that inside these two actually the ligand is actually trying to get. So what we actually observe is that most likely this methionine, leucine, and this valine, these 3 particular residues are providing hydrophobic environment to this phenol, and these 2 residues are most likely also keeping the gate open. So what we suggested is that one particular way to test whether this is the pathway that the phenol is taking and whether it is selective enough or not. What we can do certain mutation-based analysis where we can actually replace this methionine, leucine, and valine by some other residues, and all try to create a possible solve bridge to stop this particular possible ligand binding and see actually what actually happens. So this is exactly Ruchi's group did. So they carried out certain mutations. In one cases they tried to change the methionine and valine to polar substitutions. In those cases, compared to our original wild type, the KD changes significantly. It basically tells you that the ligand, the phenol binding becomes very, very difficult. We tried two different ways. In one cases, we tried to replace the methionine and the valine. In other case, we replaced methionine and leucine by two different polar residues. And in both cases, the change in the KD was drastic. There is also, we tried to do another thing that we thought that why don't we replace the non-polar residue by another non-polar residue, see whether that keeps the same protein or not. And we found that interestingly, if you replace the same methionine or leucine to something like valine, we don't see any differences in the KD. That actually tells you that this is almost like another synthetic analog of this mopper, which you can recreate. And finally, what you can do, you can try to block this particular salt bridge by a salt bridge, where you replace this methionine by a lysine and valine by this glutamine, and that actually makes sure that the binding completely gets stopped. So that actually tells you that this particular pathway, if the experiment is suggesting that this is the mutation which is basically stopping the ligand binding, that basically means that this is the pathway that the ligand is actually taking, which was originally predicted by our simulations. So with this, I think I'll just close the session. I'll basically try to point out is that we try to capture the ligand binding in two cases. In one cases, we try to answer some existing experiment. In other cases, we try to basically establish and motivate new experiment. So with this, I would like to use my time to acknowledge the persons who did the most of the work. For the T4 lysozyme project, it was done by Dr. Navjit Ahlad, who is right now a faculty in Agricultural University at Haryana, and the Satyabroto and has been the person who has taken care of the machine learning project. And the collaboration for the experiment part for that particular thing came from Dr. Pramod Polurapalli, who is my colleague at TI for Hyderabad, and Lewis K, who is a very popular NMR scientist at University of Toronto. For the MOPR project, it was mostly done by Mohammad Sahil, who is a graduate student in my lab, and Jayanti Singh, who is a graduate student in Ruchi's lab. And it was really, interesting discussion with Ruchi that actually started this project. I would like to thank my TI for Hyderabad for computing facilities and the funding from DST and the DAE and the Ramanujan Fellowships. And thank you very much for your attention.
Thank you very much, sir, for a wonderful talk. So I guess there is a question here. So the question is there a logic behind deciding the time and time steps to run an MD simulation? How we can choose the best simulation running time for metalloenzymes?
Yeah, so I think so irrespective of whether this is a metalloenzyme or any other non-metallic enzyme, actually the point is that the time step we cannot change much if you are really doing the all atom simulation, because this will be driven by the lightest atom that you actually have, which is hydrogen, correct? And hydrogen is vibrating at a particular frequency, and that needs you to make sure that you cannot change more than 1 frame per second to 2 frame per second. So that is the time step fixed. Now, how long you have to run, that will decide. That will be depending on the let us say if you have some idea from experiment what is the binding rate constant, then you can most likely go ahead and try to recalculate. Considering the concentration of the ligand that you have to do, you can most likely recalculate how much you have to take. But you have to, you have to also think about this other situation. As I tried to point out, the simulation length are not fixed for all the cases. We did not see binding in same time for all the trajectories. It actually ranges between 2 to 8 microseconds. Now, if you ask an experimentalist, they will say that 2 to 8 microseconds from their perspective is very short. But if you are going to do a computer simulation, you will say that 2 microseconds. Okay, but 8 microseconds is very, very long. So that actually you have to also think about that. So what we did in all the simulations, we kept on running it until we saw a binding.
Okay. So any other question from the audience online on YouTube? Okay. So I would like to ask a question regarding the accuracy of the force fields, because for small simulation runs, although that these methodologies, the early developers have already got Nobel Prize in 2014, and they have addressed their in their talk about the accuracy of the force field. But what is your opinion if we keep extending the time for which we keep running these simulations? Will it become the quantum effects and other non-polar effects, will they start to dominate and add to the errors in the simulation, or they are expected to
Actually,
get cancelled?
yeah, I think even if you extend the simulation, first of all, the quantum effect will not be there because there is no quantum effect in any of our empirical simulation because this is completely classical simulation, correct? So that way it will never do. But you are right about one thing is that that there used to be time when people used to look at if you run the simulation for a long time, many times you used to see that any particular protein does not even stable. Correct. So that actually used to basically used to give warning to people that okay, maybe you have some problem underlying the force field that you are trying to play with. So now what I can say is that the force field is still an evolving project, which is still a project that is being done by the groups who actually are really good at doing it actually, and people are aware of the limitations. But even with that, I will say that back compared to last 10 years, I think I'll say that now the existing force field is much more reliable because the first thing when we started here is to see is that whether the crystallographic pose and the simulation pose matches or not. Because if you get that, actually, that also tells you quite a lot about the force field. That at least something is right there. That's why you are matching things quite well because you are not putting in extra forces. Now, the second thing is that it does not also stop you to criticize the force field because many of these small molecules which basically are too small, which is might actually have other polarizable effect which is not completely present in our simulations. So those effects, if one can incorporate, definitely maybe it will be better. For example, many cation pi interactions, which we don't really consider in all these things explicitly, that might be very important. But in that actually definitely is something that hopefully will be addressable. And with the better computers, I think hopefully the polarizable force field will also become more popular actually.
And beyond that, there are other experimental large number of variables like the buffer.
Right. So that actually is also very
Experimentalists are using.
exactly
How
the
important
buffer.
thing is to simulate the exact conditions that they have in the experiment.
Right. So basically, what we try to do is that we try to ensure that the concentration of the ligand that we are trying to play with is right. If we know the certain protonation state of residues from the experiment, we try to ensure that those things are being conserved. So that we have, at least for MOPR project, we have made sure that for for triple we made sure that the concentration of the ligand that we are trying to put in is not too high. It has to be the same as what they do it in NMR. So this basic cross-check I have to do, but you are also at the same time right that the pH is something that one need to really play with. Even now the constant pH simulation is not so popular in MD simulation because these are very difficult to do. But it is becoming now almost people have started using it now. So hopefully it will be becoming more popular eventually, and that will make things more accurate.
Okay, thank you very much. I guess there are no more questions. So with that, we thank you, sir, once again for taking out time for us and showing the exciting work that you have been doing on this. And we look forward to learning from that also.
Thank you.
So thank you once again. For the audiences offline, online, we would like to announce that another session after lunch will start at 2:30 and we'll have speakers from the US, both AstraZeneca and Pfizer, which will be followed by a demo session of the Discovery Studio. Therefore, please do come back and join us then. Thank you. Are we live? Abhijit? Yes, good afternoon once again and welcome to all. Our next speaker is Dr. Prakash Rathi from AstraZeneca, UK. He is the augmented drug design engineering lead at AstraZeneca in Cambridge. He has done masters from NIPER Mohali, PhD from the University of Düsseldorf. He has postdoctoral experience at Astex Pharmaceuticals, and he expertises in drug design, molecular modeling, homology modeling, pharmaceutical research, structural bioinformatics, chemoinformatics, machine learning, and AI. And his current work is centered on augmented drug design engineering at AstraZeneca. And today he is going to talk to us about Melody, privacy preserving federated machine learning for drug discovery. So with this brief introduction, I hand it over to Prakash. Please start.
Thank you, Weber. Hello, everyone. I'm Prakash, and I'm an engineering lead. We help drug discovery scientists to run DMTA cycle, design, make, test, and analyze for early drug discovery. And we run quite a few projects and platforms within this, including Melody, which is a European collaborative project to build machine learning models using data from different pharma partners without revealing the actual molecules. So what is Melody? As I said, this is a collaborative project where both public and private partners came together in 2019. And the main objective was, can we accelerate drug design by sharing the knowledge across different pharma partners so everyone gains without sharing the actual molecules or actual targets. So this was funded by European Union Innovative Medicine project, but also pharma partners contributed into it. So 10 pharma partners and European Union funding, plus the public partners in the form of KU, Catholic University in Leuven, Ictos Pharmaceuticals, Cubometics, Ocwin, AWS, NVIDIA, and so on. So this is a 3 years project, conceptualized in 2019, but started in 2020. And each year, there were 3 objectives. The first year was to actually achieve the technical feasibility if such a machine learning platform which is privacy preserved, which means no data is revealed to any other partner, is feasible at the same time achieving better performance. So the technical feasibility was achieved in first year, which I will talk a little bit later, what's the technical architecture and how this works. And in the second year, it was to actually estimate and compare whether the models that are produced using data from multiple pharma partners are better than the models that would have been produced using only your own data. I will mainly focus on this because this has been achieved most recently, last year summer, and currently we are in the year 3 where we want to further improve the predictive capabilities of these models. With all these objectives in mind, there were some principles which were paramount, which couldn't be breached. For example, the whole platform should be secure by design. No data breach or data revelation can happen. It must be audited by third party. It must be functional. in such a way that it produces the models that are needed, and it should be able to do federated training at scale. So 10 pharma partners, billions of data points should all be handled. So as you all are aware, drug discovery is a costly affair. It takes years worth of time, but up to 2 billion euros of money to bring a drug to the market. The reason being that there are many steps to find the lead molecule and then to optimize, then bring it to the clinic and do clinical trials. It all takes so much time and money. And during the journey of getting a drug to the market, a huge amount of chemical and biological data is generated by different pharma companies because they test thousands of molecules before one of them reaches to the market. And all this data can be used to build models that can help us in future drug discovery projects. So each pharmaceutical companies, 10 of which are the partners here listed here that are partner in the MELODY project, each pharma partner has hundreds of thousands of compounds that they synthesize, they test against many different targets of their interest, be it the primary target for curing the disease, or it could be a target that could result into toxicity, for example, hug other targets. So they test their compounds against many, within many assays against many different protein targets. And you might see here a schematic representation of that there might not always be huge overlap because different pharma partners might be working on different targets, and they might have different chemical series and different molecules, but there might be some overlap. So the idea is, can all this information be added to one single model, sorry, not one single model, but can this all information be shared securely between the pharma partners such that each pharma partner gets a better model for their own targets of interest? This is where federated machine learning comes into play, wherein the data is federated to the central server, and then model is built, deep learning model is built, which I will describe in a moment, and each partner gets a model that learns not only from its own data, but also from the data that the other collaborative partner has. So this is the traditional setting where each pharma partner can build models based on their own data and they get their models fine. But how about turning this competition into cooperation, wherein the information is shared centrally and exchanged between different pharma partners securely? And I want to emphasize here, the information which is shared centrally and passed on to the other pharma partners is not the information about the is not the information in terms of what molecules each pharma partner has or which target it is working, not even directly sending any molecular descriptors without first masking them. So securely sharing that information and communicating via a secure channel, which is also cryptographically recorded so that it can be audited. Each step of the training is audited, and then each pharma partner in the end gains from the knowledge of other pharma partners. So this whole idea then sparked a thought and discussion across the pharma partner. How can we achieve it? So if you think about machine learning, and particularly about deep neural network, in a traditional setting or single task learning, what you do is you have a data at the bottom here, if you see, and you feed it into a neural network, and the neural network learns the representation of your data in some way, and then you train the model to predict one of the tasks. of your interest. Maybe you can build a model to predict binding affinity for a kinase of your interest, or you can build a model for plasma protein binding, ADDMC model, or you can build a model for hERG toxicity. So this is like a single task learning. So you have multiple models, all separate. But on the multitask deep learning neural network, what happens is that you try to predict all these tasks within the same model, which means that all the molecules that are related to different tasks are fed to the same joint representation, same deep neural network, but at the last layers of the model, it predicts different tasks. What it provides is that because there's some overlap or learning from the different tasks that can be advantageous for other tasks if there's a complementarity in those tasks. The overall all model will be more generalized. They will become better predictive. They become better at predicting new molecules, and applicability domain will improve. So this is like in a normal setting, and many pharma companies and academia, they do multitask learning, wherein you get generalized prediction, better generalized predictions than doing individual model building exercises. However, we can take one step further. This multitask learning could be done across multiple partners. So data can come from different pharma companies, and they go into shared joint deep learning representation, and then each pharma company can then predict their own task individually using this shared representation. Right. So how this works technically is that think about the neural network training. So each pharma partner sends some fingerprints, circular fingerprints of the molecules that they have, and they want to predict some properties of. They send it to a central server, or we call model dispatcher, securely to the central server, and then the central server actually aggregates all the information and then sends it back to the pharma partner. In the deep learning, how the training works is you start with a random parameters on your node, and then you try to change those parameters or weights such that your prediction accuracy of the task improves in each step. Something called backpropagation, where during the training, the weights are changed iteratively. So here, each pharma partner, during each step of the training, sends updates to the weight based on their own assays that they want to predict. So each pharma partner telling to the neural network during the training that based on my structures and my assays that I want to predict, this network should be changed in such and such manner that these weights should be changed in this direction, this much, and so on. But when 10 pharma partners are telling that based on my assays and molecules, you should change this, the central server aggregates all those weights and then sends them back to each pharma partner. Okay, you told me this is the weight for the network in the next round, but based on all of the pharma partners, this is the aggregated weight. Please use this. And then this keeps on, this carries on for many steps. Then a final model is produced for each pharma partner. And you see here, the weight updates come from different pharma partners, which means the model has information. A model has learned from molecules and assays from different pharma partners. And you see here, this is all privacy preserving, that no pharma partner shares data with the other pharma partner, but the central server. And even though the central server, I will show you later, they don't share the molecules or the assay. A little bit in more detail, if you see here, each pharma partner all share the joint representation. So it is like a head and trunk model. So here are the individual pharma partners at the top. They are predicting their assays. Pharma partner A, B, C here, for example, and they all send data to the joint representation, the lower initial layers of the model, and at the top level, they all predict their own assays. So this is how they get a model which learns from different pharma partners but predict their own assays of interest. This is the technical slide, technology slide, how this all is possible. One of the key foundational infrastructure which helped this consortium to achieve this is the cloud, of course, and we use AWS cloud. So each pharma partner has its own private AWS VPC, virtual private cloud in AWS, which is not shared with anyone else. So all data is with the pharma partner in AWS. And then everything is run in a Kubernetes container, Kubernetes cluster with a Docker container, which means all pharma partner can use same algorithm, same software for communicating with the central layer as well. And then there's an application layer provided by Oquin, which does the federated machine learning shared communications. And the algorithms for multitask is provided by Catholic University Leuven. And data for this architecture is provided by individual pharma partners, including AstraZeneca. So, as I said, for pharmaceutical companies, the data privacy is paramount because billions of dollars worth of money have been spent on generating those data, and they wouldn't want to share with anyone else for good reasons. So here there are multiple layers of data protection that was embedded into this platform. Firstly, data never leaves a pharma partner's account, AWS account, so they own the data, they keep the data. And then the data is pre-processed. The fingerprints are generated using RTKit, and then fingerprints are shuffled so that no one can even know what, even if they're leaked, they wouldn't even know what order they are in. And then the assays that the pharma partners assays that they wanted to predict were converted into classification tasks. So this is like a data preparation layer, which doesn't share the molecules here. But then there was an IT security layer. Data was all encrypted, no sensitive metadata like which assay, etc., or which molecules class is shared. Data storage is only temporary while the model training is running, and data is immediately deleted afterwards. And this is all in the isolated AWS account. On top of that, model itself has quite a few privacy protection embedded. As I showed earlier, head and trunk model architecture, which means they only talk to the central server and sends the weight updates. And that also had a systematic noise added, which only cancels out when all the data is aggregated. So even if one partner's data is breached on the way when it's going to the center server, it wouldn't reveal anything because there are 4 layers of protections. So with this in mind, I will give a little background on data preparation, and then I will talk about the results that were achieved in the year 2. So as you might imagine, there are 10 different pharma partners, and they don't want to share their data to one party, but they want to prepare data for machine learning consistently in a exactly same manner as other pharma partners have done, so that the deep learning exercise can be carried out. So what Melody has done is they prepared libraries for preparing data, and in fact that has been open sourced. You can find it on GitHub, how the data is prepared there, etc. So So these libraries, exact same versions, are shared with individual pharma partners, and they run the data preparation locally and store the prepared data, which are the shuffled fingerprints, into AWS accounts. Right, as I said, the first year run, which was in 2020, achieved its success. We achieved secure design, audited, and a functional platform, and we produced some models as a proof of concept. But the major success came last year, summer in the year 2, where we produced some models and compared them against the models that were generated using the single pharma partners. And year 3 is ongoing as we speak. Right. So how was success evaluated for the year 2 runs? So as you all are aware, in the machine learning, we can have a training set and then validation set, which can be used for hyperparameter optimization, and then the test set, which never becomes the part of the training, which is used for evaluating whether your model is predicting better than random. For example, in classification, you will see area under curve, precision recall curve, etc., or correlation coefficients, and so on. So this is the strategy used here as well, and we had 2 types of model: classification and regression. So performance was evaluated using correlation coefficients or area under curve, depending on the type of the model. But also we evaluated whether, as you see, one of the advantages of the multitask learning is that the applicability domain is extended, which means the models are more generalized than what you would produce from using only your own data. And to test that, we used conformal prediction efficiency metric to see if the models produced using multiple pharma data is better than the single pharma data. And here are the second year 2 results. So if you look here, most of the, so here you see on the bottom chart, left-hand side, these are pharma partners numbered 1 to 10 in random order, and then you see the improvement in prediction. So each bar here is telling percentage improvement in the model performance compared to the models that they generate using only their data. So positive the number, better it is the multi-pharma models. And across all pharma partners, barring one, we have seen improved performance of the federated multitask models that learned from multiple pharma partners compared to the single pharma partner. And interestingly, ADDME, subsubstance distributed metabolism, and that sort of tasks performed even better than the average on an average the multi-partner models did, because ADDME models gain more pronounced improvement because there's more overlap in the ADDME, because the actual binding affinity on the targets, usually you don't have much overlap between the pharma partner, but you would imagine that the same ADDME protocols many pharma partners must be running. Right, and then the applicability domain also increased substantially when you use the Campbell data to build the multi-pharma partner model against the single pharma partner models. Clearly, it is interesting to see is that although performance improvement looks not for second year itself, performance is improved, but the applicability domain improved even more compared to the single partner models. So there are other benefits of this federated learning, more than just getting this getting these models for these pharma partners. They have a MELODY study group which improve model predictions all the time. They improve algorithms that they share with the general public on GitHub, and this is actually the first time ever a federated machine learning run has been conducted in pharma settings. So this can set a precedent for future where multiple pharma companies, but also other industries come together, share their data securely in such a manner, and build models that help everybody. There were quite a few technical and scientific achievements have been made during the MELODY project, which would be helpful in other contexts as well, like the applicability domain metric, which I showed earlier, and how you split fold methods and evaluation, validation of your model, and the all federated machine learning head and trunk architecture running on scale has been all done for the first time, which will be used beyond building these models for these 10 pharma companies. And quite a few open source tools have been produced during the MELODY. For example, for data preparation, as I showed you earlier, MELODY Tuner, which anyone can download and use it for their machine learning projects, available on GitHub, which has information about compound standardization and generating fingerprint shuffling, etc. And then there are public data sets that they have produced to test out the system in the first place. The pharma partners wouldn't share the data even for the first run. So the benchmarking and standardizations were done on Campbell and other public data, but those data sets were prepared in such a manner that are amenable to the federated machine learning, and those sets are available for others. There are machine learning techniques that have been improved and that's also available as SparkM by the public partners on GitHub. So these improvements, and there's something in development at the moment, is to write this prediction pipeline that others can use. So please take a look at the GitHub page to see what is available for everyone there. With that, I will finally share this AstraZeneca slide to say that so what we do at AstraZeneca is, as any other big pharma partner, big pharma company would do, is target discovery, drug discovery, preclinical testing, clinical testing, approval, delivery, operations, and everything. Where we come is that we are part of the large organization within AstraZeneca called R&D IT, which help all the processes all the way with the data and AI. We have a number of positions available for data and AI, and I've linked here a few of them, which you can take a look and apply. And particularly the platform I come from, Augmented Design, Test, Make, and Analyze. We help drug discovery teams, early drug discovery teams, with the data, AI, and software needed for generating new molecular ideas, predicting properties for them, and predicting synthetic route for them, etc., and providing data at their fingertips. That's where we come, and we also have quite a few openings. Please take a look at the career page of AstraZeneca. With that, I will finish my presentation, and thank you everyone for listening.
Thank you very much, Prakash, for a wonderful talk and making us aware about what is happening at the big pharma and how they are collaborating together to come up with novel solutions to the drug discovery problems. So here in the chat, I see there is a question. What is fingerprint shuffling in pre-processing data?
Right. So to give a practical answer, so you can generate fingerprints of your molecule, which I assume most people understand is that you have a small molecule, let's say, and you can take each. You can read each atom and have some properties like what's the atomic number or hybridization and so on, and then you can generate a circular fingerprint by looking at the neighboring atoms and combining those properties into one and so on, and you can do it multiple times. So then maybe radius 3, so you do circularly keep on merging those properties, and then you get some list of numbers, and finally you can hash those numbers into a vector of a fixed length. For example, 2048 sized fingerprint. These are all then numbers between 0 and 1. So these then represents what the original molecule was. And one protection Melody used was that you don't directly use that fingerprint for machine learning training, but you shuffle them in a systematic manner that only you know, so that even if it is accidentally released, someone cannot reverse engineer, even though fingerprints are not reverse engineerable easily, but there was an extra layer of protection that you change the orders.
Okay, I guess that answers the question. There is a follow-up question: how to decide on what basis one compound will be used as training set or test set?
Right. So here, so there are many ways. If the question, I believe the question is related to Melody, but generally what you would do in machine learning is that the one is you can do random split, like take the 80% as a training set and 20% as the test set. But the problem with that is you might have quite similar molecules in your test set that were part of the training set, which means you are testing your model on the molecules which would give you very good accuracy because quite similar molecules were in the training set. But here we used scaffold split. So we did take some scaffolds that are not represented in the training set, so that you get a better generalized true accuracy.
Okay, thank you for answering that. Before we go to YouTube, is there anyone online has a question? Okay, now we can take questions from YouTube. I request Dr. Rosie to read out that to you.
Thank you, sir, for a wonderful talk. There is one question from our YouTube audience, which is Runesh Podili. He asked, what is hyperparameter optimization?
Right. Okay. So in machine learning modeling, especially deep learning, what you do is during your training run, let's say you do an experiment for solubility prediction. So you have a training set and test set, as we talked before, 80% training, 20% test. And then you try to optimize the weights of the neural network. So basically, you are training the neural network for solubility prediction. And when you do training, what you're doing is updating the weights or parameters for the model, which then becomes your model. Like set of weights is your model, and then you can predict properties. So these are the parameters of the model, right? And now you see the connection now. There's something called hyperparameter, which is beyond these parameters. For example, in deep learning, there are a few settings that your deep learning architecture itself has, like how many layers do you use in deep learning? what is the width of each layer, or what is your learning rate? How fast do you learn or change weights? Or what is your activation function? So these settings of the neural network itself are called hyperparameter. And then you can tune, because if you try different hyperparameters, so like different learning rate, you might get a different neural network which will perform better or worse. So you don't know at the start which parameters of the modeling technique itself will result in a better model for your problem. So you run multiple parallel runs with different parameters for the model. So different learning rate, different architecture of the model, different number of layers, and different size, different width of each layer in the network. So those are the hyperparameters. You try different runs and you tune them, and then you find one set of hyperparameters which is good. You use those hyperparameters to then train your model to optimize the parameters of the model.
Okay, wonderful. So I myself have one question about this stage 2 that you mentioned in your project. So this is related to the results that you got. You showed improvements of something 0.5 to 2.5%. So is this something that you were targeting, or you got more or less than what you were expecting?
That's a wonderful question, and I'll first qualify by talking more about that results we have got, and then about what criteria we had. So those results I showed was the average results across thousands of endpoints. So each pharma partner had multiple endpoints, multiple targets, and each target had classification models as well as regression, some of them regression and so on. So ADDMC and their panel models like kinases and so on. So there were thousands of endpoints that were predicted, and this was the average, right? So what I'm trying to say, there are some models that performed exceedingly well while others did not, and rightly so, because there might not be enough data with different pharma partners that can improve your model. Maybe each is contributing just 2 or 3 or 10 data points for some of the quite obscure target. So there's a distribution of prediction improvements, and this analysis going on, which sort of models perform well. In the end, in a pharma company, wouldn't really employ all those 6,000 models. We would see which worked really well, etc., and we employ that. Right. Saying that, the 1% was set as a target in the first year to meet, because that was a POC, and then in the second year it was go beyond 1%, but then see which particular domain, whether it's ADDMC or panel model, that gains the most improvement. And that's where we have achieved quite significant improvement in ADDMC, but across the board we have got more than 1%, which was one key metric.
So you are saying that there are some models and areas where you might have got 10% improvement.
Sure. And again, I want to remind that the year 2 was to show the superiority, not by a larger margin, but year 3 is the actual run where we will produce models that can be used in production setting that improves the prediction accuracy further. That will have a longer training time and some method improvements in it.
Yes, another benefit is that you mentioned that most of this basic code is there on GitHub, so it is freely available to young researchers and other teams to explore. Is that true?
That's true, yeah.
Okay, great. So once again, are there any questions from the audience? Last chance. Okay, with that, I would like to once again thank you, Prakash, for spending time with us and helping us understand what is happening in the big pharma, how they are collaborating together. So thank you very much. And so we have some time before the next speaker joins, and thus we would like to take a short break. Please do remain with us. We will be back shortly. Thank you. No, wait, let me introduce you.
Okay, all right.
So welcome back to all once again. So our next speaker is ready. We have with us Dr. Jaimin Majmudar. He is currently working in Pfizer. So he has done his master's from Naipur Mohali and did PhD from Purdue University. And he is a research fellow at the American Heart Association, University of Michigan. In 2020, he was chair of the chemical biology in the hub, and he is also board member on the organizing committee of that event. So today he is going to talk to us about the role of chemical biology in drug discovery. With this brief introduction, I hand it over to Jaimin. Please start.
Right, thanks a lot, Vaibhav, for the invitation and the kind introduction as well. Today, I would like to sort of talk about the field of chemical biology and the medicinal chemistry, chemical biology continuum as well, and its role in drug discovery. Chemical biology is a rapidly advancing field with a bunch of new techniques and technologies assisting in the effort for drug discovery. And given the kind of challenging targets that we have to tackle in this new era of drug discovery, it is imperative that we understand both the target and our molecules to the best of our abilities. And that is something that what the discipline of chemical biology does. And by way of sort of a little bit of history here, let me see if I can advance my slides. Just a little bit about my scientific journey. So I come from Ahmedabad, on the polar west side of where you guys are right now, and was fortunate enough to gain my education in different places of India, and one of them being Naipur Mohali, which was a fantastic time in my life, and I learned a lot, and I thank everyone, all my mentors there. And that took me sort of to get my PhD in medicinal chemistry. So I'm a chemist at heart by training, and that sort of led me to the University of Michigan, which is where I did a bunch of chemical biology research. And I'll sort of get into a little bit of detail on what those things were and how that have influenced what I do today. And I've been at Pfizer since 2016 in the chemical biology group. And really what we try to do is understand our molecules better and what are they doing in cell systems. Now, small molecules are very interesting things because they generally engage with proteins. And I use that term a little bit carefully because we know that there are small molecules that do engage with DNA and RNA and other living systems. But proteins, as you can imagine, perform a diverse functions in living systems, some of them being they bind to other proteins. They form these complexes. They undergo signaling or cause signaling events, and they will perform chemical reactions. They get modified and decorated in different ways, things like phosphorylation, a polar group like phosphorylation, all the way to a very hydrophobic modifications such as palmitoylation or prenylation. And these proteins are not static. They get degraded. They get resynthesized. Every protein has a half-life. Every protein has a different half-life in a different cell system and might be different in different species, which is why it's very difficult to find out what the lowest common denominator is for all these things. They do interact with drugs and small molecules and have physiological function. They affect gene transcription and are very important for life in general. So given that proteins are such critical players in the biomolecular world, it begs our attention that we pay closer attention to how our molecules interact with these molecules and what these biomolecules do in general. This is further complicated by the fact that the proteome is an incredibly complex and diverse set of biomolecules. You can imagine you have about 20 to 25,000 genes. They undergo transcription, and you get different transcripts. Which, some of these are nascent. Some of these generate small proteins, which we don't quite understand yet. But every protein also gets modified by post-translational modifications. Let's see if I can get the laser pointer. Okay, you can see that. The post-translational modifications completely change the proteome in a way that imagine a protein that can only have two modifications, a phosphorylation state and a dephosphorylated state. And this protein can only exist in one of the two forms. So you only have two forms of the protein. Now, imagine a protein with two post-translational modifications, and you're left with a protein that can be in 4 possible states. And this number is powered in the sense it's 2 to the power n. A protein like GAPDH, something that we use in Western blot controls, has to at least known to have at least about 32 different modifications. So there's a potential for GAPDH to exist in 2 to the power of 32 different forms in the cell. Which one of these forms is the most predominant one? Which one of these is the biologically relevant form? Which PTMs work together or work against each other? And these are the kind of questions that we really don't have a good grasp on at the moment, just because I don't think the computation is powerful enough at this point to even model these types of questions. And perhaps it is unlikely that all those forms of that protein are critical for biological activity. But again, these are things that we don't quite fully understand or appreciate at the moment. So that sort of brings us to the question is, can we study these proteins? What do they do in living cells and systems? How do they interact with each other and small molecules? Can we learn biology through these chemical tools and approaches? And can we apply an organic chemistry mindset to study these proteins? And can chemical biology and the C and the B are equally capitalized because the chemistry behind the biology and the biology that is inferred from that chemistry are both critical? And how can this discipline be a useful cog in the wheel of drug discovery? So that is hopefully what I would like to touch upon today. And in brief, I mean, so what is this? What is chemical biology? And sorry, I'm trying to get to my animations here. Yeah. Okay, that's how we go. All right, so this is the crystal structure of the M-Pro protease from the SARS-CoV-2 virus. And as you can see, there's a bunch of reactive amino acids such as cysteine and histidine, which form the catalytic site. And then you have some of these acidic residues which help bind the substrate. So there's rich chemistry inside a protein, and this is not unique to the M-Pro protease. This is common across most proteins that you see. And you have these binding clefts, you have binding pockets that we so like to model and get further information on what kind of small molecules can be in those pockets. So is chemical biology biochemistry, biological chemistry, medicinal chemistry? It's a bit of all of the above, but at the end of the day, it is a mindset where we would like to understand biology at the molecular level. And you could use the tools that are available to you or invent new tools, and ultimately be able to study the xenobiotic living system interaction in as granular a detail as humanly possible, such that it can explain biological phenomena that we observe. So that sort of, in a nutshell, is what chemical biology is. And to provide an example, this was some of the work that I did as a postdoc. We wanted to understand certain protein modifications. And could we leverage organic chemistry to understand these protein modifications? So at the time, I was interested in cysteines. These are just 4 of the modifications that cysteine is known to undergo. It can oxidize to sulfenic acids, which is the SOH form up here. And this can actually go ahead and further oxidize to a sulfenic acid, which is an SO2H, and that can eventually get to a non-reversible stage of a sulfonic acid. It can undergo glutathione, self-hydrylation, and nitrization. And of course, it can undergo palmitylation, prenylation, granulation, and things of that, which was part of the focus of my PhD work at the time. But this was during my postdoc, and this nitroso modification really caught our attention because there was a paper that came out of Sol Snyder's lab where they showed that there was cycling of a specific cysteine on a protein where you had palmitylation and nitrization playing out in tandem. And depending on what state the protein was in, it had a different biological function and a different localization around the neurons. So this is what piqued our interest. The methods to study nitrization at the time were pretty sparse and noisy. We wanted to see if this S-nitroso group, which looked inherently somewhat reactive, it could potentially, you have both electrophilic and nucleophilic centers around that functional group. And we tried to go through literature and see if there were any organic chemistry reactions that a nitroso group partaked in under aqueous conditions that we could then use to study this on a protein level. Because again, if you want to study proteins and modifications and do chemistry on proteins, you want that to happen in aqueous buffers. Out of surprise, and this was really a surprise because I was not expecting much there, but I guess they're super old chemistry that I think I really like to go back and appreciate at times. So this was a paper that had come out in 1980s by Terence Hart, and this is, I don't know if you can read pretty clearly, but it says S-phenylsulfonyl derivatives of L-cysteine and glutathione. And what this translated to was that a nitroso group can react with a sulfenic acid, so the SO2H. So I've shown the SOH, but if you have the SO2H, the SNO and the SO2H can react with each other to form these thiosulfonates. What we were able to do with that is so you have a reactive oxygen species acting on proteins to form the sulfenic acids. You have reactive nitrogen species reacting with thiols to give you S-nitroso proteins. But you can take a sulfenic acid conjugated to a biotin and do this reaction in lysates under PBS or phosphate buffer conditions, and you can get adducts such as the protein being modified with this thiosulfonate. You can do the converse. You can have proteins which are sulfenic acid modified, the cysteines oxidized to a SO2H, and reacted with a nitroso group that is conjugated to a biotin through a glutathione motif. And you can show that these proteins react with this handle, and now you've enriched, and the biotin lets you enrich for some of the basically perform an enrichment of the sulfonylated proteins and the nitrosated proteins. We were then able to go ahead and show that you can create, do a stoichiometry analysis comparing these to the total protein levels as to what proteins are more nitrosated versus which ones are more sulfonylated. And in this case, we see that proteins such as thyroidoxin don't necessarily have a preference. They do react with both reactive oxygen species and reactive nitrogen species, which is not surprising given that they're very reactive thiol-containing enzymes. But one of the examples that really drew our attention was this enzyme called PARC7 or DJ1. It has been implicated several times in various neuronal diseases. But this enzyme, this protein has 3 cysteines, and we were able to do this on a amino acid residue level doing site ID work here. And we found that PARC7 was heavily nitrosated, but also reasonably sulfonylated. We were able to essentially, when you purify these enzymes, this comes out as a sulfonic acid form, 1-cysteine-56, if I remember correctly. And we were able to show that the enzyme not only can be purified as a sulfonic acid form, but it also can react with a nitrosoglutathione, at least in a test tube or in an Eppendorf tube. We do not know if this happens in vivo, but the suspicion is that a sulfonylated protein may be able to react with so either nitrosated proteins to form protein-protein adducts, or even react with nitrosoglutathione, which is produced as a species during oxidative stress. So again, these are the kinds of leveraging fundamental organic chemistry principles to understand protein modifications and really improving our understanding of proteins in general. So this was some of the work that we had done as my postdoc, and then again, the key takeaway is that the fundamental organic chemistry principles, we can apply them to study biological systems, but so what? And that's all good, but how does that help drug discovery? And while drug discovery involves several disciplines and involves lots of other things which are critical, some of the key metrics for any successful discovery program are to know whether the small molecule is exposed at the site of action, whether it engages the target that we intended it to engage with, does it cause functional downstream pharmacology, and does that pharmacology translate to a relevant phenotype? So these are the 4 pillars of drug discovery, and this was published by Mark Bunich back in 2013, almost 10 years ago, but this still holds true. And where chemical biology really shines in the drug discovery paradigm, apart from helping out with early target understanding and protein understanding, but it's really around the piece of target engagement. And here I would like to highlight this activity-based proteome profiling a little bit to give you a further understanding of how we use these chemical reactivities to better get a gauge on target engagement and even do target ID work. So what is activity-based protein profiling? I mean, you have, this is a generic kinase here. You can see the DFG loop, but a protein, let's have a protein that is in 2 states that are in equilibrium, and you have an active state of the protein and the inactive state of the protein. Ideally, we would like to be able to discern how much of the protein is active versus how much is inactive. Now also imagine that some disease state pushes the equilibrium to the active state of the protein, and that is what is disease causing. We only want to study the active protein. Is it possible to do that? Can we tag the activated protein using leaving behind the inactive states, essentially just perform chemistry on the active state of the protein, not on the inactive state? And to an extent, we can do that. But can we do that in cells, intact cells or lysates, not just on isolated proteins? And can we isolate the activated form over the inactive form? That's a critical question. And once we've isolated, can we accurately quantify this isolated active protein and better understand disease phenotypes? So this is sort of the core fundamental principle of activity-based protein profiling. And so what do we need for this concept to succeed? And again, this is the same protein that I'd shown before, but this generally works better with proteins that have nucleophilic residues. Although more and more target engagement probes can be made even if it's not an enzyme, and there are ways we can get to that, but that's a separate talk. So essentially, if there's a nucleophilic residue inside the protein active site and a binding pocket, can we design complementary reactive warheads that will react with the nucleophilic residue in your enzyme or protein? Do we have a binding component, so something that brings in the Ki component of binding to this molecular framework? And finally, we need a reporter, something that either helps us with gel fluorescence or a biotin handle that we can then enrich and do mass spectrometry work on. And this concept was really pioneered by Ben Cravatt at Scripps, and this is something that his lab has taken by leaps and bounds, along with folks such as Matt Bojo and several others in the field. So with that, this is sort of the concept of activity-based protein profiling, and what you get at the end of the day with the warhead, a binding component, and a reporter is an activity-based probe that can then be used either in cells or lysates to interrogate biological activity of proteins. Now, can this be done for an entire protein family? And the answer is yes. So I'll take lots of steps back here before we dive into some data. But serine hydrolase activity-based probes are really, they've been known to be around in literature since the 2000s, when Ben Cravatt published this work early on at the turn of the millennium. But it all stems back from some of these molecules, such as physostigmine, which are essentially anticholinergic molecules. And these molecules have a fantastic history, which is what I really got me interested as well at some point, was probably a few thousand years ago or a few hundred years ago, when if you go back to a time in Central Africa, where if folks are accused of being performing witchcraft, and if they were accused of that, they were asked to grind up these calabar beans and drink that milky white liquid that comes out of it that has a significant amount of physostigmine in it. And if they survived the ordeal, they were essentially released. And if not, they succumbed and died because of physostigmine toxicity. Now, this was something interesting, just as a side history, but really the molecular mechanism of action of these molecules, such as physostigmine, which is now it's an approved drug in low concentrations for treating glaucoma, but a bunch of work done on this molecule over centuries really showed that this was an anticholinergic molecule. And in the 30s and 40s, one can imagine there were molecules that were being investigated, such as this diisopropylfluorophosphonate molecule, which had anticholinergic effects and central excitant properties and things of that nature. One can only imagine why such molecules were being investigated at the time. These are potent neurotoxins, and this was the 30s and the 40s. That said, such molecules can be not this molecule itself, but molecules that have the chemical characteristics and the reactivity of a diisopropylfluorophosphate can really help us understand serine hydrolases. These are anticholinergic. And the mechanism here is pretty well known, well established. You have the active site serine that gets coordinated with the histidine, nucleophilic attack on the peptidic backbone, get the collapse back. And once you have this covalent intermediate, that's the first reaction product, and then the water molecule leaves. The idea here is, can you make this specific step irreversible, where the collapse of the density and the eventual release of the enzyme back to its active state is prevented, essentially prevent this reaction from happening. And that is what diisopropylfluorophosphonate does. Now, this is a relatively unstable molecule. It's a potent neurotoxin. So how can this be transformed into a tool to study a whole family of serine hydrolases? Well, the breakthrough really came from the phosphate to going to the phosphonate here. And this is somewhat of a tuned reactive. It's still pretty reactive, but it has fine-tuned reactivity. The additional PEG and different types of linkers do give it some more stability, so it's not as unstable anymore. And you can put an enrichment handle on it, and it has a potential to react with several different serine hydrolases. Now, this molecule itself will not react with all of them. There are several modifications of these fluorophosphonates that are now available. And these probes essentially have helped enrich our understanding of serine hydrolases, and several modifications are available. We published this work, say last year or a year before last. We've improved the synthesis on this as well. But so what, right? So the question is, can we use this to study enzyme activity in cellular differentiation? Now, adipose tissues, adipose cells store and release energy. They dissipate energy, generate heat. The brown adipose and the white adipose, these form from the adipose duct stromal cells all the way to pre-adipocytes, and then finally convert into mature adipocytes. And this big yellow blob here is the lipid droplet that sits inside these adipocytes, and they essentially store all the lipid that is in there. You don't want this lipid to go out of the cell and have an ectopic distribution of lipid, because that can actually cause a lot of disease. As you can imagine, because these are lipid storing cells, these are implicated in a variety of diseases. But this was a work, again, I think pioneered by Ben Cravat here, where you can see that you were able to use these fluorophosphonate probes and label the cells from according to how they're differentiating. So they're going in from the pre-adipose, the stromal cell to the pre-adipocyte to the mature adipocyte. And as they differentiate, each of these bands on this protein gel and read out by fluorescence is a different serine hydrolase. So certain serine hydrolases increase in abundance over time, as is seen with a couple of these, and there are some that actually decrease over time. So the expression and activity, so you're only reading out on the active form of these serine hydrolases at this point, and there is a homeostasis going on as cells differentiate, different serine hydrolases get expressed, and all of this is fairly well controlled transcriptionally, and it's a differentiation program of these adipocytes. You can go ahead and do some mass spectrometry work on it to find out which, you could do an experiment on day 0 versus day 10 or day 15 and figure out which serine hydrolases were expressed. In this case, it was fatty acid synthase and some of the CES enzymes that were changing the most over time. So again, this is the way to understand biology and what enzymes are functional. And maybe the pre-adipocytes are something that you would like to target for a certain disease, and then you know what is your universe of enzymes that you would like to target for a certain indication. But how does this help us discover new targets specifically? So type 2 diabetes can, again, is known for insulin resistance. And one of the effects during diabetes is this increased plasma-free fatty acids, which can circulate. So they don't stay in adipocytes anymore. There's ectopic lipid deposition on top of the adipocytes, which then essentially releases into the plasma. Now, can we find molecules that reverse this phenotype? And that was a phenotypic screen that was run. And as I go next, as you can see here, that these are the pre-adipocytes on top and the adipocytes at the bottom. The yellow indicates a higher abundance of these serine hydrolases. The lower abundance is the blue, but you can see that the profile changes as you go from pre-adipocytes to adipocytes. This in-gel fluorescence is an experiment done to screen at the adipocyte level once they are fully differentiated. And from the screen, these were some of the molecules that essentially did give the phenotype of interest, that reversed the phenotype of the ectopic lipid deposition. And what you're looking for is that when you do a competition experiment with these small molecules, and then go in with the fluorophosphonate, a lack of signal in one of these bands suggests target engagement on that serine hydrolase. And using mass spectrometry to further deconvolute, the researchers were able to figure out the targets of some of these active molecules. And again, this is sort of what the screen looked like. You had vehicle treated. There is no, again, these are insulin resistant cells, so there is essentially no lipid that stays inside the cell. This is what you want. And upon treatment with rosiglitazone, the lipid kind of starts staying inside. But then some of these molecules do the same thing, only when you have a carbamate and not a urea. So this was suggestive of the fact that This was a covalent reaction with a serine hydrolase, and that was how the target deconvolution was done. Again, and the concept here was that you had DMSO-treated cells or vehicle-treated cells, and with this covalent serine hydrolase inhibitor, and you could go in with the fluorophosphonate. So if your site is preoccupied with your molecule of interest that is active, the fluorophosphonate will not be able to react, and the difference in activity is what you read out. So again, this is the loss of signal that you see, which you interpret as target engagement here. So this was one application of it. The other question is, so this was a target ID effort and understanding biology, but then do these inhibitors modify the target as intended? So these are, again, two reversible inhibitors of serine hydrolases, ML348 and 349. They target two serine hydrolases, LYPLA2 and LYPLA1, but can we study target engagement in vivo? So this was some of the work. Again, these were animals that were treated with the inhibitor, followed by after 3 hours with the fluorophosphonate probes, and then the readout was done by in-gel fluorescence. And LYPLA1 and LYPLA2 are all the way down at the bottom, that they kind of show up on top of each other. And you can do mass spec to clear, get better resolution on this, but I'm just showing in-gel fluorescence data here. And as you can see that in the heart, the molecules inhibit. So the top band is LYPLA2, the bottom one is LYPLA1, and the molecules indeed show this competition effect compared to the vehicle treated. And in this case, in all tissues, there was target engagement, but you can do this on a tissue-specific level. And we have seen that there might be tissue-specific differences in target engagement. So these kinds of tools really give us a handle on what the molecules are doing in vivo, and these were reversible, so it just doesn't work for covalence. We've shown that this can also work with reversible molecules as well. So can you develop such probes on a case-by-case basis? Not everything is a serine hydrolase, but can you do this for other proteins? And again, the answer is yes. So this was a phenotypic screen run for an Alzheimer's target to see if to essentially find molecules that reduce the levels of ApoE. And we found some molecules that were based on this template, which did what we intended them to do. There was no target known for these molecules at the time. And Paula in our group, he performed this experiment. This was sort of right at the time, about 5 years ago now, where we developed photo affinity probes. So can we leverage photochemistry to study target engagement? And this is it's a beautiful example because they made this benzophenone containing an alkyne and coupled it to the template. You can do the stable isotope labeling of amino acids in culture to get light and heavy cells. So these have light and heavy arginines and lysines, and then you can put them together and run them on the mass spec, and the differences in their ratios can be computed fairly easily by the mass spectrometer. You could take the light cells or the heavy cells, treat one with DMSO, one with compound, then go in with the photoprobe, which forms the excited state, and then couples up or latches on to amino acids close in its close vicinity. So if you have just the parent molecule and you do a competition experiment, so if the parent molecule is already sticking in the active site, the photoprobe has no place to go. But if you have this DMSO, the photoprobe can latch on, and the difference between the MSO and the compound treated can give you your targets. And in this case, the only target of this molecule came out to be LXR beta, and this was something that we did not want to pursue further, given we knew some things about the target. So at the end of the day, we were able to leverage photochemistry and chemical proteomics to do a very extensively detailed experiment to find out a target from a phenotypic screen. And we've done this for other programs, and this is something which is in our wheelhouse. So not only could we do this, there was also a binding site identification where it was found that the serine actually reacts with the radical benzophenone. And this was validated by getting a crystal structure where you can see that the serine here, the serine's down here, and you can see that the benzophenone is right here. So they're sort of on top of each other, and which validates the mass spectrometry work that we've done. Cellular thermal shift is one of the other ways to study target engagement, which is essentially an ITC kind of an experiment, but done in cells. And this is extremely useful where you get thermal stabilization of the protein of interest upon molecule binding. And this was a pretty large shift of 6 degrees, I think, and that again suggested target engagement. So we were able to leverage photochemistry, physical chemical properties, and mass spectrometry all in one, along with crystallography, to show that the molecules indeed bound LXR beta. So this was something that was published as well. So the question really becomes, like, do small molecules just inhibit proteins? And this becomes more and more challenging because the targets that come out of genome-wide association studies or advanced CRISPR screens are not necessarily enzymes, or sometimes these are proteins that have no known biological activity. They don't, even if they are an enzyme, we may not know their endogenous substrate. And sometimes we don't know, we can find binders to them, but we may not be able to find functional binders to these proteins. And molecules do more than just inhibit proteins. So again, more than inhibition, and one of the classical examples is the thalidomide example, where there's an unfortunate teratogenicity event, which everyone is familiar with. But again, the mechanism for this was not known. And it wasn't until Hiroshi Honda and colleagues really demonstrated that the molecules engage cereblon. And this was a classical chemical biology experiment that was done where compounds were put on beads, cells were treated with the compound itself, and then the lysate was flown over to see competition. And as you can see that in your elution, this thing binds cereblon pretty well along with DDB1. And what these molecules do, what these IMiDs do, is they engage with cereblon, the E3 ligase, and they change the substrates of the E3 ligase such that neo-substrates are recruited. And one of the hypotheses is that IKZF1 and the other zinc finger proteins get degraded, which may be resulting in the teratogenicity effects of these molecules. This has spawned a whole field of heterobifunctional PROTACs, where you can couple a molecule with a cereblon binder, such as thalidomide or lenalidomide, or use VHL ligands or other E3 ligases, and then bind it with a molecule that binds your target and induce its degradation. So this was, again, a very classical mechanistic understanding using the principles of chemical biology and binding at the time, which resulted in the whole field of PROTACs and molecular glues coming up. But the question is, so what? And so what here is that this molecule right here, the fourth best-selling molecule in 2020, is a molecular glue, which is an indication for oncology. So these are fundamental experiments where if we take the time to understand what our proteins do, how they interact with small molecules, what changes, using multi-omics, trying to figure out what else gets degraded using proteomics and RNA-seq and multi-omics, and really leveraging the power of the advanced computation that we have now and the advances that computational biology has made can really help us understand what molecules do on a systems level. And just to put this in perspective a little bit, this is sort of the key observations of how molecular glues came about. This is a wonderful review that was written, I believe this was a couple of years ago now, but the number of publications in the PROTAC and molecular glues field has expanded rapidly since at least the turn of the latter half of the last decade. And this has spawned in, ultimately, Arvinas, one of the companies down in Connecticut that Craig Cruz founded. They started a pivotal trial here in phase 1 for their PROTAC. And sort of it'll be interesting to see where this field goes. But small molecules can do more than just inhibit. They can disrupt protein-protein interactions. They can alter protein half-lives. This is a known phenomenon. Can they drive condensate formation and reversal, the whole field of biomolecular condensates? Can they interact with RNA? There are known examples. And can we use the multi-omics data, which is essentially proteomics done with and without molecule, with and without compound, to study protein half-lives, doing RNA-seq, looking at techniques such as ATAC QuestSeq if you're working through transcription mechanisms or ChIP-seq, and really tie up all that data together to bring to bear new targets and understand sometimes you may not need to inhibit 90% of the protein or not engage with 90% of the protein. Sometimes you just need a 10% occupancy to get you just need to tickle the protein a little bit. You don't need very high binding affinities for certain things, as long as it performs the physiological role that you want it to perform. And these are the ways that we have to be a little creative to unearth novel mechanisms of action. Because as the data are expanding, as the field is expanding, we are learning about novel functions that proteins perform and how small molecules interact with those. So I think that's sort of the key big takeaway that I would like. If there's something that you can take away back from this talk, is that small molecules can do more than inhibition. We just need to look harder and leverage multiple channels of data and really try and make sense of it. So it's really a time where there's a lot of data around from proteomics experiments around binding of small molecules with proteins. There's hundreds of papers out there. All this information is sort of buried in supplemental text somewhere. It'll probably take a Herculean task to sort of bring it all together, but I think that's a challenge for the field at the moment. So again, key takeaways: proteins and biomolecules drive cellular physiology. Some of the impact of some interactions may not be obvious at first, but again, we have to leverage our chemistry and organic chemistry understanding to really try and figure out what the fundamental biology is. And again, it's a mindset to understand protein function at the highest resolution possible, or even, for that matter, even biomolecules, because I'm sure there's a whole field of RNA chemical biology that is out there. But yeah, I think that probably was my last slide. And if you're curious, this is a website that I find pretty interesting, breakthroughs.com. It has several podcasts. It has a bunch of different articles that just talk about drug discovery in general and some of the updates that the industry is working on or is curious about. So I think this is a public resource that is out there. And with that, thank you. I will take any and all questions.
Hello. Thank you, Jamie, for a wonderful talk.
Thank you.
Are there any questions from the audience? Anyone? On YouTube, I guess we have a question. Dr. Rosie, please read that.
Hello, thank you, sir, for the wonderful talk. From YouTube audience, Shweta Sharma is asking one question. I will read it for you. What if protein structure is not known? Then how we can identify the lead molecules? Or if your target is new, then how chemical biology help to find the small molecules?
Yeah, so this is a great question, and it's pertinent because there are ways to do this. So if you go back, what I think I can talk about is the KRAS example. Now, KRAS is something that has been known in literature. People have tried to drug it for a long time. But what really came about was they had the isolated protein at the time. They probably had a structure, but this could have been done even without a structure. Now, the G12C mutation on KRAS is oncogenic. They had a cysteine instead of a glycine. And what they did was a screen with a disulfide library. Kevan Shokat did this work at UCSF, and he was able to use disulfide molecules to find, and then the readout was just by mass spectrometry at the very early stages, if I remember correctly. And you were able to find molecules that latch on to KRAS. What was not known at the time was that, because I don't think that structurally, they were structurally enabled with those molecules early on, but what they found was that these molecules, they bind to KRAS or a protein for that matter, and they open up a pocket. So they induce a pocket, the switch to pocket actually shows up because of the covalent reactivity. And this has resulted in Amgen taking this forward along with Kevan, and this This has resulted in a drug that was recently approved, the Amgen 510 molecule. So it's doable. There is a whole lot of literature around reactive cysteine and reactive lysine profiling. So there are reactive, quote unquote, reactive amino acids in the proteome. So you can interrogate those with small electrophiles, which are generally non-selective, but they can give you an idea about ligandable pockets on proteins. And it's a tough assay to run. It's the data analysis is not simple, but at the same time, I think that's one way to do it. There's other ways to do it as well, but this is one way to do it. Hopefully that answers the question.
Yes, I guess so. So yeah, so let me ask one from my side, that you mentioned about that drugs, small molecules not only just inhibit, but they do a variety of other things. So considering the sheer number of proteins and the sheer number of drug-like molecules and the amount of data that you are collecting, do you see that AI is coming up and it's going to help you at least reduce the task that you should be focusing on, or is it still that is something cannot be easily handled?
I don't doubt the ability of AI or machine learning to help us understand some of these things. I think there'll be a time for that. I think it's still early days, and part of the reason, the challenge is that the data themselves are not uniform. So what do you use to have a training set? Let's say you are screening for molecules that disrupt a protein-protein interaction. And you can do a screen and find that, but that information has to be in the public domain, or at least the screen has to be in a way that if it's a proteomic-based experiment or something along those lines, most of these data sit in discrete pockets and of the scientific literature, and they're not all uniform. So I think the first task is to identify those and sort of bring them together in a way that machine learning can be useful. Because I think that there's a way to do this. The simplest of experiments to do is really just run experiments on the right type, the cell type, maybe primary cells or something, where you do the full proteome, just full proteomics with and without compound. What changes, right? Because it is possible that adding a small molecule to a system changes abundance levels of other proteins. Now, it might be doing that through transcriptional mechanisms or through other mechanisms, but or by directly binding to the protein and stabilizing it. So those are things that I think we just don't have enough data, and whatever data is there is sort of not uniform. So I think that's the first task.
Yeah, well said. Yeah, I guess there is one more question on YouTube.
Mm-hmm.
Please read that.
Okay, sir, there is one more question from Surendra Kumar. He is asking, protein structure changes with minor change, even few degree Fahrenheit in body temperature. Then what strategy we use for design?
Oh, that's a great question. I think so. Proteins are dynamic, right? So you'd assume it would be safe. Well, you can correct me if I'm wrong, but it's sort of relatively. The hypothesis here would be that most of the protein stays close to its most energetically favorable conformation. How would you screen for something that changes with body temperature? I don't think there's a lot of data out there, right? Because if you think about it, once you sleep, your entire core body temperature goes down, and I'm sure that affects protein structure in some ways. But I don't think we know that on a global level. I don't know if MD simulations at different temperatures, if that can even be modeled in a way that. So yeah, I don't know how you would screen for that, but the cellular thermal shift assay might be something you may want to look at in a little more detail. If that, could you do a screen at a slightly elevated temperature in cells and try and get that. I'm a very strong believer of cellular assays as much as possible. Not that isolated proteins don't work, but if you can find a binder in cells, you're more likely to translate that all the way through a drug discovery pipeline. I answered the question, but I hope I give you some tools to look at.
I think at least partly you did. Yes, thank you so much, Jamin, for a wonderful talk. It was a pleasure to have you, and special thanks because you came very early in the morning. I could see you sip caffeine to just keep you up, but you might have lived the Mohali days where we used to go in the lab at 3 to synthesize glycidate esters.
I remember those days, yes, and some of our NIPER colleagues, they're all doing very well, and it's nice to be back in some ways. And thank you for the opportunity. And it's okay. I mean, I normally wake up at 4 o'clock, so today was just a little bit earlier, anyways. It's okay.
Great. So with that, we thank you once again, and
thank you.
Thank you. Bye bye. So with this, we now can move to the next section, because we already have the speaker with us. Hello, Dr. Divya, can you hear us?
Hi, good afternoon, Dr. Vaibhav Dixit, sir.
Good afternoon. So let me introduce you to the audience. Dr. Divya is an application scientist at the National Support for BioVia Discovery Studio, LTM Technologies, and she has done bachelor's in biotechnology from Anna University, master's in biotechnology, and PhD in biotechnology from Vigyan University, Guntur. She has expertise in molecular modeling of proteins and transmembrane structure and ligand based pharmacophore based drug design, molecular dynamics, and simulation. And she has earlier done work as application scientist for life sciences as Para Innovations. And today she is going to show us a demo session on using the Discovery Studio module. So over to you, madam.
Yeah, thank you so much, sir, for a brief introduction. And it's my great pleasure to be with you all today in this virtual platform. And I believe that today's session is going to be, even though it's the late evening, the session is going to be very interesting for you all. As I have heard about a previous speaker session where he was speaking about a molecular dynamic and simulation as an extension, how we can perform this molecular dynamic simulation of various temperatures or by introducing some different changes in pressures, how we can encounter and see the variations in the cells and the conformation changes. And after that, we are also going to have a little bit going to add this agenda regarding a QSAR, quantitative structure active relationship. Let us not wait and we just keep moving on and see about how this molecular dynamic simulation can be done using a software called BioVia Discovery Studio. Let me share my screen. Sharing my phone. I hope everyone, sir, can you see my screen? Can you just confirm me?
Yes, we are able to see.
Yes, thank you so much. Thank you. Before that, I would like to introduce my company. So ADDMC Technologies is a partner of Dassault Systems, PLM Solutions, Statuses, and for 3D bioprinting, and Artex 3D for 3D scanners. And we have a complete solution for CAD, CAM. Yes, sir. Are
you sharing a PowerPoint, or it's just the Microsoft Teams window?
No, I'm sharing the whole screen.
We are just able to see. You need to reshare probably.
Yeah, right now?
Yeah, now it's visible.
Yes, thank you, sir. Thank you.
Yeah, please continue.
Yeah. I'm very proud to introduce my company, Altem Technologies. It's more than a decade company who is partnered with the Dassault Systems for a PLM solutions and Statuses for 3D bioprinting technologies and Artex 3D for 3D scanners. And we have a complete solutions for CAD, CAM, and then for 3D bioprinters and 3D bioprinters technology is also part of it from Cellink. So one of the wings in life sciences has been taken by on a BioVia Dassault Systems, Discovery Studio, Material Studio, and Pipeline Palette. We have headquartered in Bangalore, and we have a various sales offices across the Chennai, Hyderabad, Pune, Kolkata, Mumbai, Chandigarh, and New Delhi. So with a various team experts, consultants, application engineers, application scientists, designers, and domain experts and software specialists are a part of our company. You can see here our one of the wings of BioVia Life Sciences is in the BioVia Discovery Studio and Material Studio. And you can see our esteemed awards has been given for us our technology. Moving forward, so dynamics and simulation is a quite very interesting topic, and everyone wants to do the dynamics. Once the people do the drug discovery process, and when we end up with 10 to 15 drug compounds or lead molecules which we have, among them, which I want to choose for my in vitro studies, or which could be potentially can be follow the stable conformations, and how the behavior will be there when you want to run nanoseconds or milliseconds like this. So for this, we should have a certain working protocols, which many of them have been very well known. But according to BioVia Discovery Studio, the user-friendly software and more than 3 decades of integrated with a charm technique and also the various force fields of charm will help you to drive your force dynamics without any constraints. Part 1, we are going to look at about protein preparation and theory behind the force fields. So first of all, we are going to have set up the system preparations. So which system you are going to have? You could choose any systems. It could be an DNA, it could be a RNA, it could be a protein or protein complex, protein DNA complex, whatever the systems you want to study and to know the variations as a function of time and energy, you can do that by using Discovery Studio. Then there comes often a significant role of energy minimization. So every steps will always start with the energy minimization. Then we are going to select an appropriate force field. Then we are going to make an either we can use implicit solvent or be explicit solvent conditions. And both the solvent conditions are part of Discovery Studio. So this is how the exact pages of Discovery Studio will look like. This is the Discovery Studio client. And we have an enriched protocols for doing a molecular dynamical simulation, feed molecular dynamics, NAMD, and free energy permutations. And finally, one can do trajectory analysis along with the radial distribution functions. So we have enriched tools in that, and users have to choose according to their application. One can pick what kind of an applications they require, and they can drive the protocol in such a way. So the basic concepts, it's very well known. It's behind of quantum mechanics and molecular mechanics, which includes the function of first order principles of mechanical techniques, and which uses basically a wave function. The wave functions are very well light and very accurate and could measure the bond breaking and formation. So which is a crucial part of molecular dynamic simulation. Molecular mechanics, which employs a definition of an atoms. Each and every atom point charges with the defined van der Waals areas are very crucial when we do dynamics. And it includes various assumptions of classical mechanics interactions, force field parameters. So now when I have lots of forums are trying to say about it, there is a problem in a force field. I cannot apply the specific force fields to the specific, if it's non-standard amino acids, I cannot apply the force field. However, the special functionalities in order to not to stop the research process, we can edit the force field parameters in Discovery Studio. And each and every function atomic positions can be captured, and you can also calculate for larger systems also. If you have very big molecules and packed with the water molecules, you can do that. And you can also identify by means of molecular mechanics the bonded and non-bonded interactions and accuracy between them. Let me start. So every dynamics will begin like this. First, starting with force field parameters and energy minimization. So there are some problems like when you want to develop an X-ray crystallography like similar structure by using an homology modeling technique, it is having lots of constraints, and we want to meet exact like an X-ray crystallography or experimental data. For that purpose, we can use energy minimization. The minimization should not be applied as such because it will affect the overall backbone of the structure also. So where, which amino acid or which location, everything needs to be identified. Then only we are going to apply the energy minimization. To do that, we have a fixed atom constraints, harmonic constraints based tools where you can fix your backbones and before running your energy minimization. Moreover, the main, I could say that purpose of this minimization is to relax the conformations and to remove the steric overlap that is produced in the system. That's what, as I mentioned, so when you have an absence of any experimental structure, the ideal geometry to do is the energy minimization technique. So we have all different types of energy minimization techniques, right from a Stephen descent, which is a slow convergence method, and the conjugate gradient. It is a more iterative, which uses a previous history of minimization steps. We have a Powell, the variations in conjugate gradient method, and smart minimizer. Say my structures having higher positive potential energy, I would like to bring in a one rand to by applying a fixed atom constraints, I want to bring my structure to have a stable conformation. So I can use smart minimizer. It will do fast convergence to achieve your local minimum. And because of its robustness, it will be a default option in most of the protocols of discovery study. How it will perform? So you have a control with the steps also. How many steps you want to do, where you want to stop your dynamics, where you want to stop your energy minimization. Everything, all the options are under your hand. You can pick up as default options, or if you want to do some changes, of course, that's how the research goes on by changing each and every options. What could be the outcome of that? So you can change that RMS gradient tolerance, and then you can run the process. So it's recommended mostly when you have your very poor conformation. So when as it comes out, the very important concept of your molecular dynamics is a force field. When we try to take in any system, when you apply the force field, if you're not getting done, we cannot proceed further with our dynamics process. So it's always have an extension of .ffml format, and each and every atom has its own force field type parameters. Depending on the system, one will apply the specific force field. So for that, we have a various ranges of force field parameters. So usually, how it is actually expressing in mathematical form, the bonded energy of is equals to bond angle, torsion, improper cross terms, and the non-bonded terms. And the force field differs in the cross terms, specific mathematical forms, and then you can also have methods of parameterization, and you can specify some parameters also. The intermolecular term, stretching, including which will be calculated using these formulas, angle bending, and rotational bonding. And impropers out of planes and uniparallel cross terms, and then the cross terms of protein backbone, C-amp corrections. And also one can calculate Coulomb's law, Lennard-Jones potential for van der Waals interactions. These all the intermolecular term will be quenchable when we go apply the force. This word. Atoms, very important, each and every atom, because when you zoom your protein is also your atom. It contains CH&OS, so all the system is made up of CH&OS. But the location where the specific structure of forming have its own particular atom different environment. So you can see your carbon in aromatic ring has a different name, or have a different name in atomic position in the force fields. And aliphatic of carbon of 2 hydrogens have different one. And aliphatic sp3 carbon with the 1 hydrogen has a different name. And as same as for carbonyl carbon also. This is how they see. This is the same carbon, but however, the force field topologies will be varying. We call Ca, Ct, Ct1, Ccc like this. So that's why the atom parameters are very important. The force field parameters are very important. So when you have a structure, all the force field parameters must be captured in your topology file. So usually that will be in RTF file. It has been the same as like an open source software also, and even Discovery Studio. So the new SHARM 36 force field as contains for all kinds of force field parameters. If you want to try it for your lipid systems, for ions, nucleic acids, carbohydrates, ethers, and then for various kinds of biological systems. So SHARM 36 force field as integrated within different atom types in Discovery Studio. So choice of force fields. So we have these ranges of force fields in Discovery Studio. It's what we have flexible protocols with this. So if you have if you're trying with a SHARM 19, which is usually done for protein and peptides, including SHARM 22, and SHARM 27, it can be used for nucleic acids and lipids. And SHARM 36 is recommended for protein, nucleotide, carbohydrate, lipid, and C generation force fields are there. And we also integrated with the commercial force fields. And mostly when you see your SHARM M, it's a heterogeneous force field. It is applicable for all the systems, including nucleic acids, DNA, and RNA also. And we have a MMF force field for organic and drug-like molecules, and CF force fields for all biological molecules and small molecule systems. It's for all atom models. The choice of force field, it's depend upon the user. So we have given you the things, which force field you want to use, that you have to decide. And then accordingly, your dynamics will be driven. So then we are speaking about the two synonym, I could say that. First synonym, I was telling about energy minimization and then force field. The third one, which we'll be always hearing in the molecular dynamics, is the solvation. How we are going to build your system with the water molecules. Either you can use explicit solvent condition, or you can use implicit solvent condition. It is up to your user. So we have two different models. You can have a solid molecule system in a periodic boundary condition, or you can use it in a vacuum electrostatic interaction space where we have a continuum dielectric methods. You can use implicit solvent conditions. So by doing this, we have various models. We have a generalized, all different kinds of generalized models or implicit solvent models of in the part of Discovery Studio. You can select one. So mostly they'll be recommended to support for the GBSW, and it's a molecular simple switching, and IBM will be support for implicit membrane systems. If you have membrane proteins and you want to do that, and if you want to run this, you can do it by using this method. And accuracy and distance-dependent dielectrics, generalized method, Poisson methods vary in accuracy with applications. So this is the implicit solvent models where we have to use and how we have to use. So if you have point energies, and if you have a minimization or molecular dynamics. So implicit solvent in dielectric or have a real accuracy level is a little bit poor. But when you look into an GBMV and then GBSW and TBSA, so if you see that the accuracy levels are very high when you implement with the implicit solvent conditions. It could be applicable for an point energies minimization as well as for molecular dynamic considerations. This is how they will try to say what you give your systems, and then select your, either you can do it for a whole system, or you can add in a spherical boundary with a center of mass for harmonic constraints for some kind of a system. So it's that's what you can choose either models. So all the models are inbuilt. Which one you want to choose, you can select and you can run your process. So just we have taken only about after preparation of proteins and applying your force field and energy minimization. Now we are getting into an actual process of molecular dynamic and simulation. How we are going to perform this process and how we are going to analyze our trajectories. Trajectories and analysis are very crucial. Only then we can see how accurately we run the dynamics, whether because it is a temperature and pressure dependent. So how changing in various temperatures will have a greater impact on your dynamics. So to perform the energy minimization, we have first order methods, as I mentioned. Second order method, we have ABNR method. It is also part of it. You can choose any method. So including a Powell methods. Apart from this, we have ABNR, which is the determination of a Hessian matrix, which is used for information of PSS curvature. You can use it to study the transition state searches. Adapted Newton-Raphson method. This is the second order method. The Powell is also available in Discovery Studio. The method doesn't require any derivatives. It will be used for small molecules. So then how you are going to apply? So anyhow, I will show you a demonstration, but I'm just putting over here. So under the tools menu, we have these options. So minimization, then you click energy minimization. Just you can clear a geometry of your protein and you can apply the energy minimization. We have a standard dynamics cascade steps. We have individual base steps which can run in a CPU mode as well as in GPU mode. Either way, you can run this process. So if you don't want to perform the dynamics step-by-step process by minimization, you can choose standard dynamics cascade steps. Initially, you can run with the cascade dynamic steps, including with the 2-step of minimization, and then further heating the complexes gradually from temperatures from 0 to 50 Kelvin, or if it depends on user, if you want to bring changes in the systems during heating, you can make it. And equilibrating the systems, and then a production, we have inbuilt with the 3 different kinds of enzymes: NPT, NVT, and then NDT. ND is very rarely used because it depends upon only energy. NVT, NPT are commonly used. So you can see here in all the protocols, you can have this algorithms will be there in the default, and smart minimizer will be a default option. But you cannot see the smart minimizer in the standard dynamics cascade because we are applying already 2 steps of minimization. That's why we don't have this smart minimizer. So performing your molecular dynamic simulation. We know that the molecules are not static. All our biological systems will try to move from the one position to another position, and there will be a dynamics will be created, and there will be a changing in position and conformation or a frames as an impact as a function of time. So we can, during the dynamics, we can read about a simple vibration to produce spectra, breathing of DNA, heme binding, allosteric transitions, and protein folding. So all these parts are dynamics, which we can do that. So the principles of molecular dynamics. It is time evolution. That's what I was keep on stressing to tell about as a function of time. So if you can examine each and every atom movements over the conformation space as a function of time. Small changes. What is the main advantage? Even small changes which is occurring in the atom that can be captured here, and you can see the differences. What is the distance? Everything you can do it in a conformation space using this kinds of methods, and it offers you the best dynamics pictures with their motions. And ignoring electronic movements and calculate energy as a function of nuclear positions. It uses Newton's second law of motion on the energy description part. Very well known phenomena. Along that, kinetic energy will be calculated using this formula. Boltzmann constant, including temperatures. So at a higher temperature and dynamics or need to be taken care well before running a dynamics. At a higher temperature, atoms move more and try to cover more conformational space. That's why the higher temperature dynamics used to overcome the barriers to explore the new local minimum. When you heat your systems very higher, and when you want to equilibrate, the time taken for equilibration will be two-folds higher than the when you are giving for the heating. So that has to be taken care. How much temperature, at which temperature your system needs to be done for the molecular dynamics and simulation, that user has to decide by looking either into paper. In case if you're not sure about it, if you have new proteins, of course you can run up to the equilibration. From the equilibration, you can take the average temperature values and you can set that temperature as for the final production analysis. That you can do that. We have that option to perform. And make sure you have to, when you have increasing your temperature, there is a possibility of secondary structure changes. Transition will occur usually. There will be helix, it could turn into a turns or coils. And there is a peptide bonds from a cis transformations to trans isomerization could occur. So make sure when you run your temperature has to be taken care when you run your dynamics. Now experimental setup. How we are going to set up this molecular dynamic constellation. The system is very simple, but input parameters that need to be addressed with carefully. Select your any system. So I have mentioned here only a protein ligand complexes. We have DNA complexes also. We can do that. And apply your force field. After applying your force field, solvate your systems. Solvate your systems. Either you can use implicit solvent, or explicit solvent condition. And do the minimization, heating, equilibration, and production. And finally, analyze your trajectory, RMSD, RMSF, and radius of variation. RMSD can be calculated for backbones and even for like if you're doing for a protein ligand complexes, you can separate your protein and ligand and you can run separate RMSD also. You can run it separately for your ligand and you can analyze. Either way, you can do this process. So, dynamics cascade. So initially, it starts with a steep and distant, and then where you try to remove the poor contact systems. And second, minimization. It ensures that initial poor contacts are revealed, and the system is having low energy point to sort your dynamic stages. Then followed by a heating of the systems, gradually heating from systems to reach a target temperature. So the next is equilibration. The main purpose of this equilibration is to ensure that all the atoms are trying to attain degrees of this. If you're increasing the temperature, then time taken by equilibration will take little bit longer than usual actually. Then finally carry out the production in a suitable thermodynamic ensembles like NVT, NPT, or NV at a given temperature based on equilibrated systems. So you can take up from the equilibrated systems, they can give a plot for that equilibration. And after taking that energy temperature plot, you can take an average and you can see the deviations in that. In that, you can fix which temperature you have to give it for production. This is how we have to do this. When you're running the process, so for example, I have not done the production. I just started my production and ended that. So after equilibration, I want to run. So I'll be end up with this two graphs. When I see this graph, temperature versus time, and you can see there is a lots of ups and downs. It goes like this curves. You can see that there's a more temperature, and when you take the average, you take the average of that. The average of the target value is around 299.3 Kelvin. You can put it as a 300 Kelvin as your dynamics temperature, and you can run the process. And you can also see here the energy seems to reach a constant values right from the beginning. There is a drastic change and try to reach it. So as a function of time, how the variations are occurring in a system, you can, each and every step, each and every atom, step by step, confirmation by confirmation, you can able to capture here. So equilibration check will help you a lot before running your dynamics. So you can also get this one when you have a temperature, at which temperature it was run, what are the changes occurring, all those things can also be captured. So consolidation before running your dynamics, be aware of your code, what we are going to answer, what questions you are trying to answer as outcome of dynamics, and what you are trying to see from that. And are you going to quantitatively information that you want to obtain? Then the equilibration phase is critical. Observe the stable temperature, kinetic energy, potential energy, and total energies. So the velocity scaling is used to readjust the systems if it's needed, and also the parameters if you're needed to be interested in equilibrium state. The production phase is actually produced, and there is no velocity scales permitted during data collection. All these things are considerations which we need to do it before running our molecular dynamic presentations. So you can have a tutorial which is a part of it, but however, I'm going to explain right now. So just you can have various tools, and just displaying the tools which is present in this Studio will help you to plot from a dynamics to the plotting of the graphs. Since I'm going to show you a demonstration, I'm not taking up this. So this is, we are trying to compare for the two different compounds, among them which one we have to choose for in vitro analysis. So we calculated the two systems. Of for example, I could say that this is done for PIK3CA that are bound with two different compounds. The root mean square deviations we are trying to see for an average amount of atoms towards the system. When you look into it, the compound one, it is trying to have an there is increasing and then decreasing and keep flowing it. But when I see into the compound two stability, at beginning there is an increase, and then when it keeps following on, it is having an fine. It is going through a better path. Similarly, I'm looking into the RMSF. So root mean square fluctuations. The root mean square fluctuations can be calculated for different kind of systems here. It's used to analyze the amino acid variations in either a backbone, or you can see it for your main chain as well as side chains. All three calculations can be done by using Discovery Studio. It will help you to understand where's a fluctuation of atoms is happening, or a transition of any secondary structures are happening or not by using root mean square fluctuations. And finally, the one of the most important parameter is the radius of gyration. Which is used to study the atoms as a function of time to ensure how the compactness of protein they entirely driven towards your protein dynamic synthesization, which also can be done. So these are very crucial, important parameters that we have it. And you can also have a C alpha plot for residual index also can be calculated. And then each and every step, if you want to capture the variations of in a Ramachandran plot, that also can be done. The respective changes in phi and psi angle of your dynamics. And you can animate and you can see the dynamics running. I will show you all these things. After analyzing, you can also get on a hydrogen bonding interaction based graph. It looks like a heat map actually. So having your compounds, and you can see here wherever you can see the green in color, that means that all the confirmation, this is the confirmation. So all the confirmations are trying to maintain its bonding till the end of the dynamics with the specific amino acid interaction. So almost 3 to 4. And wherever you don't see the green color or red in color, when you see a blue in color, that actually indicates there is a loss of bonding interactions. And apart from that, you can calculate the interaction energies, whether the interaction is being contributed more by van der Waals or by means of electrostatic, and total interaction energy also been calculated. Open the systems, open your output dynamics, and select your systems but then interface atoms, you can able to calculate the same. Then see, this is outcome of your conformations. You have interaction energies, van der Waals energies, and then electrostatic interaction energy. All three can be calculated. Then, of course, as the outcome, definitely we should know about MMPBSC and GBSC method. Both the calculation will be calculated by using this formula. Delta free binding energy is equals to GA, it is on the complex, minus GB of the ligand of the complex, and the remaining part of the complex. And finally, it will gives you the average delta G energy also. Individual frames also it will capture, and overall it's also captured. And you can also convert your conformations to molecules to calculate the RMSD deviations and to understand how the conformation various is happening as a function of time. You can convert the conformations into molecules. It moves like this. How the changes are happening. All these things you can able to capture over here. And finally, the single binding free energy will give you the information about your time, potential energy, along with that RMSD conformations, including complexes, ligand, and protein, and average delta G. So each and every frame also it calculates, and even for a whole complex is also captured. So major applications, so if you can study the protein stability, conformational changes, and then protein folding, molecular organization of DNA, RNA, and ion transportations in biology. Now we go for this demonstration. I will show you this. One second. Hope you can see my screen now. Now I'm sure. See, this is I have tried to take two different complexes. One is natural product, in fact, with the receptor binding domain of SARS-CoV-2. So I have two different complexes, so which is bound already, which is out results of C-Docker interaction energy. And I'm going for simulation. So run simulation, set up constraints, and change force field. So you can see here we have various different force fields. So I'm trying to apply the charm force field. So once I apply the charm force field, you can see all the atoms are attained. Similarly, I'm trying to do for the parallel system. You can run parallelly 2 to 3 complexes also. There is no restrictions here. You can run 2 to 3 complexes parallelly to perform your dynamics and simulation. So after applying, you can see the force field is applied. If it's not applied, you have this option here, more option, where you can edit it. So going back here under simulation. So under simulation of dynamics, we have energy minimization, force field, minimization process. You can do minimization. So smart minimizer will be always a default. Or you can choose different descent, conjugated or powell. So how many steps you want to perform, an RMS gradient, threshold, everything it is depends upon user. Along with that, if you want to apply shake constraint in this, you can apply. So I'm choosing Stephen descent and I'm trying this process. If you want to do individually, I can do this. Stephen descent. And then after doing the Stephen descents and everything, 2 steps of minimization, I can use that output as the input to a kitty. So here from a 2020 version onwards, we can run all the dynamics in the GPU mode. You can use either Intel GPU or you can use external GPU. Both options are available. So you can have heating steps, initial temperature, target temperature. And once you set up this and you can run. After that output, you can use that as an input for equilibration. So time step, how much step. You have to use the restart file. Don't forget to use your restart file. It's very important. You have to use the restart file to do the process. And then equilibration, how many steps you want to run, you have to decide. And the production, you can do either within a NAMD or within a CHARM production. We have a flexible systems in this. We have very large molecular systems or anything. You can choose either NAMD or you can go ahead with this CHARM-based dynamics. So first, we'll begin with always solvation, explicit periodic boundary condition, and you expand. We have a different cell shape, cubic, orthorhombic, and how many distance from a boundary you can select, and solve considerations you can decide. Or by default, it will be like a 0.145, or you can change a different cationic type and anionic type is also there. So I'm selecting this protein and running, and minimum distance, I'm trying to keep 3. All the values are expressed in terms of angstroms. Solvation, it's running now. Once it runs, like I added how many water molecules, and then how much sodium, how much ions are added, that also will be calculated here. And open that solvation, and then click here to view results, and how many water molecules are added, you can calculate here. Post-fill parameters is applied, and the solvated molecule. The solvated molecule of your systems, and you can label it, because I added label, that's what it looks like, blue in color in the background. So I removed that one. We have a chlorine, and then water, all these things. You can select and you can move like this. Either way. Are all are tip-3 water molecules? The 3-side, tip-3 is a 3-side water molecule. It's a part of discovery here. That is what we are using here. Okay. When you select water, you can see how much atoms are added. And once it is ready, that's what you can do by step-by-step process, or either you can use a standard dynamics cascade. Either way, you can process it. So if you want to perform, you have good GPUs, which is inbuilt, or you have external GPUs. So by using step-by-step process, it will run more than 10 folds faster compared to the CPU mode. So now I'm trying with the CPU mode. I'm using different distance, different distance 1000 steps, and then conjugate gradient, 100 steps, 1000 steps. Then the heating, I kept as such. And if you want to study want to set up your temperature, so run up to the equilibration and check for its changes, and then use that specific temperature to set up the process. That is most important part of your dynamics. Simulation, you can run up to, this is expressed in terms of picoseconds. If you want to run 1 nanosecond, increase to 10,000 means 10 nanoseconds. If it is a periodic boundary condition, automatically it goes to the type of NPT. Or if you don't want a periodic boundary condition phase, you can go for NVT and V. The respective, whatever the options that you're selecting, the respective protocols will be chosen over here. And based on that, electrostatics will be chosen. We have 2 different kinds of electrostatics, including a spherical cutoff, and then PM method, partial mesh Edward method, spherical cutoff for NVT, and NPT for a partial mesh Edward method. So which method you are choosing, you have to opt for it, or you keep automatic. And then we have integrated with the dynamics integrator, leapfrog wallet algorithm, and with the shake constraint to monitor your hydrogen bonding interactions. And if you're running in the grid or GPU, everything you can able to mention over here. So after giving the setup, everything you get going to run, just give a run. Same setup, I'm going to try it for my another molecule also, which will copy, and it's running, you can see. You can run multiple jobs, there is no restrictions. Okay. You can run multiple jobs, it starts from a step-by-step process. I'm closing the other ones. So you can see it starts with the minimization 1, 2, like that. Actually, I'm doing this in the CPU mode. You can, when you're running in a GPU mode, it will run till 10 times more faster actually. So in the step of heating, and then equilibration, and finally the production. This is how the step-by-step process will be carried out here. Then coming to the production, then it goes to steps 0%, 1%, 2% like that. Sometimes it will jump from a 10%. It depends upon the system computational cost. It will run very much faster out. Parallel, if you want to run another job, of course, you can do that. So there is no restrictions actually. You can run 2 to 3 dynamics parallelly, and you want to retrieve the results also, you can do that. So once it's already done, we will end up with so many file parameters, including trajectory, DSV, post-fill parameter, energy parameter. Suppose your system is got stuck and you are somewhere, some power short, and unusual things are happening. No need to worry about. We always capture the restart files. The restart file you can take up and you can begin your dynamics. From where you left, from that position, you can start your dynamics. And finally, you get a standard dynamics and CSV file, which captures how much starting position, what is the initial potential energy, final potential. Each and every step it will try to capture. I have run only for 10 picosecond in this. You can run for nanoseconds also. I will show you another one. You can click here to view results. So once it opens, you can see that. So this is the output. This is the molecule, molecule. I'm trying to give the different display style for that. See the interactions. And you can run. You can see the animations. How the bonding is changing, where it's interacting. Those things you can able to capture. Small steps also you can able to very well capture using this kind of networks. So for the whole systems, I'm changing display style. Okay. Then labeling, trying to choose stick, protein, changes of an atoms. And then binding of an atom. What the movements of each and everything you can capture very easily. The CTK model. This is CTK model. When I'm running it. How the atom changes are occurring. If a side chain is bonding and it is moving from position, that also you can monitor very well by stopping the position at this point animation, and you can see where is the variations are occurring. So that also you can capture very detailedly. You can very well see this. Now I'm keeping off protein to solid ribbon. I can customize. I can make it graphics. Either way, I can put a rainbow based structure changes. So this is how we'll be seeing our things after doing the dynamics interactively, and I'm more in a powerful interactors or a presentation mode. You can see your results. You can also capture this as a movie. You will have the options in the view where you can make it like a movie also, and you can export when you're going for presentations. You can play that movie. That in that way also you can do that. And when you're doing this one, and after doing this, if you can analyze your trajectory, analyzing trajectory, sum this output. So output should be must have confirmations. You can do pairwise RMSD also by comparing with all the frames, you can cluster your trajectories also. When you keep all frames, you can cluster. So I'm trying to take an input, which before running a dynamics, the static input I'm trying to take. I'm calculating RMSP, RMSF. If properties, if I want to check, initially I must monitor the ligand bonding interaction. I choose specify the frame, last frame, or within all frames. Flexibly, you can choose according to your wish. Fit trajectory, to which groups you want to fit it, hydrophobic or backbone to backbone, also you can do that. RMSD confirmations. And then you can run the process. So what always the Discovery Studio will try to do, it'll always capture your file and try to save your inputs along with your protocols and then your outputs. From the output, you'll get whatever the result. Actually, it is copying and running. So you can see it's analyzed trajectory started. And finally, you get a result like this. So either you get a graph, automatically it will try to generate a graph, or you can select, you can plot it off your own. So along with the RMSD graphs, where the interactions are happening, non-interactions are happening, everything it will try to capture and gives you an RMSF. Where the fluctuations are happening, when you select the specific region, that the 3 location also will be captured here. At whether it's happening in the loop or the helix or in the beta strand, everything it will try to capture. The ligand bond monitor will look like this. It's like a CSV file. If you want, you can put of your own of your heat map using the chart options. In the chart heat map menu, you can select a confirmation on Y and the rest of the things. X-axis. Select. Click OK. In this way, you can study this. To see wherever interactions are happening, it will be highlighted, and where there's no interactions, that it shows like a blue in color, or light interactions, smaller interactions will be in a green color. So we have an legend parameters to explain that. So closing that, you get a RMSD table along with the confirmations. All the values are expressed in terms of angstroms here. From each and every confirmation to be captured. Similarly, for an RMSF. So this is for main chain RM. Actually, this is for C alpha RMSF. We get it for main chain also separately, and side chain also will be there. Both the things will be there. Opening the results. So once they open, in this you can, I told you, we have 3 different types of RMSF. So you have it for your C alpha, main chain, as is for side chain. If you want to see the changes, you can put a plot here itself as a number of IDs items, as well as with your plots of various RMS. So you can select your name and then put together changes. If you want to look at an RMS of the alpha main chain, put together, you can have a graph. From this graph, you can see which atom or amino acid has variations in your main chain, as well as in side chain you can capture. So which has various fluctuations also you can able to capture from this graph. When you hover here, you can see the amino acids. And you just keep moving on the amino acids, will try to hover and show you the positions also. So that position, if you select, that will be automatically highlighted in the 3D window. So that way it feasibly help you to understand the fluctuations of the system. And also atomic positions, bond levels, also you can capture. And what force field type is applied, that also you can understand from this. And bonding, single bonding, what is the distance, and then the labeling, chains, residues, And then in groups, what are groups are there, how much confirmation. The confirmation tab is very important. You can export this in CSV files also. It will capture all the information and the references to RMSP deviation. And the radius of coloration, you can have it a tool menu and you can calculate. And ligand bond monitor, when I try to run this one, you can monitor and you can see the ligand bond changes. How many bonds are there? How many favorable bonds are happening that you can capture here. Similarly, when you're running, you can see the bonding variations. Not going to see here how much angle it is changing, what is the distance, category, types, that also you can capture. Similarly, in the confirmation tab, you wanted to calculate the radius of gyration. We are going to analyze from the tools menu option. Open this tab and keep it and analyze trajectory and give the radius of gyration. If you click the radius of gyration, automatically within 1 or 2 minutes, it will run behind and show you the results. It has done. So you get a radius of gyration. You can capture and then you can go hit the button. So this is how we are recalculating RMSD, RMSF, and RG values. Then as the outcome, if you want to calculate the interaction energy, so go to energy minimization, there you have interaction energy. Input your system and then select an atom. Atomic selections are very important. So interface groups, interface receptor atoms, to see the interaction between them. And then interface ligand atoms. Or if you have any residue selections also, you can do that. If you want some residues need to be, what is the interface between the residues interaction, if you want to select that also you can do that. Everything I kept to default and then run it. So once it runs, I will get the results. Closing all this one and trying to open this. So once I open, in that I will capture within the interaction energy. So van der Waals interaction energy, electrostatic energy, total interaction energy. Together, I will have the for each and every frames and snapshots that will try to capture and show. Similarly, I can use the same in output to generate for my free binding energy calculations also. If you want to run for your individual things, I can do convert confirmations to molecules. You can see the automatically it will try to superimpose, not superimposition, changes happening in the systems. I capture and I can calculate the free binding energy using MMPPSA method. All the input receptors must be my input, and then ligands all, and you can calculate the PPSA method. On the other hand, you can choose a single point energy transition, that is also part of it. So each and everything, just give your input. I'm trying to show them. You can see here it's running. Single point free energy, put your systems, and you can calculate GBSA as well as your PBSA method over here. Each and every frame, each whatever frames that is which is captured during dynamics will be processed. And finally, when you click open, it will calculate the average delta G. Average delta G for the whole systems. If you want it for a single confirmations, what is the average delta G and potential energy? That also can be captured here. From the confirmation tab, it adds another 3 columns to that. Complex for the ligand separately, complex separately, proteins for complex, protein. This is for ligand, this is for overall. So this is about our presentations for a molecular dynamic and simulation, and as well as an demonstration. Now I'm going to touch on a very simple topic of a QSAR. The QSAR, we are going to have a 15 to 20 minute presentation followed by a same kind of demonstration. I'm going to another topic, QSAR. I'm just sitting here and directly going to the slide. So very well known phenomenon that QSAR, QSPR. So when we come to this QSAR, we always try to answer two different questions. So what are the features of the molecule which affects activity, and what could be introduced to enhance this active property? So crucial part of the drug discovery process, I would say. So moreover, always the QSAR is a quantitative mathematical transformation technique. We are trying to have a correlation between mathematics and biology to understand how well the both are correlated and what are the parameters are correlated, so that we can understand the experimental property of a similar like compounds and to determine that individuality actually. So the condition goes always like this. Y is observation. It's a dependent variable or your activity value. It could be anything. It could, if you're working on a toxicity profiling, then that is called as a quantitative structure toxicity relationship, and which need to be correlated with various descriptors. Who all are called descriptors? A log P, all 2D parameters, 3D parameters, electronical, topological, NMR, dog score, fit value, pharmacophore features, whatever it is there, we call that as an independent parameters, which can be correlated, which also can be used for a correlation to reduce the noise and make a sense to have a better fit. That is how we are going to handle in a QSAR. Just history, I'm not reading this, just putting for your overview. So considerations, so minimum 20 to 30 compounds are very important. So we have to have a dependent variable. The strength of the QSAR, it depends upon your dependent variable. And then accuracy, and definitely there will be uncertainty will be there, but also we will be checking over here. And then independent variable is your choice. And then independent variables, if you're not choosing a proper independent variables, that results of poor QSAR model. QSAR models are very good to have, and developing a one such a model will take a, it is little bit, it is not hardest task, but we can do that. And then when you try to use this one overfitting, so what the people will try to do in order to have a best training set model, even this we are seeing in a QSAR and machine learning algorithms also, when I want to develop for some kind of AI-based models, try to take a good training set molecules. So when how we are training your training set molecules, it's very important, and that is a potential to identify your test set. High accuracy, that's what the Kubernetes paradox has been mentioned. The high accuracy of a training set will always lead to have a poor or low accuracy in test set molecules. To try to take a diverse set of compounds, the diverse set of compounds of various ranges of activity value will have a better efficacy of the models. Number of compounds, minimum 20 to 30. It depends upon which method that we are going to work it on. And the chemical diverse of compounds, that's why. So one is developing, say for instance, the person is developing only for chalcone derivatives of a specific model, and the other person wants to train with an maybe your glycoside tracks need to be trained with that model. So totally it is a contrast. We cannot have we cannot predict an better predicted activity value for that. Instead, there is a group of compounds having a diverse. One is having rich aromacity and lipophilicity, and then one is a palmitic acid, something like this. If you have a mixed diverse of compounds, that is very good to have a good QSAR models. So workflows of QSAR follows like this. First, start with the data preparation exploration, chemical structure preparations, dependent properties, descriptor analysis. Once you define the descriptors, divide your systems into training and test it. Various algorithms are there in the inbuilt in this, I'll show you. And finally build up to the QSAR model, do validation and analysis and prediction. Data preparation exploration.
Hello,
So chemical structure. Hello, sir.
I guess your slides are not moving. Where are you? What are you discussing now?
Yeah, 1 second. So let me check. Can you see right now, sir? Is it moving now?
Yes.
Can you confirm? Oh, yeah.
Please continue.
Thank you so much. Thank you. So data preparation exploration, chemical structure, dependent property analysis, descriptor analysis, training and test sets, QSAR model generation, validations and analysis and model predictions. The correct chemical representations are very important, which is the essential part of it. And then you can see what is the consideration of alignment, whether it's 2D or 3D, that we have to decide and try to have in a prepare your structure. That is what QSAR protocol says. Prepare your QS compounds before running your QSAR models. So chemical structure preparations we have to do. So we have a workflow-based models. So first we have to select a set of ligands, and then to create a QSAR model, it will say first prepare your ligands, input your ligands, and then generate it. Use that as the output. Dependent property, very crucial part of it. Which algorithm of QSAR that you are going to choose? Either you're going for a classification model or you're going for a continuous model. Based on that, you have to prepare your input system. Let me connect and tell you as I keep walking on. The dependent property, it needs to be predicted. Either you can put it in a binary way, binary in the sense, for instance, I have a set of compounds in my hand. In that, some compounds are less than 100 nanomolar or seems to be active, and greater than that or seems to be inactive. To be put it in a binary, I could say that is 0 or inactives, actives are 1. That is a binary classification. Classes. I can use this class-based model for random forest model. In that, what we can do, say for instance, instead of saying a binary 0 and 1, here I put it in a different way. Maybe actives as the A, moderately active as the MA, inactive as the I. I'm classifying as based on a classes. Or you can use a numeric or alphanumeric. So that totally depends on the which systems of a QSAR model you want to perform for which molecule. And your activity could be a linear or scale. The dependent property preparations. Input your systems. We have a various scaling methods in QSAR, right from the main center to scale to unit variance, that's 4. The unit range, 1 minus to 1, divide by mean, natural logarithmic, IC50 to IC50 conversions, millimolar, micromolar, and nanomolar. Input that one, and then prepare your dependent property. So you can have the scales like this also, -1, lowest score, 1 is the highest score, like this. Then descriptor analysis, very crucial. As I told, anything could be a descriptor. QSAR is not only A log P, acceptor, donor, molecular weight, that is not only descriptor. Descriptors of physiochemical properties or describe aspects of your chemical structure. And this is an independent property. It could be experimental, it could be a choice of descriptors. So Many QSAR methods are very, very sensitive to fewer descriptor selections. So we have to select the best descriptors to analyze the same. Independent property preparation, select the descriptors. We have the calculating molecular properties, select the molecular properties, run new work. So various descriptors in Discovery Studio, 1D, 2D, Cytec based descriptors or patent descriptors are part of Discovery Studio. 3-dimensional descriptors and other descriptors we have even LUMO, FOMO, based on Lee, Morphic2, and BAM3 settings based density function theory based models also be there. The 2D descriptors, right from A log P, we have various indexes also is there. 3-dimensional descriptors, dipole moment, juice, moment of inertia, surface area, volumes like this. Fingerprint descriptors, including Cytec extended connectivity fingerprints, daylight path, atom environment fingerprints, and MDL fingerprints. So what does that mean? EFC, E is the atom type, F is the functional group, L is the A log P, S is Sybil type, and then I is a prototype. And then C is in a functionals, and then E is the atom environment, E is the data item. And then output will be C will be the counts, and then F is the fingerprint, and M is the maximum border. The common fingerprints, we use the ECFCS or FCFPS. So extended will be based on the bits, and functional is based on the what is the acceptors, donors, aromatic like this. We can use other descriptors, as I mentioned. You can use semi-empirical quantum mechanics, or can use the density functional quantum mechanics. You can also include any imported properties of systems, and you can use pharmacokinetic scores, docking score function energies, pharmacophore fit values, non-border interactions also be candidate. And after that, so once you have a descriptors in your hand, you are trying to calculate within the principal component analysis. So all the parameters should need to be satisfied in a one specific column. So we try to make a first correlation statistical analysis. After doing that, we'll be doing a principal component analysis to understand the minimum variance to be there for each and every principal component analysis. And finally, deciding your descriptors. So these two parameters will help you to decide which descriptors you have to choose. So correlation statistics and principal component analysis. Principal component analysis main aim is to maintain a linear reduction of the data. Correlation, it will calculate all these things and you get a pairwise also to understand that deviations and all. In exploring your calculative properties, PC analysis for all the descriptors that you are given. And once you are done with that, we are going to select that a theoretical model must be validated internally. Either you can do internally or you can do test externally also. So the chemical space of the defined along with applicability we are trying to introduce. And then the objective of the QSAR is to predict the model predictability. So if you give us some compounds, it should have a good predictability to distinguish it also. So training and test set preparations, we have our different models, set up models in various percentages. Everything is automated. You don't need to do anything of your hand. Earlier, people do it with Excel sheets. No need to worry about it. Put your molecule systems and then choose the methods, how much percentage you want to define. And accordingly, you can get your fit up with the files, SD files. Use that file as a test and training. So we have these many models. Bayesian categorization, recursive partitioning, And then regression-based models, we have MLS method, multiple linear regression, partial least-square method, genetic function approximation, and 3D QSAR, molecular field analysis to understand the positive isosurface surface area and negative isosurface surface area. So when you want to choose a method, how we have to choose, which dependent variable we have to choose, that's what is explained here. So these things are based on continuous data, Bayesian is based on a binary classification, recursive partition is based on the multiple categorical data. So independent variable, if you handle the all, will handle the numerical data. Some is not handle the fingerprints. These two will not handle the fingerprints, and these three things will handle the fingerprints. And if you look into it, both it seems to be simple and easy to interpret, but prone to overfitting. And often, it's a better simple linear regression method. And GFA, it's a very good method, but however, there's having a prone to overfitting. Bayesian model, it could handle any large amount of data, runs very fast and easy to interpret. And recursive partitioning, you can do the multiple ways. You can use a single forest model or multi-forest model. So applicability, so any system that we are using, that it should be statistically validated. That's why we always try to include the applicability as one of the parameter. So anything the systems, if you have two descriptors which is present in this OPS, optimal prediction space, that is considered the ideal one. So good statistical model has the limitations. So prediction will not be equally good for all the samples. So that we'll try to give a similar compound set for training models. And then model applicability, MAD and OPS should be a trustworthy method. So anything outside, lying outside that one, it is not considered. So I'm trying to explain you only a GFA method. I'm trying to skip other methods. Just I'm concentrating on GFA method. So GFA method is evolutionary based method. So why the genetic is playing over? The genetic function approximation here is for the descriptors analysis. Some descriptors will try to move from generation to generation. Some will try to not travel after some period of time of generation. So that's why we are trying to use genetic function approximation methods. So algorithm goes like this. So first of all, you will choose and selected parents and do perform these mutations and try to add offspring equation to population. So parent 2 and crossover mutations. So it will try to have a crossover mutation. And then evaluate the fitness and to reduce the populations. And finally, it will choose the equations. So it goes like this. You have a multi-parameter optimization method here. Input your compounds and which model you want to build. And then what is the independent property. And additionally, linear with the linear sprints you can choose. Note because it will help you to understand a better way along with all the equations. And what is the population size? Maximum generations, we have a scoring method, single score method, and Pareto optimization method. Pareto optimization method is more accurate. And then the scoring function will be helpful to have a high possible accuracy with the complexity actually. Along with that, we are introducing scoring function. Either we can use a Friedman-Lachs or R square or adjust R square if that is. So these many scoring functions. So all these are statistical parameters which we are trying to include over here. So after giving input to test and training indicator results, either that's what you can do internal validation. If you can do internal validation, get a Q-squared value. If not, you can do external validation also. The variable counts, model descriptions, and everything. So I will explain you by showing you a demonstration because we are running out of time. I will show you the demonstration. That would be a better. Once. Yeah. So this is exactly the Discovery Studio looks. So we have a connect with the local server, tools, protocols, files like this. And I am trying to push. We have samples and you can access tools. And this is a whole big tool surface. And we have help menu and job options. Files, I already created a folder. Sniper, right click. Let us default. I have a data set. It's already divided into test and training. So I'm trying to use that one test set and training set. This is my training set. I can use this one, but I will take it from here. I have training set, which is included with an activity value. How do I include the activity value here? If you push this one, you can see the compounds. If I give control H, I can see the hierarchy. This is real-time data set. If you want to add any data to this, you have to right click here, add attribute, or maybe I want to add activity value, type activity, click OK. And either you can enter manually, or if you already had the values, you can copy paste. If I don't want to add this column, I can right click, I can remove attribute, activity. I have the training set data. After doing my test and training set preparation, I have my training set data in my hand. So go to the protocol, Discovery Studio, small molecules, QSAR. I'm running with the genetic function approximation. So here I'm trying to give the all molecules. Okay. And then I'm not using any test set. Let me show you. Underscore, I'll put like this, otherwise lots of models. Dependent property is IC50. Independent properties, I am trying to choose electronic state and then topological descriptors. Number of models, I'm trying to generate 10 different models. Additional terms, I'm choosing either quadratic or linear spins, GFE options, Pareto optimization method, scoring function, adjust R square, additional options, everything I kept as such. And then I'm trying to run for 5,000 generations. Keeping everything, just keep it. So if you're trying to see, you can see your number of generations it's trying to create. So it has reached to 1,000. It's a fast running protocol in Discovery Studio. QSAR is a topmost fast running protocol. 27 seconds. Yeah, that. Closing this one. Closing even this. Double click and open. You have a various files in this. Variable counts table. So whatever the counts, the generations will be over here. And then the counts. What are the parameters that you're given, independent parameters? All will be calculated over here. Model component will be in XC and it will be in a code file, SkyTech file actually. And then model description. So I have given 10 different models to be generated. For each 10 different models, it will try to calculate the equations section. You can see the equations. And adjust R square, R value, R square, R prediction, RMS of error. I think it will be very smaller letter. So it will try to form the equation. So R square must be higher. See that. And then each and every models and the respective variations, which one is taken, lower bound value, upper bound value, what is the P value coefficient, errors for 10 different models, it will try to calculate. And then training set informations, whatever we have given, how many variables we have given, what is standard deviations. And though we have given so many parameters of independent variables, some are excluded out actually. Which is excluded out, what is its reason? You can see it from here. It's not constant, too many zeros. So actually we have given user set is we have given these many parameters, including topological and electronic descriptors. But however, it will try to calculate it for a few things. It will tell you the reason also, why it is not calculated. And finally, we have a variable counts along with the graph. So we have a counts will be there. So you can see this. Now, finally, click here to view results. This is for your training set data. You have this. Now, I'm going to do external validation. Go to files. I have my test set and go to protocol. Small molecules, general purpose, calculate molecular properties. Here I'm selecting the all. And in other options, I'm trying to select the NIPER and then the applicability. Click OK. We have 10 different moles using that also. All molecules, tested molecules, and give it. It's done actually, finished. I'm closing this, double click and open. And you can calculate, you can bring a chart, point plot, okay, model, and then IC50, okay. Right click, trend line, select this. Here. So this is for your test set. This is for your training set. So both the values are more near 0.773 and then 0.76. In case if you don't have these values, the R squares are too much. One is 0.5, one is a 0.9. So then you have a problem with your test and training set divisions. So we have to select and there is also a problem with your you have to look on a descriptor properties also. So put together, you have to bring both are in the same platform. Those statistically validated parameters or a model you can use to predict the activity value for unknown compounds. This one we try to perform for an Hyena perform race, and you can do it for other things also. So thank you all. Closing this.
Okay, madam, thank you. That
Okay.
was right on time. I guess, are there any questions from the audience on YouTube? No, I guess, madam, when we have the software with us, we will explore and then probably students will have more questions for us.
Definitely, sir, definitely. And in meanwhile, if you have any queries also, feel free to reach us. We are happy to engage with you.
Thank you. Thank you once again for joining us.
Thank you, sir. Thank you.
So in the last, it is my pleasure and duty to present the vote of thanks. Are you able to see my screen? Okay. So with this, we come to an end of the 3-day workshop on advanced drug design and medicinal chemistry. It was organized by Department of Medicinal Chemistry at NIPER Guwahati, and it was supported by the Ministry of Electronics and Information Technology, METI, Government of India. So we are very grateful and thankful to them for their financial support. We would also like to thank our patron, our own director, Professor USN Murthy, Director NIPER Guwahati, and also chief guest in this workshop, Professor E.D. Jemis, ISC Bangalore, who in the beginning gave us good directions and gave a historical account of how quantum chemistry and semi-empirical methods evolved into the modern era. And now we are able to use machine learning with these tools, and I am sure the young generation was inspired by this his speech. Then Professor Sastry and Bhartam gave their thoughts about 3D thinking and importance of the open source tools, followed by Professor Coutinho's excellent lecture on the QSAR. Then Professor Raghava gave information about protein therapeutics and how it is evolving, followed by Professor Roy, who explained the QSAR details as well as variety of examples, and Prashant Kharkar gave useful information on variety of areas that they are working on. And today, Dr. Rathi presented a large how big pharma companies are coming together to solve difficult problems. And yesterday, Dr. Sam D. Weiser also mentioned about how quantum chemical methods are being used to understand the reactivity of metalloenzymes. And today, Dr. Majmudar explained the chemical biology and its importance in drug discovery. And also, Dr. Soni explained how protein-directed evolution and structure-based methods are helpful in discovering new reactivities of enzymes. Then also Professor Paul explained us how molecular dynamics is helping in understanding kidney disorders and getting some insights. Dr. Sirimulla explained about the applications of AI and how these are coming together to help variety of drug discovery projects. Then Professor Mandal also elaborated more on the applications of molecular dynamics methods, how they are useful in deriving the K on and K off parameters that gives us the KD values. These are expected to be very valuable in the near future. Then Professor Chakravarti also guided us in how green chemistry applications can be used in, and how medicinal chemists should think about all the organic chemistry reactions and how one can incorporate these in our drug design applications. Also, Dr. Dhakad explained basic concepts of molecular interactions. So with all these put together, I'm sure that the young minds who are watching this online, offline, have benefited, and we look forward to conducting a similar workshop next year on a larger, slightly larger scale. And this was not possible without the advice of our advisory board, and Dr. Radhakrishnananda, Dr. Tamilvanan, Dr. Banerjee, Dr. Naidu, and Dr. Hitesh Kulari. And I must mention all the members of organizing committee, faculty, staff, and students. They have worked hard behind the scene to make this event a great success. I once again thank all of them. And so we hope that you will join us in thanking all these people who have put efforts so that we could bring this successful workshop to you. Thank you once again. Goodbye. Have a great day.